and welcome to this year's Young Marine Biologist Summit. It's so wonderful to have you all here. This year, our theme, as you're going to find out, is Hidden Ocean, and we are going to be showcasing the incredible microscopic life forms, those things that we can't see with our bare eyes, but are so important for the health of the planet. We've got a fabulous lineup of speakers who we will begin in just a few minutes. I'm Helen Scales. I'm a marine biologist and a writer, and I am very pleased to be joined this year by Rory. Rory, hello, how are you?
I'm very well, Helen, and very delighted to be joining you, Dr. Scales, hosting the event today. Very exciting. So some of you, if you were here last year, might remember that I was on the careers panel and did a wee talk about albatross and other marine top predators. So I'm used to some of the bigger things of this world, but today I'm definitely celebrating the small, not quite microscopic, but I do have a sand eel hiding behind me here, which is
It's
lovely.
a double hide. I like that. I really like that you're hiding a sand eel behind you. It's fantastic, and I've got some fabulous small creatures behind me too. So we're going to be diving into the hidden realm, which is underpins all of this wonderful stuff in the ocean that we all are such big fans of. But first, before we get cracking, I think we would love to know a bit more about you guys at home. And starting off with a poll, we would love to hear where you are all tuning in from, because one of the wonderful things about this whole platform is that we can get you guys joining us, not just from the rooms where we all are scattered around here in the UK, but hopefully all around the world. So you should find a poll if you look either below you if you're on a phone, or I think to the side of the window, which I'm speaking to you right now. There should be a Q&A box, and I think there's a toggle at the top which says poll, and if you have a look at that, it will ask you where you're from. So pop in your location, your country, whatever kind of information you'd like to give us about your place that you're on. It would be great to see what we've got amongst all of you. So we'll let that run for a few minutes, and I must just say a huge, great, big thank you to the Save Our Seas Foundation, who are making all of this possible today. We're going to hear from some of them throughout the day. And also, we are making this whole event accessible as possible as we can. And you'll see that there are subtitles, hopefully, running underneath me. And we're also going to be recording all of the presentations. So if you find you want to need a bit more time to digest some of all the fantastic information we're going to be telling you, you'll be able to watch that all back. You can pause, you can read the slides, all that kind of stuff. Now, the recording is going to go up on YouTube after today, so you'll be able to look back at that. And if you're a member, then all of this content is going to be available as individual talks, and you're going to be able to go over to the My MBA parts of the website on the online platform, and all of that is going to be searchable. So basically, right now, you could just sit and enjoy all of the fantastic stuff we're going to be telling you, but you know that you can come back to it later if you want to dip in and remind yourself, write notes, write presentations for school, whatever it might be. It's all going to be there. So yes, now let's have a look. How is our poll going? I'm just going to toggle over to that. I'm also just, it's a little bit small on my screen, so I'm going to just make this a bit bigger so I can actually see what's going on. Excuse me. I should have got a nice big computer screen for this, but I didn't. Right, so we have got Europe, which seems to be the most popular place, but not the only place where you're coming from. We have got Asia, North America, Africa, South America, Antarctica 0%. Oh, sorry, no, it's all a bit small on my screen. We don't have anyone from Africa or from South America, which is a shame, or Australia, but that is fabulous. I know we've got some, I think we've got a few individual locations as well. People are tuning in from the Azores. Hello, Azores. People from Ireland. Hello, Ireland. Philippines, India. We've got Aberystwyth in Wales. Miami, Florida. Now, I am a bit jealous today. It's a beautiful sunny day here in England, but quite chilly. I imagine it's a bit nicer in Florida. We've got Bulgaria, Aberdeen. Hi, Aberdeen. We've got Somalia, Brazil. We've got France, Nigeria. That's fantastic. And some more Americans in New Jersey, Pakistan, fantastic. Sri Lanka, Austria, Alabama, back in the States, and New York as well. Trinidad and Tobago. And, oh, can I say this properly? Kissimmee, Florida. Hi. Thank you so much, everyone, for joining us. It's just so lovely to know that we've got a big international group of you all. It's probably, well, I think we're going to come to this. What time is it with some of you guys? I think that's also something I'd like to know.
We're going to get to that, yes. And well done, Helen, for that big, long list. I was hoping you were going to say good morning in every language, but we'll get on to that, right?
Yeah. I'll practice that in the break. Yeah.
So there's a poll up there just now, just for you to pop in what time of day it is where you are right now. So there's 6 a.m. to 10 a.m., 10 a.m. to 2 p.m., 2 p.m. to 6 p.m., 6 p.m. to 10 p.m., 10 p.m. to 2 a.m., and 2 a.m. to 6 a.m. And bless you, if 2 a.m. to 6 a.m. is when you're here, special award for anyone joining at that time. And thank you to everyone that submitted questions in advance for our speakers and presenters, because many of the talks today are going to be answering those questions, and others we're going to ask the presenters directly. And some we might even post the answers to in the chat, just depending on how timings are going. So thank you for those. And please keep them coming through the day, and we'll do our best to answer as many as we can. And just a wee reminder that you can find the Q&A box, I think, beside the video screen where we are if you're on a computer, or again, just below the video screen if you're on a mobile. And do remember to mention particular speakers if you're pointing a question in their direction. And you'll have to look for the program below this window if you want to see what you're in store for today. Again, look down there and see what you can see. And again, any questions, there's an FAQs document. So if it's a question about how the day is going to run, how things are working, you can see all that in the frequently asked questions section. So I think let's take a little look at the results of the poll just now. And my screen's slightly bigger, but I do wear specs like you, Helen, so I'm zooming in here.
Very close to our screen today.
It's looking like 10 a.m. to 2 p.m. is a very comprehensive winner right now, and who can be surprised given that this is the start of the day. But we do have a few 6 a.m. to 10 a.m. Shout out to them. We do have a few 10 p.m. to 2 a.m. So shout out there. We've also got 2 p.m. to 6 p.m. and a 2 a.m. to 6 a.m. Helen. So whoever that is, good on you. Amazing.
Seriously, you're doing better than we are. Thank you so much. That is absolutely stonking that you've got up in the middle of the night. Or perhaps you stayed up. I don't know. Maybe it's a big, I hope it's a party. I hope it's a young marine biologist summit party. I'm sure that's what's happening all around the world, which is just fantastic to know. And we've got so much coming up for you in the next few hours, the rest of the day, rest of the morning, the rest of the night, depending on where you are. I am particularly excited that we are going to have the return of the Sea Life Showdown. If you were with us last year, you will remember it was quite the battle as we decided which was the best species. We're going to be repeating that, I think, with some small creatures, I would imagine, to go with this year's theme of Hidden Ocean. So looking forward to that a bit later on. And hang around as well. We're going to have a fantastic careers panel. As Rory said, he was one of our fabulous careerists telling us about what they do in their work as a marine biologist. And I'm going to have a whole bunch more of fantastic people coming up later. So, you know, this is your chance to ask whatever questions you want to a whole bunch of people who are marine biologists of many different types. I think that's one thing that's, you know, quite a wonderful thing to explore, is all the different things that marine biology can lead you to do if you decide that this is something you want to do for the rest of your career or part of your career or whatever it might do to fit into your life. So yeah, the careers panel is going to be coming up later, and think of questions, whatever they might be, and we will pose them to our panel a bit later. But yeah, I think it's time we dived into the hidden ocean. Here we are. I'm enjoying being surrounded by all these wonderful creatures around me, and hopefully we'll find out some more about that. But first of all, I think what we should do is we should just hear some wonderful words from some very important people who are making all of this possible. First up, we have got a little pre-recorded piece from James Lee. Now, he is the Chief Executive Officer of the Save Our Seas Foundation, and they are a wonderful organisation. They are working all around the world on conservation education projects to basically help to create a sustainable future for sharks and rays. Now, James has a fabulous amount of experience as a marine biologist. He's a shark conservationist for over a decade. He has a PhD from the University of Plymouth. He is also working partly with the Evolutionary and Ecology Lab at the University of Cambridge. And I happen to be sitting right now in the University of Cambridge myself, although there are no sharks around at this point, which is a bit disappointing. But I think we should hear what James has to say for us. Say to us. Thanks, James.
Hello everyone, my name's James from the Save Our Seas Foundation, and welcome to the Young Marine Biologist Summit 2022, where this year it's all about hidden oceans. I think summits like this are just really important. They give us the chance to get together, the opportunity to share our knowledge, share our research, and combined collectively we can have a much greater impact for helping our oceans than we do alone. At the Save Our Seas Foundation, we support marine conservation projects all over the world, and all of these are focused on trying to make sure that these marine species, which are currently threatened through things like overfishing and climate change and habitat degradation, are here for generations to come. And a big part of that is just making sure that this biodiversity that helps keep our ecosystems functioning is conserved. One of the biggest parts of this is actually supporting early career researchers and people keen on getting into science like yourselves. And so a big part of what we do is providing opportunities for early career researchers and funding education opportunities so people like yourselves really make a difference for the future of all these threatened marine species. So if you really think about sort of how complicated ecosystems are, and yet most of us have focused on these really big charismatic species that draw a lot of attention, but there are so many aspects to every ecosystem, every marine ecosystem that sort of underpin that balance and that stability and that structure. And so much of that is within our hidden oceans that form the foundation of this. So one example I think is actually really cool is how there's this increasing power of genetics, using basically the instructions of our building blocks of life to figure out things about actually animal behavior we may not have previously realized. One of the cool things about genetics is we can use it to find hidden species that we didn't even know were separate from another one. Because of the way animals mix and breed, their genetics actually form structure, so they might be genetically different in one place versus another if they don't actually mix. And then that information you can use for, say, enforcement. If you have a trade where you have animal parts and products that you have no idea where they actually came from, you can take a tiny sample and all these hidden, invisible genes that you can sequence through fancy machines will then be able to tell you where this actually came from and help you in the enforcement of whatever restrictions might be in place for that animal. And before these genetic tools, that was completely hidden to conservationists, and so information like that is absolutely critical. So it's really because of all these different kinds of research and everyone coming together to understand, to get the most complete picture we can of all aspects of our oceans, that we really build the best picture that will help us conserve these animals for everyone for generations to come. I'm really delighted that you guys are all here to learn from all the fantastic speakers we have and to share ideas and to learn as much as we can about this sort of fascinating hidden ocean that underpins the whole of our marine ecosystems. Our passion for nurturing the next generation of marine scientists and conservationists is why it's been really important to us to partner with the Young Marine Biologists Summit. I think it's really great that it's no longer just this one event. You guys now have the opportunity to join the Young Marine Biologists Club, which will actually meet regularly throughout the year to have sort of more one-off sessions about particular topics. That's something you guys should definitely check out. Yeah, and in closing, I would just again like to say, welcome, thank you all for coming, and I really hope that you enjoy the Young Marine Biologists Summit 2022.
That was great. Thank you very much, James. And we're going to go straight into Dr. Jill Ryder, who's the president of the Marine Biological Association. She's had a varied career across the world and in lots of different industry sectors, and was awarded a CB in the 2011 Queen's Birthday Honours. So looking forward to hearing from Dr. Ryder.
Welcome to our 6th Young Marine Biologists Summit. It's a privilege once again to welcome you from all around the world. I must start with thanks to Save Our Seas Foundation for their continued support. This is their 2nd year partnering with the Young Marine Biologists Summit. Their support allows us to offer this event to you for free. Last year, we looked at some of the larger creatures of the sea. This year, we will be focusing on the smaller, but no less important, or awe-inspiring creatures of our hidden ocean. The teeniest creatures in our ocean, the plankton and microbes, underpin all life. Without them, we simply would not have our beautiful, diverse ocean. Earlier in the year, we shared some incredible facts about these awe-inspiring mini beasts of the ocean from our TikTok and Instagram accounts. Did you know that 1 teaspoon of seawater contains millions of microbes? Or that there are more algae, teeny microscopic plants, in the ocean than stars in the universe? Or even that there are certain types of plankton that emit a bright blue light? Today, thanks to some wonderful speakers, you will be able to learn even more about this hidden and mysterious world that many people will never have seen. We will introduce you to some of these beautiful and impressive creatures and hope you will find them as fascinating as we do. All around the world, we are seeing the impacts of climate change, with wildfires, sea level rises, and the loss of species. Now, more than ever, we need to learn about our ocean and what we can do to make a difference. The more we know, the more we care, and the more we are passionate to protect our blue planet. I hope you will be truly inspired today by all of our speakers, and that you will continue to learn and understand your ocean with our new monthly Young Marine Biologists Club. If you are not yet a member of the MBA, there will be information available throughout the day on how you can join. So please enjoy your conference, and remember, together we can make a difference and be the voice of marine biology.
Fantastic. It's always wonderful to hear from Dr. Ryder, and a great message there about us all coming together and being the voice for the oceans. We are all in it together. This is fantastic. Now, I think it's time we dived into our big, wonderful lineup of speakers who are going to be exploring with us today the hidden ocean, the hidden life of the ocean. I'm going to kick off actually with a pre-recorded piece from Professor Karen Lloyd. Now, she couldn't join us right now, but she's going to come along a bit later. So if there's anything about her presentation that you want to know more about, or any questions you've got, you can put them to her later on in the day. She's going to be talking to us about the mysterious life inside the Earth's ocean crust. She's from the University of Tennessee, and she studies microbes that live in really crazy, weird places, extreme environments like way down deep under the seafloor crust, in things like deep sea mud volcanoes and ancient permafrost. So let's see what Karen has to tell us about these extreme places and hidden life that exists there.
Hi, I'm Karen Lloyd. I'm an associate professor at the University of Tennessee, and I want to talk to you about mysterious life inside Earth. So have you ever wondered what life is like deep inside Earth's crust? If you have, then you're similar to me. I wonder about it every day. So that's what my research is to look and see what life is like inside Earth's crust. And if you think about, just taking what we know about life up at the surface and assuming that it's similar down there or different, that's not very satisfying. What we really need to do is actually dig down into the earth and go where the microbes are to discover what it is they're doing down there. But it's really hard to dig through rock. You can't just move through it very easily. So one way to get deep inside Earth is to go to the deep sea. So this is the Alvin submersible, which for the United States is our deepest diving scientific submersible. It can currently go down to 6,500 meters. I dove on it in 2008. This is myself and my advisor, Andreas Teska, getting on and launching. And this is what it looks like inside. There's lots of switches all around and a tiny porthole that you can sit in front of and look out all day. And what we discovered down there were lots of life, lots of giant worms that depend on chemicals instead of normal food to get by. So there were chemicals coming out of the deep earth that we were able to show through our research with the microbes are doing all kinds of things about methane. That's really what we're focused on. How does methane move through the deep earth? And in my more recent work, we're curious about what happens to this methane when it gets out into the water. Like, is it consumed by microbes immediately? How does it function? So for that, we go back to the oceans. So this is my graduate student who's carefully getting our equipment ready to toss over the back of a ship. This is one of our landers that has a laser on it to measure methane and oxygen continuously while pumping water all around. We drop this off the back of the ship. The person who's leading the dropping right here is the engineer who designed this, Howard Menlowitz. But being on research cruises is also quite fun. The nice thing is you get a gorgeous sunset every single day. So we would take sunset selfies almost every day. And we were also on board with an EK80, which is a really cool way to basically find bubbles in the ocean. So all the yellow at the bottom of this graph here are methane bubbles. And this instrument was so good that we could actually watch the line of our instruments dropping down to the bottom of the ocean. It's very, very fascinating stuff. And this is my students. Once we got our samples up on the shipboard, there was lots of work, lots of processing to do. So this is mostly what we end up doing on a cruise, is bringing in all our own equipment, setting up our own lab. And these are my 3 students working away. I had to get them to look up to take a picture of them because they were working so hard. And you try not to get sick, but sometimes you do, and that's okay. So it was a successful cruise. We had a great time. This is the full science party. We really just had an amazing number of things work. And I can't tell you about the science that came out of this yet because we just got back about a month ago. So I don't know what we're going to find just yet, but we're working on that back in our labs. But this is, going underwater is one way to look in the deep earth, but it would be nice to get inside that rock. So one thing we can do is we can wait for things to passively come back up to us in the form of hot springs. So this is looking at the side view of a subducting margin. So this is a oceanic plate that's getting buried underneath the continental plate. And this basically squeezes water that comes down and trickles down as rain and becomes ancient water that gets infused with all these deep gases. And it flushes back up through pressure differentials and makes hot springs. And as long as you do a couple of things to really designate what's recent stuff and what's very deep things, you can use this to look at the deep earth, to look at microbes in the deep earth. And so we drive around. When we do this work, we drive and get as many hot springs as we can. So this is in Chile in the Andes, looking at the hot springs there. This is in Costa Rica. Sometimes we have to hike through the jungle and find microbes. All these orange stuff right here are iron oxidizing Gallionella bacteria that are just fascinating to see in nature. This is hot springs in Iceland. This is me in the blue jacket here, perched in between 2 precarious fumaroles. You really have to watch where you step when you do this kind of work because there's fences to keep the tourists away, but we have to go beyond them with our permits and be very careful. We often do kind of crazy things, like we have to process our sampling in the car as we're driving to the next site. So my colleague Becca Tyne here was cracking up because it's really very awkward to have to sample your stuff inside the car. But sometimes we get to do our sample processing in quite nice places. So I'm sitting in Idaho in the western part of the United States doing sampling. It was quite lovely. And sometimes you catch an authentic moment where you're discovering data. So this is some of my gas geochemist colleagues who are looking at real-time data come off. And I managed to snap this picture before they saw me there. So those smiles are real. We get to go to really nice places to look for these hot springs. This is a site in Chile. Sometimes we go to really dirty places. So you kind of have to crawl in nasty holes sometimes and get crawled over by bugs and spiders and stuff, but it's okay. We live through it. And we get to see interesting animals along the way. And if sometimes we can even see volcanic eruptions. So this is in Iceland, and we're studying the hot springs around this. But let's just be quiet and listen to us laugh. We were just beside ourselves. It was so beautiful. I took this video. No telephoto. This is too hot for microbes to live. But the fact that this eruption is happening is changing the subsurface biosphere around the whole area. And that's what we were there to study. So this is me standing, that's how far away we were from the volcanic eruption. It was truly a memorable experience. We also go into active volcanoes to sample the living microbes that are in these sulfur lakes in the center. And this is Poas volcano in Costa Rica. And hiking down to it was like hiking through a moonscape. It was incredible. But it is quite dangerous because this is an eruption that happened shortly after we were down there. And we luckily were well out of there and away from it when it actually erupted. Otherwise, we would have not done well. From this work, we found very interesting things about how microbes are transforming carbon inside deep Earth that we wouldn't have found otherwise by using these passive sampling ports in the Earth. But not all parts inside Earth's crust are really hot. Some of them are permanently frozen, like permafrost in the Arctic. So this is our crew who went up this year in March to study permafrost. And I want to emphasize how international science is. Science often does not see borders as much as other disciplines do. And that's one thing I love about it, because we have to do stuff that's truly universal. So here we have folks from the United States, we have folks from the UK and Canada, and we have folks from Russia here, and we have folks from Nigeria as well. And we're all a team working together. It's kind of great. Science is like its own citizenship. And here's a picture of my students celebrating a successful drilling campaign in the Arctic. And these are the drills that we use. So this is me talking with my Russian colleague who is a master driller, and we drill into the frozen ground, and it's really just quite a stark landscape. It's really a beautiful time to go up there. So from this, we're learning about how microbes are affecting methane gas emissions, what's happening with the permafrost as it's thawing from climate change. It's changing rapidly, and so we have to move quickly and try to ask the best questions we can ask to try to answer the question of what are the feedbacks going to be from these deep subsurface microbes? Are they going to exacerbate the problems of climate change, or are they going to make it better? They could really go either way. So I hope that you learned something, or at least enjoyed me talking about my research. And if you are interested in living this kind of life and doing this kind of stuff, then I urge you to, study really hard. You get all this from hard work and really learning a lot about science. And I hope that you will, whether you choose to be a scientist or not, stay curious about everything in the natural world, because it's totally worth it. Thanks.
Hello, folks. Welcome back. And it is my distinct pleasure to introduce our next speaker, Michael Cunliffe. He's a senior research fellow and associate professor in marine microbiology and with the Marine Biological Association and the University of Plymouth, and has a particular focus on microbial biology and ecology, trying to better understand marine microbial life from the shores around Plymouth to the open oceans and way up, or indeed way down, to the polar regions. Michael is going to talk about marine yeasts. Did you know they existed? Well, now you do. And the adaptations they have developed to live in the challenging marine environment. So, Michael, over to you.
Thank you, Rory. Did you know they existed? That's my question before we start, really. Okay, thanks, Rory. So I'm going to talk about our work on marine yeasts. And like Rory said, some of you might have not even known that marine yeasts are out there. And we've been traveling the world, going to different parts of the ocean, trying to see if we can find these marine yeasts and understand more about them. Let me find my electronic clicker. So, does that work? Okay, great. Yeah, so yeasts. What are yeasts before we start to talk about marine yeasts? Well, they're basically very small single-celled fungi. And how small is very small? Well, about 5 micrometers in size. So if you're not used to hearing units like micrometers, 1 micrometer is a 1,000th of a millimeter. So very, very small. So of course, to look at these, we use microscopes. You might be familiar, when I say yeast, you might think of yeasts like baker's yeast, so yeast that you might use to make bread. But there are actually lots of different types of yeast, lots of different species of yeast that live in a range of different habitats. And yeasts are what we call osmotrophs. So that means they feed by absorbing the nutrients across the cell wall. So, as I said, yeasts in many different environments, including the marine environment. And this is one of the yeasts that we isolated recently from the shores near Plymouth. As with all organisms that live in the marine environment, from whales and dolphins down to the microbes that we're talking about today, the marine environment is a challenging place to live, and all organisms have to develop adaptations to live there. And we're particularly interested in the adaptations that these marine yeasts have to survive and to thrive in the marine ecosystem. So this is one of the yeasts that we isolated. This is a yeast called Naganitia. Naganitia we isolated from the North Atlantic Ocean. So this is a yeast that lives out in the open ocean, floating around in the water. It's part of the plankton. You can see an image in this micrograph above that what the yeast looks like. And then in the map below, this yeast in the global ocean. So these yeasts are everywhere in the ocean, or this one in particular, Naganitia. Living in the open ocean is actually a challenge for a lot of microbes because nutrients are very low. The reason why open ocean is so blue and clear is because the water is very nutrient poor. And you know, what adaptations do these marine yeasts have to survive there? And in our experiments with Naganitia, what we show is if we basically reduce the nutrients that Naganitia has, it can massively shrink down its size. So we see Naganitia easily reduced down by 30%. Why would a yeast, why would a plankton make itself smaller? If you're smaller, you basically grow much more efficiently. You need less nutrients and you can live in these low nutrient environments. Another yeast that we're interested in, and this is actually probably one of my favorites, it's a bit of a strange name. This is a yeast called Mechnia cowia. And we isolated Mechnia cowia from seawater near Plymouth. If you just look at this top picture of Mechnia cowia, you can see these needle-like specialized cells. So these are long, pointy cells, probably sharp. Why does Mechnia cowia have these pointed cells? Well, we think Mechnia cowia is probably a copepod killer. So copepods, really important group of invertebrates. I think David is going to talk about them in the next presentation. These copepods probably eat Mechnia cowia. Mechnia cowia goes into their guts because they have these sharp spores. They can peck the copepods and kill them. Really interested in Willow as part of our the Darwin Tree of Life project, and it's something that we want to work on more. So as well as going to places like locally around Plymouth and in the Atlantic, we also like to go to really foreign places as well. And we've been particularly interested in going into polar regions like the Arctic. So these are two people that I'm lucky enough to work with. This is Dr. Kimberly Bird and Dr. Berta Zanker. They both work at the MBA. And they went to the Central Arctic in 2021 to try and see if we could find some yeasts in this sea ice. And of course, if you're traveling in the Central Arctic on this sea ice, it's a challenging environment to be. And so to get on that ice, you need to go on an icebreaker. And you can see this is the icebreaker Odin, which is actually operated by Sweden. This is what Berta and Kim went on to travel on the sea ice. So this is Berta sampling the sea ice. You can see that she's sawing a piece of ice, and we were able to isolate yeasts even from this ice. And one of the yeasts that we isolated is called Captobasidium. This is a yeast that really likes it in cold temperatures. So it's something that we call psychophilic. This is cold-loving yeasts. And in fact, the sea ice where we sampled it. So how does this yeast survive in these cold conditions? They actually produce lots of lipids. So the yeasts are very fatty, and the lipids that they produce should be able to help them to survive. So I just want to wrap up, and just by wrapping up, I want to acknowledge a few people really. I've come and talked about the work that we do here at the MBA, but this is the work of lots of different people. So Poppy Diver is a PhD student at the MBA. She's worked on Naganitia, and Kimberly Bird and Berta Zanker are both doctors at the MBA. They went on the Arctic cruise and sampled the sea ice and were able to isolate these really interesting sea ice microbes. And Emily Cooper is another PhD student at the MBA, and Emily is the person that did the lipid analysis on this sea ice yeast. So I'll stop there, and I can ask any questions or anything that's in the chat.
Thanks very much, Michael. And we've got a few. Thank you very much, Michael. Blabbering over the slide that came at the end, the epic bit of noise that signals the end of your talk. So apologies there. I'll remember to wait for that next time. Michael, we've got some good questions coming in already. And the first one is a pretty important one. Finlay is asking, what are microbes?
Oh, good question for me. So microbes is a catch-all term, and it's for organisms mainly that are microscopic. So that means we have to see them with microscopes, so they're small, and usually they are single-celled. So small single-celled organisms that we probably have to use microscopes to see. And it's a huge diversity of organisms. So bacteria, these yeast that we've been talking about, single-celled algae, viruses, groups called protists, groups called archaea. So it's a huge group of diversity, much larger diversity than any other type of diversity. So there's more microbes than there are insects and fish and all other different things, but basically something that's small and something that's unicellular.
Brilliant. Thank you, Michael. And I think we've got time for one more. This came from Maz in India. What are the current and future trends in marine sciences? Where are things going?
Oh, that's a brilliant. Have we got several hours to chat about this, Rory? So one trend is what I'm interested in. I work on DNA-based tools to try and understand diversity. Because of sequencing technologies that have been made available, we can sequence these organisms that live in the sea, we can sequence their genomes, and this is giving us a completely new perspective on the biology and the ecology of marine organisms.
Brilliant. Thanks so much, Michael. Hope you all enjoyed that. I loved hearing a bit about marine yeasts. I got to say, it's new for me. I'm a marine biologist and I did not know very much about them. And I love copepods. So hearing that the copepod food is also reassuring for me. Helen, you are going to pick up and introduce our next speaker, I think.
Yes, absolutely. That was fantastic. Thank you so much for that first presentation. You've got us started off diving into our hidden ocean in an absolutely brilliant way. And it's now my great pleasure to introduce our next speaker, David Johns, who is the head of the Continuous Plankton Recorder Survey at the Marine Biological Association, which I think we're going to hear more about from your talk in a minute, David. But welcome. How long have you been working on plankton for?
A long time now, 25 years, I think.
Excellent. So you know a thing or two. What got you interested in the first place in working on this kind of thing? And one sneaky question, which I suspect kind of thing, if people ask me, I have no idea how to answer, but have you got a favourite type of plankton?
Oh, difficult one. So what got me interested? I think plankton are so varied, and you can see some of the images behind my head and behind your head, Helen, as well. They're just fascinating to look at. And despite, working in that area for 25 years, I never get bored of looking at them. I see, sometimes a common plankton, but it's just a nice view, and I'm still excited about it. As for a favourite one, I don't know. They're all pretty special. Copepods, as everybody's mentioned already, some of the copepods are incredible. There's some incredible facts about them, which I'll touch on in the talk.
Well, perhaps by the end of your talk, some of you guys at home might have some of your favourite plankton. So maybe you could share us some of your thoughts on that in the Q&A. But without further ado, I shall hand the screen over to you, David. Thanks very much.
Thank you, Helen. Hi, everybody. Thank you very much for joining us today. And thank you, everybody, for giving me this time to sort of have a chat with you all. So yes, today I'm going to talk a little bit about plankton. Small but mighty, as we've titled it. So let me just try my clicker, see if it works. Okay, so there's the same picture that's behind us, just some really nice images that we've collected of plankton. So I'll just start off with what are plankton? Well, the term comes from the Greek, meaning to drift. And generally, what we think about with plankton are they're the small plants and animals. I'll be talking about things in the sea, but you get them in fresh water as well. And they drift around, and they generally can't swim against currents, and they're generally fairly small. We can split them into two main groups, sort of the phytoplankton, the plants, which have chlorophyll in them. They're generally they're green, and they photosynthesize like plants on land. And then the zooplankton, which are the animals. And these are sort of vitally important for the marine ecosystem. And they're food for very many types of animals throughout the sort of whole food web. Some of them spend their whole life in the plankton, and we would call them holoplankton. And some of them actually settle out of the plankton into the sea floor and become things like crabs and lobsters and sea urchins and starfish, and we term them meroplankton. The plankton also includes the copepods, which we've heard mentioned here earlier. And there's this little arrow there pointing to one in this image. There's actually quite a few more in there. And an interesting fact about them is they are the most abundant multicellular or metazoan organism on the planet. So it sort of gives you an idea about why we think they're so important. Okay, let's move on to my next slide. Okay, so a bit more why they are so important. Well, if we think about the phytoplankton, the plants, they photosynthesize like plants on land, and therefore they produce oxygen. They probably produce around about 50% of the oxygen that that is produced on the planet, so a substantial amount. But also, like terrestrial plants, they draw down carbon dioxide out of the atmosphere, out of the sea, and then they can fix it, and they can basically transport that to the deep ocean when they sort of die and settle out. And we call that carbon sequestration. So they're incredibly important sort of about mitigating against CO2 level rises. We go back to my favorite, the copepod. Why is that so important? Well, it's just food for so many organisms in the sea. Cod, there's a picture of a cod here, fish, larval fish, adult fish like to eat it. Whales, basking sharks, whale sharks, all the large filter feeders, and even seabirds as well will sort of pick them off the surface. They like to eat them as well. So they play this key role in the sort of whole marine food web. But also as well, I mentioned earlier that some of them settle out to the sea floor into sort of crabs and lobsters and whatever. So they're very important to sort of fisheries for humans around the globe. There's a picture there of an edible crab, and there's also pictures of mussels. Both those organisms start their life in the plankton. Okay, so hopefully that's kind of convinced you that they're important. So we need to study them. So how can we study them? The oceans are massive; they cover most of the planet. You could, of course, just go down to the ocean and just drop a plankton net over the side, which actually I would recommend you do anyway, wherever you are, because you'll get some fascinating creatures coming up. But if you want to do it over a larger scale, you need something a little bit more, maybe a bit more industrial, I suppose. So Helen mentioned that I'm head of the Continuous Plankton Recorder Study survey, and we've been running this sort of global survey for many years, over 90 years in fact now. And that originally started by a guy called Alistair Hardy, who's a fisheries scientist in the UK, and he worked with herring fishermen in the North Sea. And they had noticed that depending on the color of the sea, weirdly enough, it would be whether or not they caught more fish or less fish. So he invented, you can see this was a flyer of the first thing he invented, called a plankton indicator, which they could tow behind their boats. And if it had plankton in it, they would know to cast their nets and catch fish. And if they didn't catch anything, they just wouldn't bother fishing. So we quite rapidly developed that to the device we use today, which is a continuous plankton recorder. You can see a little picture of it there. Now, this device is around about a meter long, probably weighs about 80 kilograms. It's a big metal beast with effectively a clockwork insides. And this is towed behind merchant vessels, what we would call sort of ships of opportunities, sort of like ferries and tankers, and they deploy it off the back of their vessel whilst they're on their regular routes through the world's oceans. I must say, we would not be able to sort of run this survey without the help of the sort of merchant shipping navy and portside staff. So they tow that behind. You can see there one just being collected. It's very robust. It flies underwater around about 6 to 9 meters. Plankton go in the front. They're captured on a silk mesh. and then it's sort of transported into the back of the device, and then where it's fixed with formalin. So it's biologically fixed. It's brought back to us in Plymouth, which is where we do most of our work. And then we have a team of people who look at those samples. So there's just a quick map there, which hopefully in a minute will rotate. Anyway, we collect samples over the whole world's oceans. Most of our work is in the North Atlantic and North Pacific, but we also go up into the Arctic. We've been down in Antarctica. We've got sister surveys all around the globe. And we have actually now towed over 7 million miles. And we've got a Guinness Book World Record for that, which we're quite impressed with. And just to put 7 million miles into perspective, that's 15 times to the moon and back. So it's a long way. And in that time, we've collected probably over 500,000 samples, and we've looked at around about 1/3 of a million of those samples. They've all been analysed for phytoplankton and zooplankton, and all of them are stored here in Plymouth, and we make them available for scientists to look at. As new ideas, like Michael mentioned things about marine yeast, which we wouldn't have thought about a few years ago. As these ideas come up, people can come to us and they can look at our samples. So I'm really hoping that there's some people listening in today who will have some sort of fantastic ideas in the future, and we'll be able to make our samples and share them with you guys. So we've got all those samples. Well, yeah, I forgot. There's my globe. It's moving around. You can see some of the samples. We can see there's lots of their different colours from our sister surveys, but you can see there's some big chunks of the oceans that we are still missing. So we're always trying to sort of fill those in. So we've collected all these samples. What have we learned? Well, most of you will be familiar with climate change, and climate change happens in the sea. Waters are warming, and because of that, in the plankton, we've noticed that a lot of plankton have moved in their distribution. They're tending to move polewards because the water's becoming slightly too warm for where they like to live. We've also noticed that new species have come into different areas where they never occurred before. And this can be because conditions have suddenly become ideal for them. Like I said, they shift northwards, or in some cases they're brought in by human activities, increased human activities. And in one case, we know that there's a plankton that has come around from the Pacific into the Atlantic due to reduced ice cover in the Arctic. And we've also seen changes through the season, so what we call the seasonality of the plankton, when they increase or when they bloom. Because of warming, this is sort of moving around. And all of these can have impacts for all the creatures that I mentioned earlier that really depend on them. I was just going to finish off with just a couple of facts, actually, which I thought were fairly amazing about some of the plankton that we look at. So we're going to go back to my favorite, the copepod. One of them in particular is called Calanus, and we get one very large, well, for us, very large Calanus. That's called Calanus hyperboreus. It's around about 1 centimeter long, which is really big for one of these guys. And they can actually live for 3 or 4 years. But the most amazing thing about them is they can go down several thousand meters in the winter, and they effectively hibernate. They go into a diapause. So they stay asleep down there. And in the following spring, they'll swim back to the surface just in time for all the plant plankton to appear so they can carry on feeding and developing. There's another type of plankton. We think of these as water fleas, Cladocerum. And I don't know if you can see in that, on the left-hand side is a big black dot, which is its eye. And the little arrows should hopefully point at other little black dots inside it, which are actually developing young. It has live young. It can reproduce asexually, and it can produce lots of little eggs, lots of little babies inside it, a bit like a little Russian doll, and they can all pop out. But if conditions suddenly get bad and there's not much food around, or temperature's bad, it can actually produce a resting egg, which you can see in the one underneath. This great big egg, which is very strong, almost like armored, and they can lay that, and that can stay in the sea, and it can actually put up with really bad adverse conditions. And then when things get right, out the baby can pop. And then the very last one, I just thought about, we mentioned this, coccolithophores. We could have hours or days just talking about coccolithophores, but this little guy here is 1/100 of a millimeter across. And if you can just make out the little plates on it, they're actually made of calcium carbonate or chalk, and they reflect sunlight. But these occur in such massive numbers, you can actually see them from space. The next image you can see, that's a satellite image just off the UK, and that sort of light greeny color is just a huge bloom of these little guys. Like I say, bearing in mind they're a hundredth of a millimeter, and we can see them from space, which is just mind-blowing. Okay, that's my super rapid go through plankton. There are my details there if anybody has any questions afterwards that I can't answer, and then there's a link to our CPR Circle website. So thank you very much.
Brilliant. Thank you so much, David. That was fantastic. It's such a fascinating way of understanding the ocean over such big scales, looking at such tiny things. And we've got a few minutes left before the break for some questions, which I'm dying to ask you from our lovely audience. So Ruben asks, what are the positive effects that plankton can have on the ocean?
Well, I suppose the positive effects are the fact that they actually feed so many organisms, so many creatures depend on them. They're vitally important food for pretty much everything you see in the sea. What I didn't touch on is they can actually have some bad effects. You can actually get some types of phytoplankton that can occur in very large numbers and produce toxins, and they can get into the shellfish and then can make people sick. But I didn't want to talk about anything negative today.
Well, on the topic of food, actually, this also leads us very nicely to Heidi's question. She's in Bulgaria. Hello, Heidi. Thank you for your question. She wants to know, how can plankton be better used as food for people? Is that something that's been thought about?
Yeah, that's a good question. It has been thought about, and it kind of has happened a little bit with krill in Antarctica and calanus from the copepods. You can extract oils from them to use in various things. But if you're asking me personally, I don't think it's a very good idea. These creatures are, like I said, they're critically important for the whole marine ecosystem, and we really do not want to be removing them. You know, we fish for the fish, there's climate change, there's pollution, there's lots of different impacts, and actually removing the food source of our marine life, to me, doesn't make much sense.
Yeah, I think that's certainly something that sounds quite concerning.
Mhm.
And I've got a couple more questions, which I'm going to wrap up in one, actually, because I think they're touching on similar things just before we let you go. Ruby wants to know how quickly do plankton reproduce? And we've also had another question, which is the lifespan of zooplankton. Do they live for very long? And then leading on from that, you know, based on how quickly they reproduce, how can there be enough of them for things like enormous whales to actually feed themselves?
Yeah, they can reproduce extremely rapidly. I don't have a figure off the top of my head, but sort of phytoplankton, they're your sort of classic single-celled organisms that can sort of divide, and then as long as they've got good conditions, they can sort of multiply exponentially very quickly. A lot of the zooplankton, they sort of have a life cycle of laying eggs and developing, but there's just so many in the sea. There's just billions and billions and billions of them. That's not to say that we shouldn't care about them or be worried about how things are changing, but yeah, there's a lot of them.
There's a lot of them, absolutely. I'm going to squeeze this one final one in as well, because I want to know the answer to this one. Why does the colour of the ocean change due to phytoplankton populations? I recently read, says our questioner, recently read an article about the colours changing due to phytoplankton changing. And you showed us that beautiful picture of coccolithophores being invisible from space. So yeah, how is that changing the colour of the ocean?
Yeah, the sort of phytoplankton, because they're generally green, once they sort of reach a certain abundance in the sea sort of changes, you know, from a blue color to a sort of a greeny blue color. And you can think about, when we think of sort of tropical oceans that are that beautiful clear blue, that's almost like the desert color for plankton. There's not much in the way of phytoplankton there, and all the zooplankton are very small. And then if you come to the UK, if you come here around spring, the water is green. It's greeny blue. You can hardly see through it if you're diving. And if you were to take a little plankton sample there, it would just be absolutely full of so many creatures. So yeah, it's basically based on the density of them. There's just so many that they change the color.
Fantastic. Well, David, thank you so much for introducing us to your work and the plankton recorder and all the wonders of these tiny creatures that live in the ocean. If you have any more questions, I think David very kindly gave his information, so you can get in touch and ask those. But thanks for all of those we've already answered. And thank you for bringing us into your particular area of research. That's fantastic. Thank you.
Thank you very much.
So Rory, I think we're due a bit of a break. I think we should let you guys go and grab a cup of tea, stretch your legs. But what have we got coming up?
Oh, we've got lots of exciting stuff coming up, and it's already been pretty exciting for me, I have to say. I am a big fangirl when it comes to the continuous plankton recorder. I did not know that you could get so much joy out of a sort of metal box, but it's dead important. And I'm glad I've got this sand eel behind me as well, actually, because copepods are really important in the diet of this little fish, which is super important in the diet of a bunch of the big marine predators that David talked about. So this information we get from the CPR is like the foundation of everything we know about marine food webs, especially in the Northeast Atlantic. So yes, I was very excited to see that presentation, and thank you to David for overseeing that awesome work. Yes, so yeah, as you say, Helen, we're going to take a short break now. The next session is going to be starting at 11:10. Our time here in the UK, wherever you are, it will be 10 past that hour, the next hour that we're coming up for. The session is going to explore seaweed, corals, diatoms, and hydrothermal vents, one of the things that first got me into marine biology. And we're going to be hearing from Mike Allen, Cher Chow, Colleen Kavanagh, and Yasmin Mida. So this is looking like a brilliant session. I'm going to take a little break, and Helen, you are very much in charge of the controls. So time for a break, grab a wee cup of tea, a drink, pop to the toilet, whatever you need to do, and we'll see you back here at 11:10.
I'm Sydney Harned, and I'm a graduate student at Nova Southeastern University's Save Our Seas Foundation Shark Research Center and Guy Harvey Research Institute. I am studying the genetics of scalloped hammerhead sharks in the Eastern Tropical Pacific, including the Galapagos Islands. Specifically, the Galapagos scalloped hammerheads are under tremendous threat from the huge amount of illegal fishing that occurs here. Hammerheads in the Galapagos form large schools of hundreds of adult female sharks, and people come from all over the world to dive with them. Unfortunately, these sharks are also a target for huge fishing fleets stationed right outside of the Galapagos Marine Reserve. Hammerhead populations have been declining rapidly in the past few decades, so it's imperative that we protect the habitats that these sharks frequent. But in order to do that, we need to understand their habitat use. That's where genetics comes in. I am investigating the genetic connection between the Galapagos adult hammerheads and hammerhead nursery sites along the Central and South American mainland coast. Nursery sites are shallow coastal habitats where pregnant sharks go to give birth and where their babies remain for the first few years of their life. By taking genetic information from adult sharks in the Galapagos and babies along the coast, I can build a map of where these sharks go and how they are connected. This can help answer important questions like, are the adult females from the Galapagos aggregation traveling to coastal nursery areas to give birth? We can also use genetics to investigate familial relationships and genetic similarities between these locations and scalloped hammerheads from other parts of the world. By answering these questions, I will have a better understanding of how this critically endangered shark species uses habitat in the Eastern Tropical Pacific, which will hopefully lead to more protections on these areas.
Sharks and rays, they are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
There are legends of the deep. Ancient and elusive, they are the keepers of balance in our oceans. Hammer. Angel. Great white. Tiger. Manta. Refined over millions of years, their powers are unique. They are super sharks. Together, they ensure the stability of our world's life support. The oceans. Our oceans need super sharks. We need them, and they need our help. Assemble the super sharks and help them save our seas.
Welcome back, everybody. I hope you had a good break, that you've got yourself some tea, some water, you've stretched your legs and you're ready to go for our lovely next session for the Hidden Ocean here at the Young Marine Biologist Summit at the Marine Biological Association. And big thanks again to our sponsors, to Save Our Seas Foundation, who are making all of this wonderful day possible and making it accessible to all of you guys coming to us from all around the world. Don't forget that we want to hear all of your questions. So you've got the chat function. So drop in any questions there you want. Do remember to use the @ symbol if you want to request questions for particular speakers. And we will get those all asked as we bring each of our speakers up in these next sessions. We're going to run another poll as well. We would like to know some more things about you and how you are watching this summit. Where are you in terms of who's around you? Are you on your own like I am? I am in a very big room all on my own. Are you with friends? Are you with family? Have you got a party of people together? Tell us about your summit experience. So you should have the poll should be there if you go to the, I think, the chat function to the side, and you've also got one that says poll. Click on that. Tell us what you're up to. Scroll down. I think if you're looking at us on your phone or on a tablet, and it should be able to find that. So do get in touch and let us know what you're up to, and we can get a better sense of this wonderful virtual audience that we're collecting all around the world, across this wonderful blue planet of ours. And while you're doing that, I'm going to have a quick chat with a very important person who has been working brilliantly behind the scenes with all of this summit, and also does some fantastic other work too, for the Marine Biological Association. That is Lizzie. Lizzie, hello.
Hi, Helen.
If we bring you on screen, there you are, we can all see you. Hi, your lovely face, thank you so much. Now, well, we want to have a chat to you about the Young Marine Biologist Club, because we've had some questions coming in already about how can I get into marine biology at a young age, and this seemed like the perfect way to bring you on screen and have a chat about the club. So what's this all about?
Yeah, so we started this Young Marine Biology Club back in July. And it's for our young marine biology members of the MBA. And essentially what it is, it's a kind of session where we bring in a speaker on a different topic. So we've had speakers from the Darwin Tree of Life project, as well as Cordelia, who is also here today. It's very exciting. And so we have kind of a presentation from them, and then we have a whole bit about their career, and to really kind of give our young marine biologists a chance to ask questions about how they can get into marine biology. And so this is a monthly club, and it's free to our members. And we have 3 really exciting clubs coming up in the next few months.
Ooh. Can you tell us any more about them, or do you is that a big secret?
No, so they're all live currently. So if you scroll down to the bottom of your viewing page, you can see the next 3 events. And there's also information about how to join our membership if you want to join these clubs. So we have the great Professor John Spicer joining us in December. And he's going to talk to us all about his work working in Antarctica, which is really exciting. And then in January, we've got Alan Hesse, who's going to be doing a really cool workshop on how you can work with science communication in kind of illustration and comic book style. So it's a workshop on how you can create your own 1-2 page comic book all about science communication, which I'm really excited about.
That sounds fantastic. I think I'm going to have to come to that one. I love the idea of making comic books. Yeah, definitely sign me up. Can I come? I'm not very young anymore though. Fantastic. And there's one more. Is there one more coming up as well?
Yes. So we have Aaron Brazer from CFAF, who is going to be talking to us about how he got into becoming a fishery scientist. So that's really exciting.
Fantastic. Brilliant, brilliant. So it's all about showing you guys what marine biology can be, what kind of things, what different paths it can take people down, which is just fantastic. Because I mean, I remember when I first got interested in the ocean at quite a young age, I didn't have any idea of all of the different sorts of paths that you could take, and even had no idea what I would end up doing what I do, writing books and talking to people, doing things like this. So it's just really lovely to have that opportunity to see just how diverse marine biology can be in all different ways in our lives. This is a really wonderful thing.
Yeah, definitely.
Fantastic. Thank you so much, Lizzie. I'll let you go back behind the scenes to carry on making all of this work so brilliantly. But thanks so much for talking to us
thank
about
you so
the club.
much, Helen.
Fantastic. Brilliant, brilliant stuff. Now, how is our poll getting on? Have we got any results so far in terms of, have you been telling us all about where you guys are watching this from? I'll just let that spool up on screen for a couple of seconds. Oh, there we go. Let me just change again. I've still got my issue with my tiny screen in front of me. So here we go. We have got quite a lot of you on your own. So am I. So I feel that we are all here together. So that is what this is about. And I do love that by the means of the magic, the technologies that we have, we can all be talking to each other and seeing each other today. So none of us are on our own. We are all one part of a big family. But 18% of you are with your family at home. So hello, families. Thank you for being here too. This is all very important. Some of you are with friends, and a few of you are with large groups of people, which I just also love the idea that we've got this big global party. All in aid of the oceans and the hidden oceans, and this wonderful day when we can all share our nerdiness and our fascination with this wonderful place that is the ocean around us. So thank you so much for sharing those, your whereabouts and what you're up to in our poll. You can if you want to know more about what we've got coming up the rest of the day, the program is still, I think, below me or to the side of me, one of the sides. You should be able to see what's happening. Get your questions, keep them coming in for our speakers. As you'll see, and I'm sure, as I mentioned earlier, I'm very excited about the Sea Life Showdown that's coming up later on, and we're going to find out which is the best species this year, and you're all going to be voting on that, hopefully. And again, our mini careers panel, again, this idea of helping you to understand a bit more about what marine biology is, what you can do with it, your chance to ask whatever you want to a whole panel of fabulous marine biologists who are themselves doing a whole array of different things. And we can get to that later in the day. So without further ado, let's crack on with our next session of fabulous speakers. We are diving this session into the world. Well, beginning off with seaweeds. Now, I have some sad news. Unfortunately, Mike Allen, who was supposed to be coming to join us, couldn't make it. But we have an intrepid Duncan Smallman has jumped in to be replacement Mike. Mike's not feeling too well, but we have whipped in his place someone who's going to do a fabulous job of telling us about seaweed and seaweed generation. Mike, who can't make it, is a professor at the University of Exeter, but Duncan is here to take his place. Hello, Duncan. Thank you so much for stepping into the breach.
Hello.
Hello.
Hi, Helen. Hello, everyone. It's honoured and delighted to be able to represent Mike. I just don't have a sombrero, so, and probably not quite as cheerful as Mike usually is, but yeah, sadly he has no voice at the moment and is curled up in bed.
Oh, Mike, get better soon. We're very sorry to hear that. We do hope you are back on your feet and talking very soon. But Duncan, we are thrilled to have you with us. I think best if we just throw over to you, and then we'll have hopefully some questions at the end. So thank you so much.
Yes, so I have gone through, as you can see, it says it's with Professor Mike Allen. Unfortunately, you've got Dr. Duncan Smallman, and we're going to be talking a bit about more problematic seaweeds, because you might remember from some, well, actually this was quite a long time ago, wasn't it? The Beijing Olympics. And maybe also in the coast around you might remember you sometimes get green seaweeds washing up that wash up onto the beaches and they cause a big stink. But the one that we're focused on is something called sargassum, and that has been washing up in West Africa, in the Caribbean, Gulf of Mexico, in fact a lot of places in the tropical Atlantic. And so today I'm going to be talking a bit about our work there. So wonderful. I do apologize. Today is also a very important day where you will learn the lesson of what proper preparation that means. So I do apologize if I stumble here a bit. So Mike has spent over 20 years of his life in research looking at microalgae and how to use like phytoplankton, and also how the last 8 years or so figuring out ways of using this problematic sargassum, which started washing up in various parts of the Gulf of Mexico and the Caribbean in around about 2011. So he's this is just a slide talking about bits of where he has been looking at fertilizer, looking at kind of turning it into a biocrude, so like crude oil, and using it as a fuel. But this is this hasn't quite worked. Sargassum is a major problem. It's a genus of brown seaweed. Seaweeds are broadly grouped into 3 groups. So you get, and they're all based on colors. And humans, we like to put things in boxes, and the Victorians were brilliant at it. So they were broadly put into browns, reds, and green seaweeds. And red seaweeds and green seaweeds are sort of plants. Brown seaweeds, not at all related. They're about as closely related to red and green seaweeds as we are related to red and green seaweeds. I find that really fascinating. And for any of our women, wannabe women scientists out there, a lot of the best phycologists and people who study seaweed are called phycologists were actually women. So some of the best drawings, some of the best diagrams were actually that were done in the early days of phycology were actually done by female scientists. So next slide. Yeah, so sargassum is this shows where the current sargassum belt is. There are 2 areas that sargassum is found. So naturally it occurs in the Sargasso Sea, and then this new belt where along the tropical Atlantic is called the Great Sargassum Belt, and it's kind of originated in roughly about 2010, 2011, and it's purely a human man-made issue. I think that's what the next slide talks about, but yes, it stems from the, oh no, the next slide doesn't talk about that. But it stems from the Atlantic, and it goes from the Atlantic to, it's carried over to like the equatorial currents take it into the Caribbean and into the Gulf of Mexico, and it just washes up. And then this year there was an estimated 20 million tons of it washing up. It also washes up into West Africa, so it chokes ecosystems. It kills a lot of animals. Sorry, this is going to sound really gloomy, but if you like sea turtles, sargassum is not your friend on this front, sadly. Yeah, so we will go to the next slide. So we kind of looking at sargassum in ways of being able to sink it for removing carbon dioxide, because the oceans themselves, as you're probably familiar, that we're facing issues with ocean acidification, and that's because this water, just the seawater itself, just passively takes up large quantities of carbon dioxide. About approximately 900 gigatons of carbon per year is taken up by the oceans, and for many, the atmospheric carbon dioxide and surface carbon dioxide, surface ocean carbon dioxide are kind of roughly the same carbon pool. They're exchanging all the time, and the oceans are some people say about 30%. It's kind of closer to 40% of our carbon emissions are taken up by the seas every year. And so this is all due to our use of fossil fuels. Okay, let's go to next slide. I do apologize if there's a bit of background noise. There are little people running around. So the sargassum is due to nutrient runoff, soil erosion, untreated sewage, basically poor farming practices in around the Amazon basin, the Orinoco River basins, and the Congo River basins. And it's just large amounts of nutrients going into the seas and then causing this huge boom. Kind of what the thought is that the climate has changed enough that it caused a weakening in the North Atlantic currents. And so some of the sargassum that was in the Sargasso Sea sort of was shipped around the Atlantic and went down into the equatorial. Some of it just survived into this kind of suddenly finding this really band of high nutrients, and then the currents just took it in, and as soon as it hits the coastal waters, especially around the Amazon, it gets everything it needs, and this sargassum just goes booms. So it's growing, and it's taking up the carbon dioxide in the surface waters, and the IPCC recently put out their projections that we actually need to remove carbon dioxide. We don't just need to cut our emissions and stop using fossil fuels. We need to remove about 10 billion tons of carbon dioxide a year. And so we figured, well, there's a problem. that is going to affect habitats, really important habitats like mangroves and seagrass beds. I'm really partial to seagrass beds. Seagrass are amazing. They're like 35% more efficient at carbon dioxide removal, like fixing carbon, than trees. So I'm a big fan of everything in the sea. Trees are, you know, they're just there. I mean, they're very cool, but let's face it, that this is the Marine Biology Association. I'm going to say the sea is the best. So anyway, so yeah, the seaweed absorbs CO2, and it's potentially better at absorbing CO2 than rainforests. So we go on to the next slide. What our solution is, we're going to take the sargassum. Right, there we go. And we're going to sink it to depths of below 2,000 meters, because the deep ocean is the largest carbon sink on Earth. In fact, marine sediments are some of the biggest carbon sinks, but just the deep ocean itself contains 30 trillion tons of carbon, and adding everything that we've produced in industrial times in the last 270 years or so would only increase 2% of the deep ocean carbon. So yes, it is essentially putting a seaweed problem up here into the deep ocean, but everything is a lot slower. It's essentially, we're speeding up the long carbon sink. If you think where oil and coal comes from, coal is fossilized plants from many hundreds of millions of years ago, and oil is basically fossilized marine life from hundreds of millions of years ago. And that is everything that's sunk down previously and just been compressed and buried. And that is essentially what we're looking at with the sargassum. It's just crazy to think how much the deep ocean could take, and we probably can do it without a high impact. So next slide. All we need to do is is that tons of sargassum shore, if it ever gets near the coast, and sink it into deep water and leave it for all eternity. But obviously, I do apologize for my internet, it's a bit unstable. Obviously, there are certain challenges we face. We've faced the engineering, political, regulatory, economic challenges, and because we are sort of talking about geoengineering, more so than we've done already, but this time sort of more focused, deliberate, and monitored geoengineering than what we did with the Industrial Revolution, which was just a wee bit, well, let's just go ahead. So the next slide is how we're going to do this. And essentially, we're building a robot. We're calling it the Algaray, and we like to think of it as a mix between a Roomba and a Pac-Man that comes along and munches up the automatically comes along, munches up the sargassum, and then we lower the Algaray, and it sort of goes down into the water. So at the moment, we're going to be using this kind of robotic. The plan is to use a catamaran like this, fully solar powered, fully solar powered, fully remote controlled, and eventually automated to run through the sargassum mats, and then the rays are lowered by a winch into the deep, and we drop the sargassum like that. Eventually, the rays will look something like the GUP-D from Octonauts, and it will just sort of tootle around doing it its own way. And the next slide, hopefully, is a video to show that we've sort of made a start. It's flipping. So on one side, on the left side, is the ray doing its flip. On the other side, Mike told me that there was meant to be a video of where we could actually see it being towed, but we have got it working. We've got the ray working, and we've even got the remote control catamaran working at the moment. So thumbs up, we're getting in the right direction. Where we are operating is in the kind of the next slide. We're actually going to be in Antigua and Barbuda, but you can see that this is kind of the political landscape. And Antigua Barbuda has quite a large exclusive economic zone. So this map is just showing the waters that belong to the different countries. And you can see the number of different places that are impacted by sargassum. Next slide. I probably have overladen you with too much information and probably gone on a bit too long. So if you are interested in what we're doing, we've got a website. We're at seaweedgeneration.com. We've got a whole too much information page, which is basically our frequently asked questions. And you know, Seaweed Generation we were only founded in 2021. So we've only actually really been around for a year. Mike and I founded it. We're brought together by our CEO, Patricia, or Paddy. And yeah, so we've only been developing this for the last year or so, and it's been a very exciting journey. So yes, and I never properly introduced myself. I did actually study marine biology. I did it because I was wanting to study sharks, and I ended up studying parasites of salmon. So I would recommend you find your favorite marine parasite because they're really fascinating. And then I ended up in seaweed, and yeah, I've spent 10 years in the seaweed industry, finding other uses for different seaweeds. So anyway, next slide. Thank you very much for your time. These are some of the contact information. This is all for Mike. We are on Twitter at Seaweed Generation. Mike is that, and my Twitter handle is at CPT_liceboy, Captain_liceboy, if you like, because I study sea lice. So thank you very much for your time, and I hope that was all right.
That was fabulous. Thank you so much, Duncan. It is so hard to step in when you're at the last minute. You did a fabulous job introducing your wonderful work. So interesting. And we have got some questions if we can keep you for a couple more minutes. I like this one because, again, I get asked it about all sorts of things in the sea and I never know quite what to answer. But what's your favorite type of seaweed?
My favorite well, to eat is nori, is larva, which is Porphyra and Pyropia. It's a red seaweed. It's a lovely, beautiful sheet. But purely on aesthetics is probably something called sea beach, Delaceria sanguinea. It's beautiful. And the other one I really like eating is Dulse, Palmaria palmata.
Fantastic. And that actually answers the next question, which is brilliant, partly, which is what type of seaweed is the ones that you can eat? So there are lots of seaweeds we can eat, aren't there? A lot of them are edible.
Yes. So that's the other problem with sargassum. You can't really eat it because it's got incredibly high inorganic arsenic content. So it's a good way of poisoning people if you really want to. But sorry, I shouldn't encourage that. In the UK, all seaweeds are edible. Some are just tastier than others. So it's not like mushrooms where you're going to do yourself, or even land plants where you're going to do yourself a mischief if you misidentify anything. It's just you've got different things. They've all got different flavors. So yeah, you're fine to go out nibbling.
Fantastic. That sounds great. Question from Sophie is, do red seaweeds contain chloroplasts?
Yes.
There we go. Nice and easy. But it's fascinating what you were saying about how differently, how much this is a big group of different organisms that we call seaweeds. It isn't just one thing by any means.
No, they are. So they've all got different. So yes, they all contain chloroplasts. So red seaweeds are kind of considered a very ancient group of plants. So whether they're within the plant kingdom or not is not totally sure. That's kind of like a taxonomic issue that might or might not be important. But so the green seaweeds are really closely related to plants. So they're kind of within the plant kingdom, but the reds are sort of their own thing. But the reds have there's just different types of chlorophyll, and the reds have and let me get this right, chlorophyll A and chlorophyll B. The greens have chlorophyll A, and the browns use chlorophyll A and chlorophyll C. But the reds do have these secondary pigments that help with protecting them, or the light, with light capture. So they've got some really funky kind of protein pigments called phycobilins, and they're they can be beautiful. Like some of them will give a blue iridescence. A really nice way of identifying something like Irish moss, which is Chondrus crispus, is it'll have a blue iridescence at certain times of year in the rock pools, and it's just a brilliant blue. So, you know, you can actually sort of get blue seaweeds if you know how to do it.
Those ones are absolutely beautiful. I was definitely enjoying looking at seaweeds over the summer in France and seeing all different colours, and that iridescence on Chondrus crispus is absolutely gorgeous. Duncan, thank you so much for joining us. You've done an absolutely stellar job of stepping into the breach. We wish Mike all the very best. Hope he feels better soon, and best of luck with your work. We look forward to hearing how that all goes and whether you figure out how to deal with this issue of Sargassuma, perhaps turning it towards helping the planet as a whole as well. So thank you.
You're very welcome, and thank you for having me on.
Fantastic. Well, our next speaker is not joining us live, and that was always planned. I think she's fit and well. This is Cher Chow, who is doing her PhD currently at the Centre for Biological Diversity at the University of St Andrews. She is a coral reef specialist. She studies how biodiversity in coral reefs affects how they work. And she has recorded us her lovely presentation, and she's going to tell us all about the complexity of life on coral reefs.
I'm Cher Chow. I'm a coral reef ecologist based in Scotland at St Andrews, and today I'm going to be talking to you guys about the hidden power of corals. So as I said, I study corals. I'm a coral reef ecologist. I study how complex living things are in these amazing ecosystems. So I study the complexity of life there. And also I study why coral reef ecosystems work so well. And one of the places that I work in is the Great Barrier Reef, which is really well known to be able to be seen from space. And now I'm not here with you guys today because actually right now, by the time you see this recording, I'm on the island where that arrow is pointing at right now. I'm in the Great Barrier Reef at the moment, so I can't get enough internet to come and talk to you guys. But I study these amazing coral reefs that are built by these animals, corals, that they build these reefs so big that you can see them from space. And when we zoom in, coral reefs are made of corals. All of these coral colonies that you can see here, they live together in this community and build these hard structures that fishes, crabs, and other animals can live in. And when we look really closely, all of these coral colonies are just composed of these tiny, tiny organisms that are animals, and they're just related to jellyfish and anemones. And if we look in, you can see those little arms poking out, and they're coral polyps. So these huge structures that we can see from space are actually built and created by these tiny, tiny creatures. And that is their hidden power. And when we think about corals, we often think about them in colonies of these big groups. They're actually groups of thousands and thousands of animals, and they can come in all of these different shapes and sizes. And so that's why I study complexity, because there's so much diversity that you see here with the shapes and how big or small corals can grow. So they can be anywhere from boulder size, bigger than me, like this one on the left here, to ones that look almost like cauliflower and broccoli, and then ones that look like tree branches. So corals build these incredible different structures all over the reef. And when we zoom in, there you can see thousands of little coral polyps that make up these coral colonies. And each of these is an animal on its own, believe it or not. So these huge reefs that you can see, they're actually built by the tiniest animals that you can find in a coral reef ecosystem. They just have their arms where they grab food, bring it into their stomach, and they sit in the skeleton that they built. And the skeleton is the coolest part about corals, in my opinion, because I study how much these skeletons grow. And then because of the skeletons growing, they build a reef. And so they build this skeleton by just sort of putting like calcium carbonate underneath them. And then by putting it underneath them, they're building up their own exoskeleton around them. And when you have thousands of coral polyps doing this for a colony, we have a colony growing tiny bit by tiny bit over the years. And then when we have millions and billions of these small builders do this over time, it builds giant reefs. So what we see in the photos before, those giant reefs, those are built over hundreds and thousands of years just by the work of these tiny, tiny polyps that we find. And so because these coral polyps are so small, it's really hard to find out how fast they're growing, how much they're growing, just by looking at them. You can't measure it at that tiny of a scale. So what the lab that I work at does is that we create 3D models of reefs like this. So we can see like the entire structure of the reef. We copy that onto our computers. We can move it around and we can analyze it. And because we have it for a whole reef over the course of many, many years, we can stack that on top and say, okay, these tiny things we can't measure, but we can measure how they're growing all together, all at once. So this is something really cool that we've been seeing in the research that I've been doing. But also, because these are such small animals, they can also be very vulnerable. Even though they can build these beautiful ecosystems and structures, corals themselves are just, in reality, very small animals. And when we hear about coral bleaching happening, that's because with climate change, each summer around the world in the tropics, the seawater is getting warmer and warmer because all of that seawater is absorbing a lot of the sun's energy. And with every summer getting warmer and warmer, it actually stresses out these really small polyps. And when it gets to a temperature that's really uncomfortable for them and getting stressed out, like if you and I go hiking and it's a really hot day, we don't feel as comfortable either. We respond by sweating. But these corals, they respond by kicking out the thing inside them that helps them photosynthesize and create energy. And so when that happens, these corals can no longer get their food or energy, and then they get even more stressed out. And when this is long enough and happens over a period of time, they eventually die. And so that's what we mean when we hear about mass coral bleaching, either in Australia, Hawaii, other Pacific islands. This is what has been happening for the past 5 to 30 years. And it's really sad. But what I'm saying is corals have hidden powers. And when this happened at our site in Great Barrier Reef, of course, it was really sad. But as we going back, we still have to do our research. We kept going back and we waited. At first, it looked like this. The corals died initially. And so because the animals weren't there to keep the skeleton growing anymore, they were starting to break apart and things looked really flat at first. But as the years went by and we still had some corals surviving, we looked under those corals and we found these tiny babies starting to grow back. This is a photo I took 2 years ago where you start seeing these tiny, tiny blobs of corals coming back. And if we think back to our 3D maps, we do them every year, even if the reef doesn't look too happy or healthy. And this is what it started to look like. So focus on that coral where the star is. You'll see all the corals start changing around it year to year. Every click is a different year. We started seeing things grow back. So these corals, they're tiny creatures. And even though they could be vulnerable to something like as small as a temperature change, they could also be really resilient. They can recover and grow back. And last April, I went back to this same site that you see here on the map. We went back. And this is what the reef looks like now. So I just wanted to tell you guys about this hidden power of corals growing and building reefs, to show you that don't underestimate them. They've been building these incredible ecosystems. So thank you so much for having me today. I know we hear bad news about coral reefs a lot of the time, but all we need to do is give them the time so that they can recover, and just believe in them, because they grow really hidden and tucked away. And then in the blink of an eye, you come back and they're eventually grown back. So thank you so much. And yeah, thank you.
Well, I hope you enjoyed Cher's fantastic talk, and perhaps felt, if you're like anything like me here in England, felt a little bit warmer with the idea of diving in those beautiful tropical waters in the Great Barrier Reef. And hopefully she's having a fantastic time doing some very important research out there with those wonderful corals. Now, it is my very great pleasure to introduce our next speaker, who joins us very early in the morning, which is incredibly generous of her to join us. This is a great pleasure for me. I have to say, I'm very excited to meet you. This is Colleen Cavenagh, who is a personal hero of mine. I've recently wrote a book about the deep sea, and I have to say, reading about your work and writing about it was a huge pleasure. So thank you for joining us, Colleen. Welcome to the summit.
I'm all set.
Fantastic. Yes. Well, first of all, I should just briefly introduce you to our listeners and to our viewers at home. Colleen is an expert in deep sea hydrothermal vents, those extraordinary extreme ecosystems down in the deep ocean. And in particular, she studies the symbiotic microbes that power these amazing ecosystems. Now, Colleen, I would love to know how you first got interested in studying ocean science, and especially the stuff that's so far down deep beneath the waves.
Well, actually, I grew up in Michigan, which is surrounded by the Great Lakes, and did not even see an ocean until I was a sophomore in college. But Michigan is how I got interested. They offered a marine ecology course at the Marine Biological Laboratory, which is comparable to the MBA, and took that, and we were expected to come up with a project on our own. I actually studied horseshoe crab mating behavior. But I fell in love with research. I found out that not everything is known in textbooks or in scientific articles even, and that anyone can pose questions, and that it is a hidden world. It's like being a detective. And I ended up staying in Woods Hole, going to graduate school, and being there when the vents were being discovered. Well, they'd been discovered, but a very excitement in the Woods Hole community. And I'll tell a little bit about that.
Fantastic. I can't wait to hear that. And I have to ask you, before I let you take over the screen and give us your presentation, you have been down into the deep sea. I believe you've been diving inside of incredible submersibles like Alvin. What is it like?
Oh, it is amazing. They're using a lot more remote vehicles, but it's absolutely true that they don't have the 3D impression of being actually in the sub. Some of the practical aspects are, it's a very small space with a lot of equipment. So you're like this with 3 people, and there are no bathroom facilities, and you are down 8 to 10 hours. So so that's an interesting aspect, and everything in the sub is recorded, all noises.
Okay, good. So let's think about that for a minute.
But it is truly amazing. And there are opportunities now with subs, at coastal resorts, and I recommend going down, doesn't matter what depth.
Absolutely. No, there's wherever you get in the ocean, there's always wonders to see, and they just get stranger and stranger the deeper down, I think, the deeper down you go. So that's really such an inspiring place to explore. And I think perfect time for me to hand over to you, Colleen. And the screen and the audience is yours.
Okay, thank you. And thank you all for signing in. As just mentioned, I've been working on hydrothermal vents started at Michigan and swore I would be true to the Great Lakes. But in my opinion, the Great Lakes, because you can't see the other side, are just like oceans. So today what I want to do is talk about the hidden worlds within hidden worlds that I study. And that is that, as was just mentioned, we're looking at from the deep sea, which no one can see from the surface, I'm working at 2.5 kilometers down, but also animals that were discovered there that turn out to have these bacterial symbionts. And of course, when a zoologist looks at an animal, they're not thinking about bacteria, internal bacteria. We think about it a lot more with the human microbiome now. But it actually took a number of techniques that are actually just like diving in Alvin, where you go deeper and deeper and deeper to be able to visualize bacteria. And I'll talk a little bit about those discoveries from the point of view of the techniques that you have to use to study things in the deep sea besides Alvin. Oops, there we go. So first I want to talk about the microbial world, just so that you know. Oh, I lost my spoon. So there are 1 million bacteria in a milliliter of water, and it doesn't matter if it's seawater or fresh water, tap water or bottled water. So that means that you are showering with millions and millions of bacteria. And to amaze your family and friends, in a teaspoon, there are 5 million bacteria. So you have lots of them around you. The amazing thing is that they are so diverse in their metabolism. And this is in large part because they've been around for almost 3 billion years, and they have co-evolved with the evolution of our Earth's planet and geochemistry. So you have bacteria that can fix nitrogen gas out of the air into fertilizer like ammonium, and others that are chemosynthetic that I'll talk about. The other, as I mentioned, digging deeper and deeper into our understanding at the molecular level, is that there's been a revolution in microbiology which has involved using DNA sequencing to identify microbes and other organisms. And this is very important for microbiology because the vast majority of bacteria cannot be cultured. So we know nothing about them. Now we can actually look at their origins, their relationships to other bacteria, and also what they're capable of, both genetically, excuse me, and metabolically. So this has been, it's an amazing time to live in a microbial marine world. So deep sea hydrothermal vents, I want to mention quickly, were discovered in 1970s, excuse me. Pardon me, I'm sorry. And these are hot springs that are along the mid-ocean ridges where the plates are separating. And they're, if you ever get a chance to go to Yellowstone Springs, those are those sorts of springs, but at the surface. And these were explored with geologists initially who were looking for these springs, but had no idea that there was anything biological to look at, but in fact discovered flourishing ecosystems with these tube worms that are amazing in their red plumes, so-called Yeti crabs, this fuzzy looking crab, giant clams the size of dinner plates, mussels, and there are hundreds of new species that have been discovered here. But one of the big questions, and the base of the food chain is chemosynthesis. It was postulated because there was a lot of hydrogen sulfide in the water samples, and chemosynthesis is a process that only bacteria and archaea can do. And this is using inorganic compounds such as sulfide or iron or hydrogen as their energy source. And it parallels photosynthesis, chemosynthesis, in that plants and algae can use sunlight as an energy source, and carbon dioxide, just like chemosynthesis, as their carbon source. So they convert CO2 in the seawater or CO2 in the air into sugars that then feed themselves and make new bodies. Excuse me. But the question remained when I was a first-year graduate student, how are these giant tubers making a living? They have discovered, it was discovered, they have no mouth and no gut. They get up to 2 meters in length. And there was virtually no food at the vents. There was a lot of particulate matter, but how could they eat? And this is where I came on the scene. Meredith Jones, the curator of worms at the Smithsonian, gave a talk at Harvard and mentioned that they had elemental sulfur crystals in this tissue that fills their coelomic cavity. And it was at that point that I jumped up and said, it's clear they must have chemosynthetic bacteria that use sulfide, converting it to elemental sulfur to fix CO2 and feed the coral, feed the tube worm inside. And that's just like what was just being talked about. The corals, which have algae within their cells, are fixing CO2, making sugars, and feeding their animal host. Fortunately, he sent me some tissue and I was able to show that there are bacteria. And this took a variety of both microscopic and chemical analyses, because to postulate something like this, you have to be able to prove it. And honestly, no one believed me. But using light microscopy, a DNA stain, and then scanning electron microscopy, which is the picture that shows all the balls, was able to show that there are lots of bacteria there and that they are intracellular. Let's see. And this is basically effectively nutritional symbiosis. The host is benefiting from the carbon that the animals are making, and they have a remarkable adaptations, including that red is due to a hemoglobin that transports not only the oxygen that the bacteria need, but also the sulfide. So they're bringing it into the bacteria inside of their tissues. And then the bacteria in turn, let me show the next one. The bacteria in turn are bridging the anoxic-oxic interface. The sulfide is coming from below at these vents. The oxygen is in the seawater above, and the host is actually bringing both of those together to feed the worms. So around that same time, there was a paper in Science called Gutless Bivalves. So of course I was like, oh, they must have symbionts. And indeed, they live in sediments, coastal sediments to slightly deeper, that are that muddy. If you go to the beach, or not the beach, the mudflat, and you smell that rotten egg smell, that is food to sulfur bacteria. And Solomia velum, a little clam that's in that group, lived right off Woods Hole, and I was able to collect them, and with the help of many, able to show indeed that they also have these bacterial symbionts. The rod-shaped organisms are within a cell there, and that the round thing is a nucleus. So, and the crazy thing is they had been shown both in the fossil record and the modern record that the bivalves make these Y-shaped burrows. And in fact, that's the perfect adaptation, because they're getting the sulfide from below and they're pumping oxygen from the above to the overlying seawater, from the overlying seawater. So they're providing for their host. They're providing for the symbionts are providing for the host. Well, indeed, these symbioses are now found in numerous animal phyla and also protists. This is an evolutionary tree based on the symbionts relationships. And you can see based on the host, they're found in clams, sea anemones, mussels, nematodes, oligochaetes. And these are ranging from diverse habitats from across the coastal ocean to the deep sea. The deeps at the hydrothermal vents, the chemosynthetic symbioses are extremely widespread. Virtually all of the macrobes, that is the large organisms as we call them, have the symbioses, the symbionts, and they effectively are the forest. They're providing not only for themselves, but they're providing the structure for the ecosystems. In addition, I just want to point out, this allows both host and the symbionts to colonize otherwise inhospitable environments where there's virtually no food, or there's high levels of sulfide, which the bacteria can also help to detoxify. And so it's no wonder that these are widespread. And it really took, from a hidden world point of view, it took diving to the deep sea, 2.5, 3.5 kilometers depth, to discover these ecosystems that are based on primary production using inorganic compounds emanating from the earth versus photosynthesis on the surface. But it took that to illuminate the world, our world, to see that these symbioses were widespread right under our noses and didn't recognize them. Let me see. So I just want to close with, for both marine biology, for microbial biology, for science across the board, that not everything is known. Dig deep into your reading, starting at the encyclopedia or Wikipedia level, popular literature, and then ultimately getting into the scientific literature. And you will find, as you get into this hidden world, that there is so much that is unknown. And it just opens a whole world, and I and you can expect a discovery. Thank you. And these are some of my personal symbionts I was acknowledging.
Do you want to put that back? Should we have
No, it was just, well,
There we
sure,
go.
I can show
There
it
we
for a
go.
minute. It's science is, as they always say, you're standing on the shoulders of giants, the people that came before you. And these are members, some of the members of my labs, my mentors, and of course, the captains and crews, the chief scientist, and the Alvin support group that make all of this possible, especially in the deep sea.
Well, Colleen, thank you so much. You are indeed a great giant, and I know many people stand on your shoulders as well. So thank you for that wonderful presentation, sharing the wonders of the deep and how that's opened up so much more of our understanding of the whole planet. And we have some questions, if we can throw them your way, just for a couple of minutes. Some great questions coming in. One, which is, does chemosynthesis only happen in invertebrates?
No, was discovered over 100 years ago in free living bacteria. By a Dutch scientist called Beyering, named Beyering. And you can actually, if you're near the shore, or you have access to, you can even do this if you have a seawater table, or a seawater aquarium, get a mussel from the fish store, kill it, and just let it rot, and you'll find a veil of sulfur bacteria develop over it, a white veil. And this is another adaptation by free living bacteria to trap the sulfide from below and to have the oxygen from above. And there are a variety of these sorts of adaptations by free living bacteria.
Fantastic. So they are all over the place, and you can get your hands on them as well if you want to see them for yourself. We have another question here from Karina, who says that she's learnt how the origin of cells could have come from within deep sea hydrothermal vents. Do you think this is true? And do you know if there's any current research on this?
This, it had been thought that life originated in the, kind of shallow shores with seawater and some of the chemistry, methane, and then lightning zapping it and creating nucleic acids. There are lots of opportunities in the deep sea for this sorts of chemistry to go on. And you have all of the building blocks, the sulfide, the methane, the CO2. So it has been postulated. As you know, we can never really get at the origin of life. So you can have work that supports it. And there are still people on both sides of the camp. But yes, there is definitely research, some of it theoretical, because you have to postulate what was going on way back then. But it opened up a whole nother world of thinking on how life originated.
Fantastic. It is fantastic stuff. To think that back far in time, we can even imagine what might have happened to bring us all here today, talking around the world and this fantastic technology we have. Here's a question from Charlotte. I don't know how you feel about this one. It's certainly quite an active topic at the moment amongst many scientists, which is, will deep sea mining be detrimental to these organisms down on the deep seabed in hydrothermal vents?
Yes. In a word, there is extensive research going on by many of my colleagues. Some have turned it really into activists. Unfortunately, because of the chemistry of the vents, which is bringing a lot of precious metals and rare metals to the surface from the very deep, you're forming things like manganese nodules and lots of deposits that make it very lucrative to mine. And they actually have machines now that are they're like bulldozers that just go, just trample over the bottom and have these conveyor belts and other methods that then send the material to the surface. And there are a lot of efforts to try to make these sanctuaries, because if they're outside of the economic zone of nations, then it's like a free-for-all, or the nations are able to license. So yes, it's a huge issue, as I know from your book is.
It's an ongoing issue. I wrote my book partly about deep sea mining, and that was a couple of years ago, and it's still a very current issue, and a lot of things are changing. So if that's something any of you guys are interested in, it's definitely happening now, and in terms of its unfolding, and we'll see what happens in the coming years. But it's because, thanks to work of people, wonderful people like Colleen, that we know about what's down in the deep ocean and just how important it is for all of life on Earth. So so Colleen, thank you so much for taking time to speak with us. I know we have loads more questions, but I think we're going to have to move on. So I shall just say thanks again, and enjoy the rest of your day.
Okay, well, thank you. Enjoy the summit. This is fantastic.
Right, so we have one more fabulous speaker before we take a little break for lunchtime, certainly lunchtime here in England. And I'm very pleased now to introduce you to Yasmin Meeder. Yasmin is a PhD student at the Marine Biological Association and at the University of Exeter. She is an expert in tiny plant-like creatures called diatoms, which I think she's going to tell us all about. Yasmin, welcome. I'd love to hear from you just briefly. How did you get into being a marine biologist and into this particular area of research in the tiny wonders of the open waters?
Thank you so much for the introduction, and it's great to be here today, and especially talking to all of you that are so excited about the ocean. So how did I really get into this topic? Well, for me, it was kind of understanding this really complex system that we have as the ocean, and it's just, I really wanted to dig deep into this, but I didn't go into it in the normal or natural route. I studied biology as my undergrad, and then I sort of branched into this unique life of doing sort of tiny microbial work. And I think this came about mainly because I really like to do biomedical science, and I also really liked ecology. And I wanted to sort of bridge the gap and use the skills that I could do sort of in the biochemistry or molecular biology, and then using that and studying the ocean.
That's fantastic. So a real mixture, which tells us that marine biology doesn't have to sit in a box. It can be connected, and it is connected to so many other areas of research and other areas of our world. And something else you do, Yasmin, is science communication. How did you get involved in that, and why is that something that you think is important?
I think I really just stumbled into science communication. I think it was really important that my family didn't really understand what it is that I do, and I created an Instagram page that really was just for family and friends to say, this is what I'm up to. I don't live in my hometown anymore. This is just a way to connect and show people what I'm up to and how scientists live, or what we do and what we do in the lab, because there's a misconception that all marine biologists go out on a boat or go diving. I'm not a great swimmer. I don't go diving. I don't even have a scuba diving license or anything. I'm purely a lab biologist. And it was mainly just to talk about this and communicate it. And from there, it kind of grew, and there's a lot of people that really are interested in this aspect of it. And I think it's so important to make science accessible for all different kinds of people. And that's really how I got into it. It was almost a bit of a fluke. I just started it and went from there.
Oh, fabulous. I love flukes. That's pretty much my life. It described all over the place, just doing different things that at the time I think, yeah, I'll give that a go. And you never know where it'll lead you. And it's a very exciting journey to take. So Yasmin, you've very kindly recorded a presentation for us. And then hopefully we'll have a little bit of time at the end for some questions. So if you've got any questions for Yasmin, stick them in the chat and we shall have her back on in a few minutes. But first of all, let's have a look at your presentation.
Thank you very much.
Hey everyone, I'm Yasmin Meeder, a PhD student at the Marine Biological Association and the University of Exeter. Thanks for joining for this talk. So I'm going to start with this really famous quote that I think you all should know about. And it is not the strongest of the species that survives, nor the most intelligent. It is the one most adaptable to change. And this is from Charles Darwin, who you may have heard of. And this is really setting the scene for my talk, which is about change, adaptability, how organisms can respond to their environment. There are loads of types of algae, and what do you think of when you hear about algae? Is it this macroalgae, this seaweed that you often see? Or is it stuff that you consume in your food and drink, such as spirulina? Or is it this really slimy, sludgy, horrible thing that you see on ponds? Because a lot of the time, no one really cares about algae because it doesn't look so nice. And why would you? I study a type of microalgae known as marine diatoms, and they're super important because they can absorb carbon dioxide and produce oxygen, so the oxygen that we breathe, through the process known as photosynthesis. And these marine diatoms come in all shapes and sizes, and they're known as the jewels of the ocean or jewels of the sea because they have a silica cell wall around them. And this silica cell wall is essentially that they live in a glass house. And they're super important as they form the base of the food chain. They're known as the primary producers, and they are a type of phytoplankton that you'd see here. And so this is where they can absorb the sun's energy and take up this carbon dioxide. And they're really essential for the top carnivores or top predators, such as the sharks or dolphins that we all know and love in marine biology. Even though marine diatoms are really teeny tiny, they have a big impact. And the Sustainable Development Goals that I've listed here show you loads of different examples of how diatoms can have an impact to society. For example, they're really important for climate action or for clean energy and for helping to treat and clean water. As a diatom, you're exposed to the ocean, and the ocean can change a lot. And that is just like you and I. When we're walking in the street, sometimes we're really hot, sometimes we're really cold. We're able to change and adapt by maybe putting on a layer or taking off a coat. So how do diatoms really respond? Because they've still got these changes and these things that impact them. Well, firstly, what impacts them? Temperature, just like we face. Salinity, we don't really have this because we're not marine or we don't live in the ocean. But if you live in the ocean and you've got different types of salt concentrations, this can also impact your ability to survive. And also nutrients. If your parents are always telling you need to eat your veg, it is true, but also you need those vitamins, these nutrients, and so do diatoms. They need this nitrogen, they need this phosphorus and this phosphates. And my research focuses on understanding more about this phosphorus. Even though diatoms are teeny tiny and you need to use a microscope to see them, they're absolutely incredible and they have these adaptations that they can survive changing environments. So when there's really limited phosphorus in the ocean, the diatoms can actually really just like conserve their growth and say, hold on a minute, we have no phosphorus, we're going to stop growing. And just like you see in this image here, you have a couple of diatoms, they're like, we haven't got phosphorus, we're going to chill. But then as soon as phosphorus gets into the ocean, they can sense this change and they can rapidly grow. And I find this insane because they're only one cell, a single cell. So how do they respond and detect these changes? As a researcher and a marine microbiologist, this brings me to my project where I'm understanding diatoms and their environment. But hold on, diatoms don't have a brain, so how do they sense these changes? You and I have this central nervous system that can detect when we have a change. Like if we touch something really hot, we're like, whoa, that's really hot. We'll move away instantly because we've got these signals that send it to our brain. But diatoms don't have a brain. So my project is really understanding how they can do this and how they can sense these changes without a brain and without a nervous system. So what we do know is that diatoms have this really complex mechanism that they can use to detect phosphates or phosphorus. So what we have in this image here is that phosphorus is now entered the environment. Diatoms are like, whoa, okay, we've got phosphorus. And what they can then do is detect this change in the environment and they can send signals in the cell. Now, they don't have a central nervous system, but they do use calcium ions. So calcium can come into the cell and tell the cell what changes to make, such as improving their growth or being able to regulate their bodily functions, or if that's what you put it. And so they have this cell recovery responses. So as part of our research, we wanted to understand, well, what's going on, what kind of genes are involved, how is the cell telling itself to start growing, stop growing, what's it doing? So what we do is we can grow cells under really low or limited phosphate, and then we can look at the gene expression. So the gene expression essentially means the genes that are present and regulated or upregulated and genes that aren't really there or there's less of them. So what we can do is there's different gene names and we'll see, oh, there's loads of these genes present under low phosphate, whereas this other gene, there may not be that many. Or if we've got a different stress such as nitrate or nitrogen, maybe we don't have these genes present. So what this can tell us is perhaps these genes are involved in something to do with this low phosphate because they're there, aren't they? So we're really looking at understanding this a little bit more. One way we can do this is through gene engineering. Now, when you hear the word gene engineering, gene editing, or GMO, a lot of the time it's with a lot of bad press. Or what you may have seen from a Netflix show is this unnatural selection where some dude literally injects himself and it's really wild. I would recommend to watch it, but take it with a pinch of salt because it's quite an interesting show. But essentially we can use these molecular scissors to edit a gene, and it isn't super crazy. It's not going to design a baby or anything. We just want to understand gene function. And so here is something that we've been doing, and we wanted to look at their growth rates. Growth rates is a measure of cells that are present in the environment. And what we did is when we get rid of a gene, we want to see how it performs. So here is two of the cells that we've got rid of this specific gene. And so in the normal gene, you can see here it's grown loads, you get normal growth. But when you get rid of these genes, they don't grow as well. So maybe this gene is responsible for their ability to grow, which is really, really cool, especially in my opinion, because we've now been able to pinpoint a specific gene that has a role and a function. So the key messages from this talk that I want you to take away with you today is that diatoms are really, really important for absorbing carbon dioxide from the atmosphere and being able to produce oxygen, the oxygen that me and you actually breathe. And they're also really important for the base of the food chain to help these big organisms that we see in the ocean. They wouldn't be there unless there were these phytoplankton. And diatoms must sense a change in their environment, especially with nutrients, because that's what they rely on to be able to survive. And also this pathway that we're identifying is really complex, but also it is important to then inform different things like the Sustainable Development Goals. If we don't know enough about these diatoms, how are we ever going to be able to push forward for things like clean energy and technology to help fight climate change? And then also these mutants that we've made, it's not this really crazy complex thing that we've done that's insane. We're really just understanding gene function. And our gene function has shown that it's involved in diatoms' ability to grow and sense their environment. And with that, I just want to thank you all so much for being here and good luck on your journey to being a marine scientist. It is so incredible. And if you have any questions, either send me a message on my Instagram. You can use the scanner here to find out about it. I post about science, marine science, and help people on their science journey. So thank you, and I welcome any questions.
Very quick questions, and I think you also very kindly said you'd answer a few more in the chat, so keep the questions coming. Yasmin will deal with those. But before we let you go, we have a question here which is from Quinn, who wants to know how many types of diatoms are there?
That's a brilliant question. I think there are loads of different diatoms. So we have kind of with the diatoms that I'm currently studying, we have two main groups. We have the pennate diatoms and we have the centric diatoms, and a lot of them have different shapes and sizes. And so I don't actually know the number of species, but I'd say there are so many, and they all can respond in different ways. So even though what my research is doing, I'm just looking at one particular species, and through evolutionary processes, they can all respond and react quite different to each other.
Brilliant. Yes, it's the diversity that breaks my brain. I think that's the shapes and sizes and everything about them is just fantastic. Okay, one very quick one as well from Ruben, who wants to know, what's the most important thing in your view about marine diatoms?
Oh, I would definitely say that it is their role in cycling carbon. They are obviously able to take up carbon and store it, and as they sink or die and take it to the sea floor, they're able to remove this carbon dioxide from the ocean and the atmosphere, which is really, really important, especially in the face of climate change at the moment.
It really is. Yasmin, thank you so much for joining us at the summit, for sharing such a wonderful view on this important part of our ocean and on your important work. And thanks for offering to answer some more questions. If you do have any more questions for Yasmin, pop them in the chat and she's going to very kindly have a bit of a go at answering them over there. So thank you so much.
Thank you so much for having me. It's been a pleasure, and I look forward to the rest of the talk.
Fantastic.
Thank you.
Now,
Bye.
Bye. Now, we are running over a little bit, so I'm sorry we've kept you from your very quick break, but we are now going to let you go for a few minutes before we start the next session, which we are due to start in just over 10 minutes time at 12:30 here in the UK, when we are coming back for a fantastic a batch of more talks. We are going to be exploring microorganisms, poop, viruses, elasmobranchs, viruses in elasmobranchs, I should say, and plankton. So off you go and have a break, grab something to eat, and we'll see you back here in just over 10 minutes. Thanks very much. See you soon.
Sydney Harned, and I'm a graduate student at Nova Southeastern University's Save Our Seas Foundation Shark Research Center and Guy Harvey Research Institute. I am studying the genetics of scalloped hammerhead sharks in the Eastern Tropical Pacific, including the Galapagos Islands. Specifically, the Galapagos scalloped hammerheads are under tremendous threat from the huge amount of illegal fishing that occurs here. Hammerheads in the Galapagos form large schools of hundreds of adult female sharks, and people come from all over the world to dive with them. Unfortunately, these sharks are also a target for huge fishing fleets stationed right outside of the Galapagos Marine Reserve. Hammerhead populations have been declining rapidly in the past few decades, so it's imperative that we protect the habitats that these sharks frequent. But in order to do that, we need to understand their habitat use. That's where genetics comes in. I am investigating the genetic connection between the Galapagos adult hammerheads and hammerhead nursery sites along the Central and South American mainland coast. Nursery sites are shallow coastal habitats where pregnant sharks go to give birth and where their babies remain for the first few years of their life. By taking genetic information from adult sharks in the Galapagos and babies along the coast, I can build a map of where these sharks go and how they are connected. This can help answer important questions like, are the adult females from the Galapagos aggregation traveling to coastal nursery areas to give birth? We can also use genetics to investigate familial relationships and genetic similarities between these locations and scalloped hammerheads from other parts of the world. By answering these questions, I will have a better understanding of how this critically endangered shark species uses habitat in the Eastern Tropical Pacific, which will hopefully lead to more protections on these areas.
Sharks and rays. They are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference. Sharks and rays, they are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Hello, everyone. Hope you have had a pleasant break and you're back and ready to dive back into the salty soup of marine joy with us. Now, remember to get involved in the chat. Tell us what you've learned today that you didn't know before. Drop your favorite hidden ocean facts in there. Tell us about the most beautiful planktonic organism you've seen today. Now, this session, we're going to hear from 4 speakers who study different aspects of our hidden oceans. But before we get into that, I'd like to once again thank, first of all, the Save Our Seas Foundation for their support in making this event happen. I'd also like to remind you all that in case you forgot over the break, you can ask questions via the chat just next to or below this video box, depending if you're on your computer or on a mobile device. Remember to mention particular speakers if you're directing questions at them. And the program for the rest of the day is below me right now. And any issues you might be having, you can check that out, see where we're at. You can also check out the FAQs to see if that helps resolve any problems you might be having with the live feed or the Q&A function or anything else. Now, like as is customary, we're going to kick off with a poll. And that's in the same box as the chat box next to me or below me. And we're asking what interests you most about the hidden ocean. And the options are the animals, plants, and other species that live there, the processes that sustain the ecosystems, how we can use what we learn to help humans, all of these or something else. So do get voting there. And while we wait for those results to come in, we're going to speak to Liza from the Marine Biological Association about membership. Liza, can you tell us a bit about becoming a member of the MBA?
Hi, Rory. Yeah, thank you. Hello again. So I'm just going to really quickly talk to you about our membership options. So as I spoke to you about before, we have our Young Marine Biologist membership, and that's for those aged 18 and below, which is really exciting. We have other categories. We have students, we have postgraduates, we have early careers, we have professionals, and we have associates, and we have fellows. So there's a category for everyone. Everyone is welcome. And we like to see it as a community, which currently we have members from over 40 countries. So it's quite exciting.
Amazing. It's brilliant. 40 countries is incredible. How do people join, Liza?
Yes, so there will be a link, a big banner with the cuttlefish at the bottom of this event page that says become a member. You can also find at the top of the page, it says our membership. And you can also find some more information on our website. And I think we should hopefully be posting in the chat as well how you can join.
Great. Follow that cuttlefish, folks. And what are the benefits, apart from having the cool title of being a young marine biologist? What are the other benefits?
So we're forming this community of like-minded individuals, those studying marine biology, those interested in marine biology, and those working in the field. As you can see, the platform you're currently on has hidden areas which are member-only access. These include community groups, which we're updating at the moment, which is very exciting. We have resources, so you get access to the Marine Biologist magazine 4 times a year, and you can also access all of the back copies. And there's lots of other kind of goodies and the bits of information that we're constantly adding. So yeah.
Amazing. Well, sounds fantastic. Like I say, when I was a kid, I wish I had the wherewithal to join something like that. It sounds brilliant. So get involved, have a look, folks, and thanks, Liza, for pointing that out. And any questions, of course, just drop them in the chat, and Liza and co will be able to help out. Now, let's see how that poll is looking. What interests everyone most about our hidden oceans? Let's have a look. See, let me just make my screen a bit bigger so I can see it. Okay, well, would you look at that? If I'd had to guess, I would have said it'd be the animals and co, the species found there, and that is winning out. But actually, a lot of you said all of these. That's a good level of enthusiasm for the animals that live there, the plants and other species, the processes that sustain the ecosystems, and how we can use what we learn to help humans. So, well, it's still a bit of shuffling around though. All of these catching up there. The poll is still moving. Don't know where it's going to settle. It's looking pretty tight. Exciting stuff. Anyway, there is going to, of course, be a very important poll coming up shortly in the next session, which is the moment you've all been waiting for, the Sea Life Showdown. Which tiny ocean beastie is going to take the crown? So we're going to see which of our speakers can convince you to vote for their particular creature. Let's see who's going to win out. Now, I think it's high time that we get on with the session and hear some talks. So we've got some truly brilliant speakers coming up, and we're going to start with a stroll into the microbial metropolis with Sally Waring. There we go, Sally. How's that for a dramatic entry? A whole noise, the sea, the ocean.
That was beautiful. Thank you.
Welcome. So, Sally, you're a postdoctoral student at Earlham Institute in Norwich, right?
That's right.
And I'm going to try and say there's a lot of big words involved in describing what you do, and I'm going to try and get them all right. Give me a little tick if I get them all correct, and if I don't, please correct me. So Sally is a protistologist, a scientist who works with unicellular eukaryotic organisms called protists. So she works with genomic data in the lab and on the computer to try and understand more about the evolution and diversity of protists, especially through photos and videos. So she's going to talk about the complex lives of these unicellular organisms. Sally, before you get started, how on earth did you realize that you liked single-celled creatures? Because you can't easily see them if you take a stroll down on the beach, can you?
You can't. In fact, I didn't even know anything about these organisms until I was doing my undergrad degree when I was 20 years old. So the first time I saw them was in a undergraduate class that was all about protists that I took kind of on a whim, not knowing anything about them, and I just fell in love, and that's now almost 15 years ago, and I'm still working with these organisms. So it didn't take much to get me hooked, but it didn't happen until quite late in my education.
It's brilliant. So those of you listening, it might yet be the thing that gets you excited about the oceans as well, unicellular organisms. And I think to convince us, Sally, it's time to show us your movies of microorganisms. So please take it away.
Thank you very much. So there are big words here, and protist is a word that a lot of people haven't heard very much, right? I'm a protistologist, and I study these organisms called protists. I've got a little introduction to talk about what protists are and how you can know if you're looking at a protist or not. So what I've got here is a phylogenetic tree of life. So on each branch of this tree is a group of organisms. And the closer an organism is on this tree to another organism, the more closely related those organisms are. So each branch is a group of organisms and they share some sort of characteristic or relatedness. And we can divide this tree up a little bit. Well, we can do this as top to bottom. So at the bottom, we've got some massive groups of organisms, the archaea and the bacteria. And these are prokaryotes, the organisms that have cells that don't have nuclei. And above that line, we have the eukaryotes. So these are organisms that have cells that do have nuclei, and that includes us and some other groups of organisms that we're really familiar with. These are groups of organisms like the plants, right? So they belong to this group, the Chloroplastida. So this includes all plants, trees, mosses, crops, things that are really, really small and things that are really, really big. We also have a group called the fungi, which you're probably familiar with, which is everything that forms a mushroom, as long as a bunch of microscopic things that form hyphae that live under the ground. And of course, we have the animals, which are over here in this group called the metazoa. And this includes lots of things we're really familiar with that live in the ocean and on land, insects, birds, fish, mammals. But every other branch in this eukaryotic part of this tree of life represents something that is a protist. So I want you to have a look and see how many additional branches there are here, right? And how many different groups of protists there are. And we really know very little about them compared to these three groups that we're more familiar with. All right, so why don't we know very much about protists compared to plants, animals, and fungi? Well, that's because they're really small. So what I'm going to show here is a video of me out in the field. This is when I used to work in New York City. So I'm actually in Prospect Park here in New York City. I'm collecting a water sample from a pond, and that water sample is full of plants. And what I'm doing here is I'm just filming what's in this jar using my phone. So we're not even at the microscopic level here, but you can see that there's some snails here. And if you look closely on these snails, you can see some kind of white things coming off them, right? And these, we're going to see a bit closer soon, are protists, right? So these are actually really big protists, right? Because I can see them with my naked eye. I'm not using any kind of magnification, just an iPhone. I'm filming the snail and I can see that it's covered in these little things. But without a microscope, it's really hard to appreciate the detail of these organisms. So right now, here are the ciliates under the microscope. So these are protists called ciliates. They're living in a colony. Each one is a single cell, and they're like little filter feeders. So they're kind of buzzing around these things called cilia that live around the edge of the cell, and that's filtering small particles out of the water, which they then swallow up and eat. All right, so they might be small, but they're really complex. This is another type of cell called a ciliate, and it's a predator, right? So it also eats other organisms. So what we're going to see is a small other protist come down from the right-hand side of the screen, and this cell called a ciliate is going to swallow it up. Here we go. Here it comes. That's the prey coming in now. Boom, got eaten, right? So that prey cell is now inside that ciliate cell and it's being swallowed up, and it happens really fast. So these organisms are small, but they're capable of really complex behaviors, right? That cilia is able to sense its environment. It can feel that there's a cell in its environment that's small enough for it to eat. It can feel where that cell is, and it spins around and brings the part of its cell that acts as a mouth into range so it can swallow up that tiny little cell. Now, lots of microscopic organisms are predators. This is another group here. These ones are also ciliates, but there are different species of ciliate, and they're called colepsiliates. And I love these ones because they kind of act like a pack. So what's happening here is we have a green cell in the middle, which is a green alga. And that green alga has got quite a hard cell wall around it. So it's quite hard for these ciliates to penetrate, and they want to eat the inside of the cell, basically. So they're all surrounding it, and they're using complex mouth parts on their cell to latch onto that alga, and then they'll pump out digestive juices, punch a hole in that algal cell, and then suck out its insides. So there's one cell kind of towards the bottom of the alga, kind of locked on there now, sucking away at that green cell. So these cells are capable of not only complex behaviors on their own, but they can work together. And it helps these cells to work together because the more cells that they're pumping out digestive juices, the quicker maybe they can break into that green algal cell and get the food that they want. Okay, so lots of different protists live together and cooperate. This video is of a protist called a diatom. It's called a Basilaria diatom. And each kind of, there's a whole lot of single cell units here. So each unit has got a cell wall, and inside each cell is some brown chloroplasts. And what you can see when these cells move is that they slide alongside each other. So each kind of long unit looks a bit like a needle as a cell, but all these cells are living together and they're sliding along the surface of each other's cell. And they're able to excrete chemicals in order to make the surfaces slidey so that they can slide along them. And in a bit, we're going to go to a lower magnification here so you can see why that behavior is important. So what's happening here, these cells are using each other to allow the whole colony to move, and kind of like it moves like a carpenter's ruler, kind of. This diatom is commonly known as the carpenter's rule diatom. And so by living together and using each other to allow their motion, this colony is able to move much greater distances much faster than each individual cell could on their own. So these cells, they live together to cooperate, and they live together to hunt. And we're going to see some more ways that they live together. So this is an example of a protist where that living together has become so complex that you've actually started to get almost a multicellular organism here. This is an organism called Volvox, and this is actually one of the first protists that I ever saw under the microscope, and this is what got me into studying protists in general. So in this case, we have cells living together. So every little green dot is an individual cell, and those cells are capable of living on their own. But in Volvox, they live together in a colony that's held together with this very fine mucilage. And then inside the colony, you can see larger green bundles of cells. So those are individual cells within the colony that have differentiated from some of the others, and they are going through sexual reproduction, whereas all the other cells will not go through reproduction. So those cells in the middle will produce new colonies that look exactly like these adults, while the ones on the outside will never do that. They contribute to the way the colony can move around, and they will help the cell to provide itself with the things that it needs to live, but they will not reproduce. So what's happened here is that we have some cells that are able to reproduce the next generation and some that are not, right? Because they're all genetically similar, it's thought that they're all contributing to all their genes are getting through to the next generation, but some individual cells are sacrificing themselves for the survival of the others, and only some get to reproduce. So those are the new colonies forming inside, and soon what will happen is that adult colony will pop open. Those daughter colonies, they're called, will pop out, and they'll look just like the adults, and then the whole process will repeat and go through another one. All right, but not all protists live together. Some of them are solitary, and they live all on their own. This is one of my favorites. This is another cell that I saw early on in my career, and I thought it was extremely beautiful. And I just wanted to show it to you because it's an example of how beautiful and elegant and complex these cells can be. So this is an organism called a euglena. It's a Phacus euglena it's called. And I want you to look at how it moves through the water. So whenever I see these cells, they actually come in lots of different shapes, but I always think they look like falling leaves drifting through the water. And I love them. I love to see them, and I think they're beautiful. But not only are they beautiful, they also contribute a lot to each environment. So all through the ocean or any kind of freshwater body that you come across, there will be many, many protists in that water body. And ones like this euglena are photosynthesizing. So they're trapping carbon and they're producing oxygen. They're acting as a primary food source for slightly larger organisms that might eat them. And then other ones like the ciliates we saw, they were eating other organisms, are helping to break down organisms, to recycle nutrients, and contributing to just the turnover of nutrients in these environments, right? So these cells are beautiful, they're complex, they have complex behaviors, and they play a really important role in all these environments, particularly the ocean. All right, so just to sum up, we've seen a few different protists, but only from very few groups. So we've seen some ciliates, which are in this group called the alveolata. We've seen some euglena, which are in the, we've seen some diatoms, which are in this group, the SAR. We've seen we saw the volvox, which is in this group, the Chloroplastida. So it's actually in the same group as the land plants that we saw right at the beginning. And we've seen the euglena, which is in this group, the excavator, right? So we saw a few different organisms and they all look different and they're all very diverse, right? But we only explored a few of these branches on our protist tree of life, right? So there is a lot more diversity out there. If you ever want to learn more about the different protists that are out there, you can check out some of the work we do. I have a website and Instagram where we post videos and information about the different protists. And I'm working on a big protist project where we study the genetics of these organisms at the Earlham Institute. And this is part of a big project called the Darwin Tree of Life. So you can look at those resources if you want to hear more about all these different protists that feature on these different trees. And that's it. Thank you.
That was awesome, Sally. Thank you so much. And I've got to say, euglena, it's also my new favorite as well now. That's beautiful. It was moving like a ray or a skate. I thought it was very lovely.
Yeah. Some of the other ones have kind of a body that's able to change shape. It looks like a blob of jelly that can kind of get narrower and wider depending on how it's moving and what it's moving through.
Amazing. And that ciliate that you showed at first that sort of gobbled something that did a backflip. Brilliant. What is the difference? So this is the first question we've got come in. Sally, what's the difference between multiple cells living and working together and a multicellular organism?
That's a good question. And sometimes it's not that easy to tell the line, right? You do have examples of the line between kind of a colony of organisms where you might have multiple cells working together and a multicellular organism. Generally, the distinction is thought to be when you have differentiation of those cells. So often in a colony of organisms, every cell is capable of reproduction and every cell could technically live on its own if it needed to and reproduce. In a multicellular organism, you lose the ability for the cells to all live on their own, and not all cells can reproduce. So Volvox that I showed is a true multicellular organism because not all those cells can reproduce. Whereas Basilaria, which was the diatom colony, all of those cells can produce new cells and each cell could live on its own. But it's a bit like the line's a little bit wavy and it's not always perfect, but that's the general distinction.
Like many things in biology, the lines are wavy. Sally, that was great. I've got time just to squeeze in this question, which I think you've answered already, but let's just be sure. What is your favorite protist?
My favorite protist? Well, I do love Volvox mainly because it's the first one I ever saw. And I just remember it being in the lab and seeing it under the microscope and I just didn't realize that microscopic life could be that beautiful. I'd never really seen microscopic life move around before. I'd always seen just kind of static images of microbes. And I'd never really thought about the fact that they could behave in complex ways or have complex life histories. So I do have a soft spot for Volvox. I also like Volvox because you don't actually need a microscope to see it. It's big enough that you can just see it with the naked eye, which is sometimes fun if you find it in the wild.
Great. Sally, thank you so much. Queen of the protists, everyone give it up for Sally.
Thank you.
Now, next up, we have Cordelia Roberts. Hello.
Hi, Rory. How are you?
I'm very well, thank you. Cordelia, I think I'm right in saying you're an ARIES PhD researcher at the University of Plymouth Marine Biological Association, and the ARIES stands for Advanced Research and Innovation in the Environmental Sciences. It's not your star sign, correct?
No, my star sign is Taurus, actually.
That's good to know. I should be checking with everyone the star sign if we're giving out yours. But anyway, we know you're a Taurus now. Cordelia is a microbial oceanographer interested in how organisms interact with dead and decaying organic matter in coastal and open ocean environments. But Cordelia, you've given your talk a slightly different title, including the words poop and death. Now, I'm slightly worried about asking how on earth you got interested in these subjects, but I am going to ask you, how on earth did you get interested in poop and death?
Well, in my first year of university, I started learning about something called marine snow, which you might have heard of, which is basically when everything dies, we have this like most stupendous snowfall that happens from the surface of the ocean to the deep sea. And then I kind of got more and more into that. And then I worked out actually marine snow isn't just one kind of thing, and that includes fecal pellets as well. And so kind of have some in my talk and show you, and I think they're pretty cool. And so, yeah, really that's where microbes thrive and survive.
Great. Well, I think without further ado then, let's get moving. Cordelia, let's have a look at the world of poop and death.
Hello everyone, thank you for joining me in what will be a presentation about poop and death in the ocean and where microbes are thriving. Let's get into it. So I'm going to start you off by thinking about something that you might be familiar with on land. So this idea of a classical food chain. So we have a primary producer, a herbivore, a secondary consumer, and then something like a predator. And that exists in the marine environment. That is something that does exist. We have microscopic algae known as phytoplankton. They're photosynthesizing, they're getting CO2, and they're taking up carbon from the atmosphere that as humans we've become very good at putting into the atmosphere. And that's a major purpose of the ocean and their microbes is that they take up carbon dioxide and store it away from the atmosphere for some time, depending on how deep it gets into the ocean. But grazing on that, we have these small crustaceans known as zooplankton. On them, we have the fish that graze on them, and then all the way up to predators such as sharks. But we can begin to explore even just a small fraction of that food chain and expand it out into a food web. So we can start breaking down the different species that we see and different size classes. We have small phytoplankton known as picophytoplankton and large phytoplankton, and that can then in turn feed different size organisms within the zooplankton. So we can have even smaller zooplankton, so microzooplankton that get grazed on by these copepods, which then get grazed upon by krill, which are these larger kind of shrimp-like organisms that live in the Southern Ocean. But they'll also graze on the diatoms or the picophytoplankton, and there's a whole host of interactions that take place there. And this is just one part of that. It gets even wider. And so what I'm interested in as a researcher is thinking about this step below here in the twilight zone. And so in the sunlit surface ocean, we have this primary production from the phytoplankton and grazing by zooplankton. But I'm interested in what happens when it gets out of the surface ocean. And particular organic material is produced from dead and dying phytoplankton, from pooping and excreting zooplankton, and what happens in the dark depths of the ocean, and why that might be important for us as humans and as a global ocean. So the production of this particulate organic matter, or POM as we call it, is incredibly important for transferring carbon from the surface of the ocean into the deep sea. And that's important because we need carbon dioxide removed from the atmosphere as we keep pumping more carbon dioxide into it, and we need it taking out of the atmosphere and stored away for as long as possible. And so these particles, or this POM, is incredibly important and a useful tool to sink from the surface to the deep sea. And so depending on the different types of particles that we get, depends on how effective that these particles are at sinking. So we can have fecal pellets, which are these kind of central ones here, and we can get those from zooplankton and fish. We can get different types of detritus. So these particles here and these particles here are quite different, but that can depend on whether we're close to the shore, to the coastal and terrestrial land environments, a material that might get washed in if it's rainy or stormy, what a material gets taken into the ocean, but also out into the open ocean. We can get different kinds of species composition or different diversity of species that result in completely different material that is produced. And we can also get these carcasses where here we see a copepod species that's died, but we can get their carapaces, so they molt their kind of exoskeleton shell, and those can sink quite quickly along with fecal pellets and transfer the material to the deep sea. So when we think about who is thriving on these colonized particles, the first thing that quite often comes to mind is bacteria. So this here is a picture of a particle with a dye on it, and everything that's in green is bacteria that are fluorescing under this light. And so we can also see in this little video here, this spinning around, we can see how patchy they are. Not all particles are equal in the amount of nutritious value that they have, and so we may get these pockets where there are a lot of bacteria in comparison to other areas where there aren't so many. But this is one kind of particle that has been made within a laboratory environment. And that's very uniform. But in the open ocean, it's not as straightforward as that. So these particles that are just getting lit up right now have been taken from the marine environment, taken from the open ocean, and they've been sliced and stained to look for the bacteria. And you can see that there's a wide range of different types of particles, and it's kind of interesting to see panel D, which is a dead copepod, and you can see the bacteria that have colonized on their legs. But bacteria aren't the only kind of things. We have other species that are eukaryote species, so these are more similar to us actually, and they're more closely related to us, but these include fungal groups and fungal-like groups. So these pictures here, you can just see there are some little sort of hyphal networks, which are how fungi grow. And you can see here that we have some kind of brewing, something a bit close to like brewer's yeast or baker's yeast, these little fungal yeast cells that exist that have found themselves onto these particles. So why are these microbes even important? Well, one thing is that when microbes degrade organic matter is that they're respiring. And what does respiring do? It produces carbon dioxide. So depending on where a particle has sunk to within the water, depends on where that carbon dioxide is going to sit and how long that's going to come out from the atmosphere. And ideally, we want a particle to sink as far as it can into the deep sea so it can be locked out of the atmosphere, that carbon, for as long as possible. But that doesn't always happen, and sometimes carbon is respired within the surface ocean, and then that carbon is released back into the atmosphere as soon. But it's not all bad. We can have this kind of enhancement of our cycle, of our food web. So when we have things like copepods that are grazing on particles, if they break them up in a process called microbial gardening, they can actually allow more organisms to colonize and increase that nutritional value. So that encourages this kind of circular circularity of organisms feeding and then feeding on another one, and actually that carbon being kind of kept within the ocean rather than making its way back out. And ideally, we want all of that carbon to stay in the ocean and help mitigate the impacts of climate change.
Absolutely brilliant. The power of poop in combating climate change. Who'd have thought? Really loved that. I really loved the slide in particular where you'd arranged all those little poops into a beautiful shape. Who'd have thought fecal pellets could be so beautiful? Now, straight on to some questions for you. And the first one we have is a great one. Are microbes extremophile?
Well, that is a really good question. And not all microbes are extremophiles, but I guess you could say in some ways they are, because to live so smallly could be a bit extreme. But, you know, we do have extremophiles. We have tiny microbes that live in the deep sea, which they're facing like incredible amounts of pressure. Some parts of the deep sea are really, really cold. So they're facing cold temperatures. Some parts are really hot, like we've already heard about these deep sea hydrothermal vents. They can be hundreds and hundreds of degrees in temperature, and they've got to face that environment. Sometimes they can live in an anoxic environment, so there's a lack of oxygen there. I'd say they're all pretty extreme things. The ones in Antarctica, in Arctic oceans, you know, they're also pretty extreme. So yeah, we definitely do have extremophiles.
Hardest organisms in the world, I reckon, with all those environments covered. That's truly impressive. We had a question in advance, Cordelia, about evolution in the oceans that I know you had an interesting story about. Could you share that with us?
Yes, so evolution in the oceans does happen, and we've actually been able to measure over the space of around 100 years evolution happening in nature. So there was a researcher at the Marine Biological Association about 100 years ago who started studying some worms and how tolerant they were to salinity. And so they would kind of see how they would deal with different salinity, so how salty the water was, and just in Plymouth at Wembury Beach. And they kind of were able to show that they had a tolerance, so they had a limit to how salty the water could be. And then 100 years later, some people did exactly the same research, and they were able to show that their ability to tolerate more saline water, so more salty water, and that's something that we're expecting with climate change for salt water to kind of like intrude into fresher ecosystems. So into rivers, we expect there to be this saltwater intrusion. And they're actually able to show that that tolerance increased and that they were able to tolerate that salty water. And so over a 100-year time period, researchers in Plymouth were able to see that species evolving over time. And these worms are little flatworms, which are pretty cool. I don't think they get enough attention, so yeah.
I love your enthusiasm for the undersung heroes of the ocean, Cordelia. I'm all about it. I think we got time for one more, which is, when did you realise that this quite specific field, and quite specific is doing a lot of work there, isn't it? That was what interested you the most, Dr. Roberts?
Oh, I'm not Dr. Roberts yet.
Oh, sorry, of course. Your PhD is
I'm still PhD. I've got a while. I've got to pass my PhD first. But it's a really good question. I think I always found the unsung heroes of the ocean more interesting than things like whales and dolphins. I've always found tiny things, and I found myself asking these questions about all of these unusual parts of the ocean, and then I kind of just fell into this idea of marine snow in my first year of university, and then kind of never fell back out. So yeah, that's how that happened.
Love it. Well, it's a great story. And for anyone listening who's felt inspired by that, I hope you can follow the marine snow down deep into the Hadal Benthos. Thank you so much, Cordelia. It's been great. And let's move on to the next presentation. So Joining me now is Fabiana, who looks at viruses in sharks and rays. Now, Fabiana, I believe you're a postdoc researcher in Porto, in Portugal, with a PhD in pathology and molecular genetics. And this is really testing me. All the qualifications I'm dealing with here in this session, some serious status all of you have got. I've got to wrap my tongue around a lot of difficult words here. Fabiana focuses on elasmobranch immunobiology with a particular interest in understanding how pathogens shape a host's immune diversity. So basically this is, in short, viruses in sharks and rays. Is that fair to say, Fabiana?
Yes, indeed. So yeah, first I start to study the host immune system. So the immune system, or how elasmobranchs can protect themselves from all the aggressors. But then now that I know how these genes are evolving, I want to know what are the pathogens. So I start with the viruses, and here I am.
I love it. It's brilliant. So I think without further ado, once again, we've got a lot of shark fans here today. Fabiana, I think folk are going to be fascinated to hear about the viruses that live within.
Thank you. Hi, I'm Fabiana, and today I will talk about my ongoing work, the virome diversity and composition in elasmobranchs. Elasmobranchs comprise sharks, rays, and skates, and are the oldest living jawed vertebrates presenting both innate and adaptive immune systems, turning them excellent models to study the evolution of the immune system. Elasmobranchs exploit different habitats and present different habits, being thus exposed to different stressors and pathogens, including viruses. Viruses are the most abundant entities on the planet, able to infect all living organisms, and as obligatory intracellular parasites, they need a host to complete their life cycle. Viruses are amazingly diverse in terms of shape, composition, and geometry, but overall they have a protein coat surrounding their genetic material that can be either DNA or RNA, double-stranded, or single-stranded. While some viruses can have an envelope, other viruses do not have. The main goal of this work is to provide the first baseline on the viral composition and diversity on the species, and for that I will study 11 species of sharks, rays, and skates that differ in their habits and habitats by performing next-generation sequencing. The metagenomic approach used is synthesized here in this slide. So briefly, I extract viral DNA from the shark tissues and then perform the next-generation sequencing. All the raw reads obtained were then analyzed using different bioinformatic tools. In our first attempt, we used fin tissues from 4 different species, 2 sharks and 2 rays, and for each of the species, I pulled 15 individuals and then proceeded for the next generation sequencing. We retrieved for each of the species several million of reads that were assembled into different contexts with different lengths. However, when we were analyzing these results, we verified that most of the hits were not viruses and were instead host-related, mostly the host mitochondrial genome. In this table, we have all the non-host related hits retrieved, and as you can see, they are all phages. Phages are viruses that infect bacteria. Obviously, we are not expecting these results since we are studying sharks and rays, so we start thinking what could have happened. Maybe the type of tissue that you used, or even something failed during the extraction methodology. Regarding the extraction methodology, it is largely used and described by several different authors, so we believe that there are no problems there. So we truly think that the type of the tissue is the main problem. Indeed, we use fin clips from the tip of the fin, where probably there is no irrigation or very low irrigation, and consequently there are no viruses, or they are very low. The abundance is very low. So in our second attempt, we only test one species, the blue shark, and use splint tissues instead. Splint is a key organ where some viruses are able to replicate there, being thus perfect for this type of study. The methodology was maintained. In our results, we retrieved 630 million reads that were assembled into 3 million contigs with lengths between 200 and 33,000 base pairs. All these contigs were aligned against viral reference databases. We used Diamond, and we only analyzed the contigs with a length above 1,000 base pairs. I want to highlight that the results presented here are very preliminary, that need further analysis. Nonetheless, we verify that blue shark presents a high number of single-stranded RNA viruses from the Retroviridae and Metaviridae families. And we also have 20% of double-stranded DNA viruses, mostly from the Iridoviridae family, and also have some single-stranded DNA and double-stranded DNA RT viruses. So, and now what? Now we intend to first go deeper in these results and verify really what we have there, but also we need to proceed with the next generation sequence for the remaining 10 species. And once we have the closed data set, we want to understand if the phylogeny, habit, or habitats affect the viral diversity in these species. We also believe that this study has potential to study the impact of this viral diversity on the host immune response, mainly on TLR genes and MHC genes. So thank you all for listening. I want also to thank for all the funding that support this work, as well my working team. Thank you.
Thank you, Fabiana. Another fascinating and enlightening talk there. And we've got some questions rolling in for you, and it's tough questions. It's tough questions always. So I'm hoping you've got the answers. So I guess a good one to begin with is, what is the importance of studying immunology in sharks and rays?
The immunology. So immune genes are the genes important for the host defense against everything. So once some host sharks are able to
Okay, folks, I'm afraid we lost Fabiana there right in the middle of her answering that question. So we're going to shimmy things along. We might be able to get Fabiana back by the end, and we could hopefully ask some of your questions. But yes, we will shuffle on while we try and get a hold of her. Again, this is the nature of doing things online. Sometimes this happens. So we're going to go next to the owner of perhaps the best mustache in marine biology. It belongs to Dr. Russell Arnott, who's going to be presenting with us today. Russell's here now, that you can see that moustache. It is spectacular. He is an educational director at Incredible Oceans, studied marine science at the National Oceanography Centre in Southampton, and worked as a commercial oceanographer before retraining as a science teacher. So combining his love of the ocean and education, he formed Incredible Oceans, a marine education non-profit organisation. And his research now focuses on the overlap between physics and biology, and he seeks to find out how phytoplankton are influenced by different water environments. Welcome, Russell.
Thank you. All good.
Now, I understand you're going to talk about the variety and shapes and sizes of plankton and the tricks they've got up their sleeves for survival. Not that plankton have sleeves, but if they did, there would be tricks in there. But before we get going, do you have a favourite plankton? And if so, why?
I do have a favourite plankton, and it's behind me, and it forms part of my talk, so I don't want to like spoil my talk, the end of it, because I think it's pretty cool. But it kind of, if anyone saw Sally's talk at the beginning of this session, she talks a little bit about a species that's quite closely related to it. So, but mine's cooler.
Oh. Shots fired immediately from Russell on the plankton front. Well, I don't want any spoilers, so I say on with the show and let's hear all about it.
Cool. Right. Okay. I'm going to hope that this clicky thing works. Let's see. So this is it. I'm going to talk about why plankton are different shapes. I really like this picture. This was done by an illustrator in Bristol called Scott Masson, who I work with, and told him all about some of the cool plankton that I've seen. And he made this awesome illustration for me. So that was really cool. So I'm going to kind of start off by saying that I'm not a proper plankton scientist. I'm like a pretend one. Okay, so I'm like, so everyone's like, what? But you have a PhD in this. I'm like, okay, well, I'm going to just kind of let people in on a secret, really. And the secret is that I didn't do any science A levels. Okay, so everyone's like, how did you trick the system? How are you able to do this? So basically, I was able to do this I had maths and geography. So I was able to sneak onto an oceanography course. So I kind of learned about biology accidentally, but it's been really cool because as well as being able to play around on ships, and so I went down to the Southern Ocean. I've also got a lab where I grow plankton and I make 3D printed models of them, which I'll talk about in a bit. And I also get to kind of travel around. And so I did a load of my experiments in these things called mesocosms, which I'll talk about in a bit later. But the other kind of cool thing about my job is that I also run this organization called Incredible Oceans, which I do alongside my research. So basically we go to festivals and schools all around Europe and the UK, and we do kind of cool talks about the ocean. So if anyone is interested in getting us into the school, you can just go to that website and like just give me a buzz and we'll see what we can sort out. But anyway, it's kind of cool. Who doesn't like dressing up as a pirate? So yeah, so this is my research, and this was a really interesting thing. So it is, why are plankton all kinds of weird and wonderful shapes? Because in theory, when you are tiny, and you think about like most plankton, I say most plankton that I study, phytoplankton, are about the same thickness as a human hair. So at that level, when you're so tiny, water is like thick, goopy, viscous material. So the idea behind it, when you look at the physics of it, is that it doesn't matter what shape you are, because it doesn't work like normal water. It works like sticky, goopy honey. So there's no benefit to being streamlined. So what scientists do when they try to computer model how plankton behave is they just simplify things, and they say, oh, let's just make it easy. We'll make them all spheres. Some people go crazy and make them like rugby ball shaped. And other people are like, no, maybe we can make some that are cylinders. But then you're like, well, hold on a sec. Like, if there was no benefit to being like a funny shape, then why have we got so many weird and wonderful shapes in the plankton? So we were like, okay, so let's see if we can do an experiment to find this out. And the only other experiment that someone's done about this, it's a really lovely, really simple, but really fun experiment, is this experiment where they made loads of little plankton models using Fimo clay, and they got like a tube of thick goopy glycerol, and they drop a cylinder in, and time how long it took to sink. And then they'd get a cylinder, sorry, they drop a sphere in, time how long it took to sink. Then they would remold it into the shape of a phytoplankton and see how the shape of it changed, how long it took to sink. And so we get this idea of like drag coefficient. And so it's basically, if you're a parachute shape, you're going to sink slower. And so this provides benefit to plankton, because it means that if you're heavy and you can't swim and you want to stay in the photic layer where the light is so you can photosynthesize, if you're like a parachute shape or you've got bits sticking out of you that slow how you sink, you can stay in the top layer for longer and get that sunlight to help you grow. So we were like, okay, so this is really interesting about sinking, but not all of the ocean is nice and calm where things sink. Things can be like really turbulent and really mixed up. So we were like, okay, this is interesting. We could do something quite interesting about this. So essentially what's happening in the ocean, and if you've ever been swimming somewhere really nice and warm, what you get is this top layer, which is nice and warm, which and then we have this temperature density gradient, which is called a thermocline. Underneath it, we've got cold water. So you might have encountered this if you go somewhere on holiday and you swim on the surface, it's nice and warm, and then you put your legs down a bit and you're like, oh, it's breathing down there. So if you're a phytoplankton, and you can't talk about plankton without bringing this guy in. So, and you end up in the top, then you're like, yeah, I'm in the top, and you can get all the sunlight. But if you end up being trapped at the bottom, you're like, mate, I'm at the bottom, I can't grow as well because it's colder and I haven't got as much light. So the one at the top is like, don't worry, because I'm in the top and I can bloom and I can go crazy. And then they actually end up blooming in the top and stopping light coming through even more to the bottom layer. They do something called overshade it. So we're like, okay, so this is really interesting. And because of climate change, it's making that top layer warmer, which means that that barrier between the two is getting stronger. So if you're a plankton that can get up into the top layer because you can swim up or you can float up into it, then you've got an advantage. Now, the bad news is, again, about climate change is that we've got millions of species, right, of plankton. But what it is doing is certain areas of the world are becoming calmer and other areas are becoming more mixed. So typically what we get in calm water, the creatures that like that, the groups, are the ones that can float up, which tend to be cyanobacteria. They've got a little air bubble inside them called a vesicle, which helps them float up, or something that can swim, which we call motile. If you swim, you're a motile species, and this tends to be the dinoflagellates. So they really like it when it's nice and calm and they do well. But if it's really stormy or it's mixed, then these groups like it. And these tend to be the ones that are heavy and the ones that can't swim or float. And because what happens is they get mixed up, back up into the top, where they're like, yeah, I'm at the top where the sunlight is. So they can do really well. So what we're seeing because of climate change is actually changing the type of plankton that can live in different areas, which has obviously big impacts for fisheries that can grow there because they form the basis of the food chains. But unfortunately, there's also nasty plankton, and there's 200 species of plankton that are toxic, and they give off these toxins that they're just not very nice. They give off these toxins as a competitive advantage to stop other phytoplankton from growing. And they form these things called harmful algal blooms. And sometimes we call them red tides because they turn the water red. And if you were to go swimming in this, it would make you really itchy all over. And if you swallowed some, it would make you sick. But the problem is that if this washes into a place where there's mussels or oysters or fish that you would want to eat, then that toxin builds up inside the organism in a thing called bioaccumulation. And so then if you were to eat that, it can actually paralyze you. And in extreme cases, it can kill you. And the bad news is that climate change is causing these type of plankton blooms to happen more often and more frequently. So we wanted to see what role a shape played in helping plankton do well in these different, whether it was calm or whether it was mixed. So we came up with these crazy ideas of ways of doing it. So we were like, okay, I'm going to get this really cool thing called a confocal microscope, and it lets me scan and do cross sections through different plankton species and then combine them back together inside a computer so I can build a 3D model of what that plankton looks like. And then I was like, hold on, I've got a 3D model of it on a computer. Can I make a 3D print of that? So I spent ages messing around with 3D printers. And then I was like, okay, what I'm going to do, kind of similar to that experiment where they were dropping the plankton and sinking and timing how long they took to sink. I was like, I could make a tank that could test how they perform under turbulence. So what I was going to do is get a big tank full of goop, and I was going to put a grid at the bottom that shook up and down to make it mixed. I was going to get some cameras to track how my model went. So then what I would do is I put a ball into the tank, and then I'd be like, okay, in comes the ball. Then I turn on the turbulence, wiggle the grid up and down, chugga, 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 chugga. The ball would move around and I'd map where the ball went. Then I'd get the ball, move that out and swap it in for a phytoplankton kind of model of the same mass and same weight, turn on the turbulence again and see how it performed differently because of its shape. I was like, this is a great idea. And then I couldn't get the camera system to work. And then I was having all these problems trying to use the microscope. And then someone else said, I tell you what, let's just do it with living plankton. And so they were like, there's this awesome facility up in Sweden in a place called Umeå, which is the northern Sweden. And I had to go to this village called Nordbun. And this is what it looked like there. It was really cold. And inside it, they had these awesome tanks called mesocosms. And these are massive. They're like 5 meters high. So each one holds 2 tons of water, and I could pump them up full of water from the Baltic, and I could set up different mixing conditions inside the tanks to see how the phytoplankton reacted. So I ran this experiment for like 2 weeks, and then I would take samples of phytoplankton at different depths, and then I'd use the coolest machine ever, right? It's this thing called a flow cam. And like I said before at the beginning of my talk, I'm not a proper plankton biologist. I never did biology. Don't tell the Marine Biological Association, okay? So I never did this, right? So I was like, well, I don't know how to identify different plankton. And so I was there with a microscope and it was taking me ages. And then someone from the Marine Biological Association said, have you seen this thing called a flow cam? What you can do is you can just pour your seawater in the top, and every time a plankton cell cuts a laser, it takes a photograph of it. So whereas this is it happening live, so whereas I was counting them, I could do 300 in like an hour and a half. This thing, I could photograph 6,000 plankton cells in 10 minutes. I was like, what? This is crazy. And then you build up these big image libraries on your computer of all these different plankton cells. You can sort them all out. So whereas other people have got like nice photos of their holiday and stuff on their computer, on my laptop, I have got, and I'm so proud of this, 462,000 images of phytoplankton. And because I've got all of these things and they're digital things, I can analyze them to see like, okay, did the round ones do well? Did the long ones do well? Did the ones that could swim do well? Did the ones that could float do well? And so I was able to go through and kind of tease that apart. And it was a really cool experiment. So again, I showed you at the beginning of my talk, that illustrator, Scott Masson. He was really excited about my research. He actually made this picture with the mesocosms and some of the phytoplankton species that I saw in there, which was really cool. So thought I'd talk a little bit about some of the species. Now, as I said before, the funny thing about being someone that knows about physics, but doesn't really know anything about biology, but I have to pretend that I know things about biology, is I had all these photographs and I didn't know what the names of them were for ages. So I was like, okay, now I know they're called Periduniella and Pauliella and things like that. But at the time, I was like, well, I don't know what these things are, but I need to start until I can send the pictures to a proper plankton taxonomist who can tell me what these are, because, you know, it wouldn't stand up to scientific scrutiny, me doing it. So I called them things like centipedes and microchips and DNA and canoes, things like that, which I thought was, and so me and my supervisors, also physicists, would be like, well, it looks like the canoes are going up, but the microchips are going down and things. So it was, I think what I like about this is there's this idea that scientists know everything about everything. and you don't at all. And you kind of have to make it up a little bit as you go along. So I know a lot about these species, the species I study, but if I'm honest, I don't really know that much about other species outside. I kind of look at how phytoplankton on the whole behave. So I thought I'd show my favorite plankton ever, and this is also one of the ones that I had. And this plankton just blew my mind as a kind of physicist that didn't know anything about plankton. Okay, so it's called Mesodynium rubrum. And from the top, it looks like a sunflower. I was like, that's cool when I was looking at it under the microscope. But then from the side, it looks like this. And it's basically two balls on top of each other. And so what it does when it's just hanging out in the water, it's got 6 arms that it kind of pokes out like that. And that stops it from sinking. So it kind of increases its surface area to stop it dragging. If it feels something swim towards it, it can detect the pressure wave using that belt, that tutu that goes around the middle of it between the 2 balls. And then so it detects it. Then what it does is it wraps those 6 tentacles around the top half, and then it just flaps its tutu and it can jump. Okay, so bear in mind, this is something that is the thickness of 1 to 2 human hairs. And what this can do is it can jump 1.2 centimeters. Okay, so to scale that up, that is like us being able to jump up in the air and like high five an aeroplane that's jumping overhead. And you think about that we live in, air. This thing lives in water, which for it is as thick as treacle. So I was just like, this is absolutely mental. And then as I started looking into it more, I was like, what's this thing down the bottom here? That is a mouth, right? So when I ran my experiment, I put a filter on so I didn't have any zooplankton coming in and eating my phytoplankton, because I wanted to see how the phytoplankton responded. I didn't want to know about that annoying animal stuff. And what I was not expecting is the phytoplankton to eat each other. So one of the things that make a phytoplankton is they photosynthesize. They can use the sunlight to get energy to survive. When Mesodynium is first created, it doesn't have any chloroplasts, so it can't photosynthesize. So what it does is it eats another cell and steals the chloroplast from inside it. So this is like you being able to eat a lettuce and then absorb the chlorophyll into your skin, so you'd kind of go a bit greener, and then just standing outside in the sunlight and just making your own sugar. I was like, this is crazy. So I'm hoping I've got some videos here of it doing its thing. So here it is, much like Sally's videos. It's just hanging out and it can't see, and it uses that tutu belt to detect a cell swimming near it, and then it's just like, nah, mate, I'm gonna eat you. And then this one again, I like, there should be another video come up now, and I'm hoping, here we go. So this one, the stupid cell hasn't realized that it's going around. As soon as it hits, boom, there we go. It touched that tutu, and Mesodynium ate it. But again, before we knew that this was Mesodynium, we knew that it was eating things, and we thought they looked like sunflowers. So for ages, we just call these chompy suns. And so I still call these chompy suns. So on my bucket list is to get a Mesodynium tattoo. I have yet to do that. So there we go. So that's kind of cool. Thought I'd end just with a shameless plug. Christmas is coming up. And if anyone needs an awesome Christmas present, there's this book that I wrote that's just full of really pretty pictures and loads of cool stuff. And obviously, I try to talk about plankton as much as I possibly can in it. There we go. But anyway, I'm going to stop waffling on now. And this has been me, and I'm going to do some questions. And if anyone wants to get in touch with me after this, there's my email address, and you can get in touch with the website as well. But thank you.
Fascinating stuff, Russell. Thank you. And before we get into some questions, a reminder to everyone that the hashtag for this is YMB Summit 22 on Twitter and other social media. So get involved, and don't forget to tag at MBA UK as well. And Russell, delighted to see Plankton, my old friend from SpongeBob, cropping up there. I've always wondered what species Plankton actually is. They never quite define it.
He's a copepod, because not many people know that Stephen Hillenburg, creator of SpongeBob, was actually a marine biologist before he did that. So plankton is actually a copepod.
Love it. Love it. I think we've got time for one question, Russell. And the one I'm going to select is this question we got earlier, which was, do some plankton need different nutrients to survive?
Yes, so just the same way that all plants that grow on land need slightly different nutrients. Some need phosphorus and phosphates, and some need nitrogens, and some need silicon. So diatoms, which we'd heard about, they make this like big skeleton, big heavy skeleton out of pure silica. So they need silica to grow. So if there's a place where they don't have any silicon, diatoms don't do very well. Mesodynium, for example, that really likes phosphate. Most plankton need like a mixture of all of these. And what's quite interesting, there's actually a micronutrient, which is iron, which is needed in a lot of photosynthesis. So this took scientists a little while to realize, because they would go to the middle of the Pacific and they were like, well, hold on, we're measuring really high levels of all of these nutrients. So why haven't we got any phytoplankton growing here? And the reason is you need iron as a micronutrient. So there's places where you get big plankton blooms. One of the places is off the west coast of Africa. And there's plankton bloom there that's caused by Saharan desert sand, which is really high in iron, which gives it that red color. And that Saharan desert sand blows into the seawater there and causes phytoplankton to bloom. So there's one kind of school of thought that I'm not sure how scientific it is, but there's certain evidence that's pointing to this, that whale poo is really high in iron. So what whales do is they take iron from one area, and as they go around and swim around the ocean, they poop en route and effectively fertilize the ocean. So there's one school of thinking that potentially, because we hunted whales so aggressively in the last century, that have we reduced the primary productivity in the ocean by reducing the amount of iron in it? Well, there we go.
Fascinating. And again, Russell, a fulsome answer. I would expect nothing less. A brilliant presentation and always full of insight. Loved it and beautifully presented. And you're going to be back for our careers panel as well. So thank you very much and look forward to seeing you there.
No worries,
and
thank you.
Thank you. And we've got Fabiana back. Thankfully, the gremlins in the technology are behaving themselves. And I'm gonna, Fabiana, you got cut off as you were just starting to answer the question of the importance of studying immunology in sharks and rays. So if you wouldn't mind picking that up again, that would be great.
Of course, sorry for but I had a blackout, so it was completely off. Yeah, so as I was saying, the immune system is the basis to protect the host from any kind of pathogens. And sharks and rays, and also chimeras, are the species, most basal vertebrates to have a complete immune system, as we know in other vertebrates such as mammals or humans. They have both adaptive and innate immune system. So they are the basis or the initial of the immune system. So they are crucial to understand how the immune system evolved during the tree of life. So
amazing. So basically, this can tell us it's like looking back in time almost to understand how immune systems have
starts.
developed.
Yeah.
Absolutely remarkable. There you go. Well, a great answer to a very interesting question. We are going to have to move on, Fabiana. Unfortunately, we've just run out of time, but thank you so much for coming back to answer the questions, and thanks for your brilliant presentation too. So folks, we are coming to the end of this session now. The Sea Life Showdown is what's coming next. I know you're all excited about it. It's starting at 5 minutes to the hour. That's 5 minutes to 2 here in the UK, or 5 minutes to whatever the upcoming hour is, wherever you are. The session, as I think you know, pits one organism against the other, where scientists and young people will battle it out to have their species crowned the winner. And today, pitching for their species, we've got Jasmine for tardigrades, we've got Maz for Calanus, personal favourite, just saying. Alosha is going to be talking about sea sparkles, and Victoria about cyanobacteria. So we've got some exciting organisms, we've got some brilliant people. Come back for 5-2 and get involved in the Sea Life Showdown. But until then, have a nice break.
Sharks and rays. They are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Hi, I'm Sydney Harned, and I'm a graduate student at Nova Southeastern University's Save Our Seas Foundation Shark Research Center and Guy Harvey Research Institute. I am studying the genetics of scalloped hammerhead sharks in the Eastern Tropical Pacific, including the Galapagos Islands. Specifically, the Galapagos scalloped hammerheads are under tremendous threat from the huge amount of illegal fishing that occurs here. Hammerheads in the Galapagos form large schools of hundreds of adult female sharks, and people come from all over the world to dive with them. Unfortunately, these sharks are also a target for huge fishing fleets stationed right outside of the Galapagos Marine Reserve. Hammerhead populations have been declining rapidly in the past few decades, so it's imperative that we protect the habitats that these sharks frequent. But in order to do that, we need to understand their habitat use. That's where genetics comes in. I am investigating the genetic connection between the Galapagos adult hammerheads and hammerhead nursery sites along the Central and South American mainland coast. Nursery sites are shallow coastal habitats where pregnant sharks go to give birth and where their babies remain for the first few years of their life. By taking genetic information from adult sharks in the Galapagos and babies along the coast, I can build a map of where these sharks go and how they are connected. This can help answer important questions like, are the adult females from the Galapagos aggregation traveling to coastal nursery areas to give birth? We can also use genetics to investigate familial relationships and genetic similarities between these locations and scalloped hammerheads from other parts of the world. By answering these questions, I will have a better understanding of how this critically endangered shark species uses habitat in the Eastern Tropical Pacific, which will hopefully lead to more protections on these areas.
Sharks and rays. They are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Welcome back, everyone, and welcome back, Helen, too.
Hello, hi, I'm back.
She's here. She's in one piece, and she's still surrounded by plankton. Folks, don't get consumed. It's back, hands down, the most popular part of our 2020 and 2021 summits were the Sea Life Showdown. We're going to watch as two passionate scientists and two passionate young people battle it out to try and convince you that their favourite species is the most awesome. So following introductions, contestants are going to have 3 minutes to present their favourite species from the hidden ocean. And we're going to see who's going to win with votes from you. And we'll get into all the rules and details with Helen in just a moment. But another reminder before we do that, to use the hashtag on social media, YMB Summit 22. And just to give you a bit of the usual housekeeping before we get going, the programme and FAQs, I think, are down there somewhere. And if you've got any issues or something, you're stuck about something, you can go and check out those. The chat box is, of course, there as well, either next to us if you're on a computer or down below if you're using a mobile. But most importantly, that is where your voting is going to take place for the Sea Life Showdown, which Helen is going to explain to us.
Yes,
Right.
so this exciting time when we are all going to get involved, we're going to briefly meet our contestants this year, introduce them and have a quick chat, and then each of them are going to have 3 minutes, and I mean 3 minutes. We're going to cut you off if you take any longer. 3 minutes to introduce their favourite species. We might not cut you off. We'll see how we get with time. They will introduce their species, and then you guys at home are going to vote for which one you think deserves to win the title of this year's best species in the Sea Life Showdown. Now, the poll's going to come up at the end. I think that's right. So you'll have a chance all the way through the next break to have a think about it, put in your votes, and then we'll announce the winners when we come back in the final session. So if you're watching on YouTube or Facebook, sadly we haven't figured out how to get your votes to actually count towards that, but we do still really want to know what you think and who you think should win. So do keep sharing your thoughts on the chat, so we'll keep an eye on that as well. But for those of you who are on the online platform, there should be a poll that will pop up, as I say, at the end of all of this, and that's how we're going to collect your votes. I think that's it. Yes, I think if we're all ready, it's time to bring on this year's speakers for the Sea Life Showdown. Here they all are. Look at them all, beautiful faces from all around the world. We're going to meet them all now. Yes, who should we start with? Rory, who should we start with? Who should we get?
Let's go to Jasmine first. I've got a list here, and Jasmine's first on the list. So Jasmine, good to see you. How are you doing?
I'm good.
Are you ready to talk
Yes.
the talk for your species of choice?
I'm always ready to talk about tardigrades.
Say no more. No more reveals. No more reveals. No more
Oh
reveals.
no!
You can't get
Spoiler.
no extra campaigning time. Now, Jasmine, am I right in thinking you are a postdoctoral fellow at All Souls College at Oxford?
I am.
Great.
Yes.
Can you tell us a bit about your studies?
Yeah, so I am a biophysicist, and I think about how things move through complex environments. So animals can move through lots and lots of different environments, and they have to deal with lots of fluctuations in their environment. And I think about all the ways that they're able to do that and how we can use that understanding to help species that are dealing with more and more changes due to climate change, and how we can use the knowledge of how animals do it to build robots and synthetic systems that can do that for, say, search and rescue.
Wow. That is some remarkable stuff. Totally fascinating. And thinking about the leaps between the natural and the technological there as well. It feels like it's growing and growing, right?
It's growing, and we're learning learning a lot in both directions. I think we can learn a lot from robotic systems about what biological systems are doing, because we can develop more simplified things where we can test things in the lab that we wouldn't be able to test on animals. And biology gives us a lot of inspiration for the robotic systems that we can build. So it's a kind of mutually beneficial collaboration, I think.
Love it. A two-way street.
Yeah.
Helen, anything you'd like to ask Jasmine before we move on to our next
Fantastic.
competitor?
No, I think let's move on and bring up Maz, who is our next contestant, our next presenter, I should say, speaker for this section. Maz is a senior plankton analyst at the Continuous Plankton Research Survey, which we heard about this morning from David. So welcome, Maz. Thank you so much for joining us. Tell us a little bit about how you're involved in the CPR and all the wonderful stuff that David was telling us about earlier.
Thank you. Hello, everyone. It's wonderful to be here. Thank you for inviting me. Yeah, so I'm senior plankton analyst, and what on earth does that mean? Well, unlike Jasmine, I'm just solely to do with animals. I just look at just creatures, which sounds, well, just creatures. It's a little bit old-fashioned. I guess what you call me is a taxonomist. So I focus on how to identify things and how to identify things properly, which is a little bit tricky in the plankton because, of course, they're so tiny and there's thousands of different species and they all look pretty similar, but they all do slightly different jobs. So I help other people learn how to identify plankton. And I teach it here, and I make sure that everyone here is doing their job properly too.
Fantastic. And taxonomy, I mean, it's a long word, but it's a very important part of science and marine biology, isn't it? It's an ancient science that's been going on for hundreds of years of identifying what's out there. But I guess it's becoming, we've got a lot more tools available now to do this kind of thing, and especially identify these teeny tiny creatures that you're dealing with. So it must be changing, I assume, and bringing it into the 21st century and using all sorts of exciting technologies as well, I assume.
Absolutely, yeah. So classically we use our microscopes, and as you just said, they've been used for hundreds of years, and we still use them. They're still an incredibly important tool in species identification. But now we've got the bonus where we've got new technology, and we can use things like molecular tools, looking at genes as well, to help identify creatures. We can even look at things called stable isotopes, the chemical makeup of different organisms, and we can tell by looking at that what they've been feeding on. It's fascinating, really. New tech has opened up a whole new world for us in terms of species and species ID.
That's wonderful. And just one word, like a shout out for taxonomists, because I get the sense that maybe this is something that we need more people involved in. It is such an important area to simply know what's in the ocean and what lives there. Is this something that you need to bring in new fresh blood for, and you need new enthusiasm for people wanting to do this kind of work?
Absolutely. Taxonomy, it sounds quite boring, but actually I get to see hundreds of different animals every day, and I've been doing the job 20 years. I'm still learning something new every day. Without good taxonomists, we can't understand biodiversity. It's as simple as that. And we all know how important biodiversity is in keeping the planet sustainable.
Absolutely, Maz. Thank you so much. Brilliant introduction to taxonomy. Rory, back over to you for our next speaker.
Thank you, and pleased again for another shout out for the continuous plankton recorder. I need an air horn for every time I hear CPR. Here we go. So next up is Alosha. Alosha, I love your t-shirt. If you could show everyone your t-shirt, it lets us know that you are a young marine scientist, and indeed you are a young marine biologist member as well, aren't you?
Yeah.
What is it you love about marine biology and marine science?
So I love to do about sharks. It's like the most interesting thing for me. And so not only that, basically everything in the ocean interests me.
That's a good start, isn't it? Liking everything in the ocean, because you're in good company here. We all think everything in the ocean is great. Of course, some of us have really nerded out on different aspects of that, and some of us, like me, have gone a little bit above the ocean to look at birds and stuff, but they still rely on the ocean fundamentally, seabirds. Do you have, apart from sharks, if I say, right, sharks are to the side for one moment, do you have a favorite ocean creature, except from the one that you're going to talk about today?
It's a shark.
What shark? What shark species is it?
Let me think. It's a great white.
Great white, classic, sort of the quintessential predator, isn't it? I love that. And you are indeed a global youth shark ambassador, is that right, Alosha?
Yeah.
What does that mean?
So, for the shark, I just spread information to people about sharks and why they should be protected, and they are not man-killing monsters, which everyone thinks they are.
They're wonderful creatures, aren't they?
And I'm currently doing research about Sri Lankan river sharks, especially the pond and cherry shark. And I have published a research about bioluminescent sharks in the deep sea.
Wow. Alosha, when I was your age, I'm not sure I had done a 1/1,000,000th of the things you've achieved for the ocean. I probably hadn't even really thought about what the ocean was. So we need young people like you doing great things, publishing research, looking at bioluminescence in deep sea. I mean, remarkable, absolutely remarkable. So it's a pleasure to have you to represent the young people and the young contingent. I'm wishing you all the best as we move into the contest shortly. And I'm going to hand back over to Helen to introduce our final. We keep saying contestant, not contestant. I don't know if the creature is the contestant or the person.
It's a good question, actually. Our final speaker, we can welcome in. We're very pleased to have with us another fantastic young marine biologist member, Victoria Huber. Welcome, Victoria. It's lovely to have you with us.
Hello, I'm super excited to be here today.
Fantastic. Now, tell me, you are also an ambassador for the Marine Conservation Network. Tell us a little bit about what that is.
So along my journey of learning more about the ocean, I help talk about Marine Conservation Network and spread awareness about the organization. And then in turn, that organization helps more people find me. So it's kind of like a mutual relationship. I'm also an ambassador for Earth Echo and the American Shark Conservancy. So it's essentially the same thing for both of those organizations. But what's really cool is that in Earth Echo, since I'm a water challenge ambassador, I test water monthly, and we get to use those results to help scientists learn more information about what's in the water.
Fantastic. And whereabouts are you speaking to us? And where is the water that you're studying? Whereabouts in the world are you?
So I'm in Florida. The body of water that I prefer to test is called Turnbull Creek because it's near the Indian River Lagoon, which is really important to me. I attended a really nice training about surveying seagrass populations. So that's why I want to make sure that water is healthy because it's going through some trouble right now.
Right. Yes, as many coastal waters are, and there's all sorts of issues with all of. We were hearing earlier about harmful algal blooms and some of the troubles we get if there's the wrong types of algae and plankton in places where we don't want them. And Victoria, you're a real passionate advocate for ocean literacy. Tell us a little bit about how important for you this is in terms of getting other people to know about the ocean and to care about it.
So the entire planet relies on the ocean to survive, and if we don't inspire the younger generation to care about it, no one's going to be willing to protect the ocean, and there's no chance. So really the people who are important to the survival of our planet are the youth, because we can inspire them and then they're going to fight to protect that ocean like we need to.
Fantastic. Exactly. And that's what this summit is all about as well. It's bringing like-minded young marine biologists together, and we can show that we are a big group of people all around the world, and we can add to that, and people can join us, and we can do all we can to help this wonderful ocean of ours. So fantastic. Well, thank you, Victoria. Welcome to the Sea Life Showdown, all of our wonderful speakers. I think it's time for the big challenge to begin. Don't you think, Rory? I think
absolutely. It's time. It is time. Let's get moving. So first up, we have Jasmine. She's trailed a little bit her organism already. You didn't get any information on it. Jasmine is going to be first. Jasmine, you have 3 minutes to sell your creature for the Sea Life Showdown. So let the fun begin.
All right. Hi, everybody. I am going to be selling the tardigrades. So tardigrades are an entire phylum of microscopic organisms, and you might have heard of them being called water bears, and you might have seen them in the news recently because they are able to survive in lots and lots of different environments, in high radiation and high temperatures. And they were even in the news a couple of months ago for crash landing on the moon. And I think that that's amazing. And it's part of the reason that they're really, really studied a lot by a lot of scientists. And the reason that they're able to do this kind of amazing survival in both Earth and space is because they have the ability to go into a hibernating state called a ton. And the way that they're able to do this is they can turn all their proteins into dehydrated forms and then just hibernate for up to even hundreds or maybe even 1,000 years and just wait for conditions to get better. But I also think it's really, really amazing how well they can survive in their day-to-day life. The tardigrades, like I mentioned, are a phylum, and there's over 1,000 species of them, and they're found in every ocean on Earth, in every lake, in every puddle that you see. If you just scoop up a little bit of water, you can find a tardigrade. And what's amazing about them is, despite the fact that they're surviving across all of these different environments, their body plan is pretty much the same. They all kind of look like little lumbering bears, and that's why they're all called water bears. And they've looked this way for tens and tens of millions of years. So this is a fossil that was from about 15 million years ago, and you can see the body plan is conserved. So they found what they like, found what works for them, and they've stuck with it for a really, really, really long time. And what this means is that they're really great to study in the lab because they're so hardy, and because the body plan has been conserved for so long, we can learn a lot about how things walk and how ancient things that no longer exist might have walked, because they've been doing the same thing for so long. We can understand a lot about what the world, how things used to move through the world a really long time ago, and how arthropods and other things that are able to move through land and water have evolved and diversified in all the tens of millions of years since tardigrades showed up. So not only are tardigrades a great species in themselves, they're also a little porthole into a lot of the diversity that we have both in the oceans and on land.
Oh, hi. Okay, so Calanus the copepod, here he is. Why should you vote for Calanus? Well, this little dude, here he is. He tells us a huge amount about the health of our ocean. He provides food for, well, a wealth of different organisms in the sea. And he's linked to, well, a British institution, really. It's what makes us British. I'm not talking football. I'm talking fish and chips. Much more important. So, Calanus the copepod, what is it? Well, it's a microcrustacean. It's a few millimeters long from head to tail. They're part of the plankton. They're small, but they're found in huge numbers. In thousands, these swarms can form thousands, millions, in fact, in a swarm. And they're found from the surface of the ocean all the way down to the deep sea to 1,000 meters. And not only that, they occupy every ocean in the sea. Every ocean on the planet. So that means they occupy a huge amount of the marine pelagic realm. They're incredibly abundant, incredibly important. Not only that, but they're veggies, they're vegetarian mostly. That means that they're the link between phytoplankton, the plant plankton, and much bigger, higher organisms. So these guys will feed on the plant plankton like the grass of the ocean. They get nice and fat, and we can see this little guy, that tube inside him, that's a tube of fat actually. They can be up to 80% fat. They're so nutritious that they're food for lots of different organisms, including baby fish. Of course, baby fish in turn grow up to be bigger fish, and bigger fish are then food for, well, things like tuna, sharks, whales, dolphins, all sorts of organisms are completely reliant upon Calanus for their diet. Some animals will feed directly upon Calanus, so things like big baleen whales, even some little seabirds will feed directly on Calanus too. So they're incredibly abundant, incredibly nutritious. So nutritious, in fact, that even humans, us, we've made a food supplement from them. Yes, you can buy Calanus oil. Who knew? Anyway, back to the ocean. So they're incredibly sensitive to temperature changes. This makes them excellent indicators of change, not just climate change, but at a planetary level, we can see changes happening. And these guys reflect the changes in the ocean. They're sensitive to temperature. So some of them like colder waters, and we've seen some of them have to move further north in distribution to find the colder waters where they're happy. And this means then that other animals that feed on Calanus, well, either they have to move too, or their populations decline. And we've seen that. We've seen a couple of examples. So the northern right whale off the east coast of the USA, Their populations have declined, and it's because colonists have had to move further north out of the area. They can't feed on them anymore. Same thing around Europe. We've seen fish populations, cod in particular, decline because colonists has had to move further north to find the colder waters. So colonists can have a huge impact on other marine life. Well, why should you vote for colonists then? Well, I think they're cute, and I'm sorry the video isn't playing, but if you could see them moving and twitching around, they're incredibly cute. But not only that, they tell us a huge amount about change in the ocean. They tell us about global planetary changes that are happening. They're food for thousands of different types of organisms out there. Without colonists, there would be no basking sharks, there would be no tuna, no dolphins, no seals, no penguins. I could go on. Actually, without colonists, there would be no fish like cod. Well, no cod, no fish and chips. Oh dear me. No fish and chips. Well, what would the British do? On a tea time on a Friday and Saturday night, it's a British institution. No colonists, no cod, no fish and chips. What would we do? I don't know. One thing left to say, and that's thank you. And thank you, Callinus de Copepod. Well, just for sustaining life in the ocean. Fish and chips. Anyone feeling hungry? Thank you.
Okay. Well, okay. Have you ever seen the beaches glow at night? Like they've been enchanted with some sort of spell? If you have, you've seen the sea sparkle, or known as Noctulia scintillans. One of the wonders of the ocean is a little creature called the sea sparkle, or known as the Noctulia scintillans. Noctulia scintillans is a single-celled organism known for its ability to bioluminesce. They can exist in a green or red form. Sea sparkles are dinoflagellates, and dinoflagellates are the foundation of the food chain. Dinoflagellates are usually considered algae and marine plankton. Sea sparkles range from 400 to 1,500 micrometers in length. There are two colors, red and green. The red form is heterotroph. The green form has a photosynthetic symbiont inside called Pedomonius noctiliae. We don't know if Noctulia scintillans is a plant or an animal. They contain no chlorophyll but have the photosynthetic symbiont inside, which makes it a little more confusing. And they have the characteristics of animals. They feed on smaller phytoplankton, zooplankton, and fish eggs. These guys propel with flagella. One is large while the other is insignificant. These guys don't need to use their flagella because they move with the current and don't really swim. They manage their buoyancy by having their bodies like a gas bag or a balloon, so they float right on the surface of the ocean. So, how is their bioluminescence made? First, luciferase and luciferin combine with oxygen to create an oxidized complex, which then the luciferin produces a photon, which makes light. Not only a bright flash confuses predators, it also attracts larger predators which scare off the original threat. So sea sparkles look like they're out of this world in a dream. They look like magic, but it's all real, and it's all because of sea sparkles. Now you know about sea sparkles. Thank you for being here to listen about sea sparkles. And please help save our oceans to help creatures like sea sparkles and many other animals. Thank you.
My apologies. Get ready to meet the coolest cyanobacteria in the ocean, the Prochlorococcus. Next slide, please. Every 1 in 5 breaths, you can thank the Prochlorococcus because it's single-handedly responsible for up to 20% of the oxygen in our biosphere. And when you consider that the fact that the ocean that covers 70% of our planet produces 50 to 80%, that's a huge chunk that we would not be able to live without. It also forms the entire base of the ocean food web, which us on land rely on. Since it's the most abundant photosynthetic organism in the ocean, it's the primary producer of the seas. That means it forms the bulk of most of the first trophic level of the food chain. It's also super small. It's the smallest cyanobacteria in the ocean, and you can line 100 of them up and only be left with something around the width of a human hair. But they're super abundant. There's around 3 octillion Prochlorococcus in the sea. That's 3 followed by 27 zeros and the number you see on screen. And if you weighed all of the Prochlorococcus in the sea, you would be left with something around the weight of 220 million Volkswagen Beetles. And they play an important role in this thing called the carbon cycle. As carbon is absorbed during the photosynthetic process, it's then passed throughout the food chain by predators eating prey. When a predator dies and sinks to the bottom of the ocean, it takes the carbon in its body with it. And since the Prochlorococcus is the most abundant photosynthetic organism in the sea, it is essentially what the entire process hinges on. And without it, the ocean wouldn't be able to absorb so much carbon, and our climate would be in a much worse place than it is right now. Next slide, please. It's also thought to be responsible for evolution. Most people know about the Cambrian explosion, where we had the first explosion of life in our oceans. But before the Cambrian explosion, all we had on our planet was prokaryotes, and that's what the Prochlorococcus is. Billions of years ago, the predecessors to its cells had mutations, allowing them to first discover photosynthesis and put the first amounts of oxygen into the atmosphere. This formed the building blocks for life on the ocean and life in land to take shape. So you should vote for the Prochlorococcus because your existence depends on it.
Wow. Helen, what to say there? It's
I
very...
can't say anything. Have my microphones off. I'm speechless. Literally speechless.
You are technically and actually and in every way, figuratively, literally speechless, unable to comment. Any immediate thoughts having seen all that? I mean, I don't want you to say which is your preferred, but any thoughts?
Well, I'm glad you haven't asked because I haven't decided, because how can you possibly decide? But we want you to. So quick recap. I mean, we have so much to choose from. We have the extraordinary tardigrades, the water bears, which is such a big favorite. I mean, goodness me, anything that can go to space, freeze, that's been around for that long and just hasn't bothered changing in its evolution, just sticking on with that cute little bear shape. I mean, come on, that's just wonderful. So that's a very strong start from Jasmine.
That's compelling, but then Maz came along and she was leaning heavily on the fish and chips angle. And I'd like to note as well that both the adults in this conversation were doing a lot of pushing of the cute agenda, which neither of our younger biologists went for. So that was interesting to me.
That is interesting, isn't it? Yeah, maybe we should think more on that. Yeah, that we should leave that behind, perhaps. I don't know, I still do love the cute, but it is not all about cute, definitely. And I am feeling a little bit hungry after all this talk about fish and chips, but also, I mean, just the idea that these copepods feed so many other animals. I mean, that we wouldn't have basking sharks and whales and dolphins and tuna and everything else that these incredible crustaceans feed. I feel like they all just kind of tie into each other there. We could probably make some great food web just out of all these animals and all these different organisms we've been talking about just now. So how to pick? So we have the food web, the mid part of the food web, I guess, is our Calanus, and then we hop down to the bottom to the producers, the sea sparkles and the Prochlorococcus as well. So those also very important and still very beautiful creatures, I think. So
Absolutely. I mean, truly beautiful. Sea sparkles definitely bringing a bit of magic. And
they
I mean,
are. Have you ever experienced that yourself, Rory?
I have not. I'm very jealously watching some of those videos and feeling very left out. Have you, Helen?
I have, yeah. One of my favorite experiences, actually. I mean, I've been very fortunate to be out at sea at night quite a bit and seeing kind of just the movement of the water in the bows and so on. But I've also done some dives at night when there was, I don't know if it was sea sparkles or another type of bioluminescent plankton, but when you agitate the water, so you can turn yourself into like a magician, really, and you can just like throw bolts of light by moving, which, you know, or just like stir up the water in front of you and it just glows, which is wonderful. And then actually one time I did that and I came back out after the dive, and it was clearly something had landed on my skin and settled, so that if I just touched, like drew a finger across my skin, it glowed. Like I was covered in magic glowing paint. It was really, really cool.
That's great. I mean, I've not had an experience like that. I did see a swarm of Calanus copepods off the Western Isles of Scotland. I watched the food chain occurring. I saw the copepods. I saw young sand eels out gobbling them up. Then I saw an Arctic tern come along. It was fishing for sand eels just right in front of me. And then an Arctic skua came and was chasing the Arctic tern to get it to throw up its sand eels. So I've watched the food chain go right the way up before my eyes, right here on my doorstep in Scotland.
I love it. I love it. Ecology before your eyes. I was up in Shetland this summer and I was hoping to see the sand eels and I wasn't in the right place at the right time. So I'm going to just have to come back and keep looking. Yeah.
You must. And let's not forget Prochlorococcus that Victoria spoke to. And I think a special shout out for anyone able to repeatedly say Prochlorococcus and not trip over their tongue. So props for that, Victoria. Very impressive. And a compelling case there as well, Helen.
Absolutely, yeah. Don't let that long name put you off if you're thinking of voting for Prochlorococcus. There you go, I did it. Clearly, just an absolutely pivotal, important organism on our planet. Where would we be without it? Another extraordinary piece of the food web and our daily lives. It's hard to, I do find it hard to kind of really imagine what that actually means, that that 1 in 5 breaths. It's extraordinary to actually think that that's what's around all of us. Now, in this room right now, there is oxygen that was produced, by these extraordinary organisms in the ocean. It blows my mind. Definitely blows my mind.
And I think if people tell one thing to another person about the ocean, it's that we need the ocean to breathe. We
really do. We really, really do. And we need all of these organisms. So, I mean, goodness me, you guys at home have got a bit of a tough call on your hands to vote for just one favorite, but I hope that you will. We are going to launch the poll, I think, any minute now. So if you check out just the same way we've been doing the polls beforehand, I think look down on your device. I think that's where you'll find it. If you're on a phone, look to the side of us and it should pop up there. And you've got a bit of time though, so you don't need to make any rash decisions. We're gonna have a bit of a break in a minute, and so you can have a bit of a think. Will it be the tardigrades? Will it be Prochlorococcus? Will it be the copepods? Or will it be the wonderful sea sparkles? I mean, just such a tough choice. But the choice is going to be yours. And we're very excited. We'll be bringing back all of our wonderful speakers after the break. So to find out the results and have a quick word with the winner. And it's going to sound really cheesy, but you guys are all winners already, I have to say.
Oh, Helen, I love it. You're all winners.
Sorry.
But there can only be one winner.
So in 10 minutes time, we're going to be back at 2:40 to give you those results. Take your time, get voting, and we will see you in 10 minutes for the Sea Life Showdown winner.
Sharks and rays. They are ancient, enigmatic, misunderstood, and essential to our oceans. But these important animals are under immense pressure. As the Save Our Seas Foundation, we're here to help. Through supporting research, conservation, and education, we work to ensure a future for sharks and rays. The Save Our Seas Foundation. Together, we can make a difference.
Hi, I'm Sydney Harned, and I'm a graduate student at Nova Southeastern University's Save Our Seas Foundation Shark Research Center and Guy Harvey Research Institute. I am studying the genetics of scalloped hammerhead sharks in the Eastern Tropical Pacific, including the Galapagos Islands. Specifically, the Galapagos scalloped hammerheads are under tremendous threat from the huge amount of illegal fishing that occurs here. Hammerheads in the Galapagos form large schools of hundreds of adult female sharks, and people come from all over the world to dive with them. Unfortunately, these sharks are also a target for huge fishing fleets stationed right outside of the Galapagos Marine Reserve. Hammerhead populations have been declining rapidly in the past few decades, so it's imperative that we protect the habitats that these sharks frequent. But in order to do that, we need to understand their habitat use. That's where genetics comes in. I am investigating the genetic connection between the Galapagos adult hammerheads and hammerhead nursery sites along the Central and South American mainland coast. Nursery sites are shallow coastal habitats where pregnant sharks go to give birth and where their babies remain for the first few years of their life. By taking genetic information from adult sharks in the Galapagos and babies along the coast, I can build a map of where these sharks go and how they are connected. This can help answer important questions like, are the adult females from the Galapagos aggregation traveling to coastal nursery areas to give birth? We can also use genetics to investigate familial relationships and genetic similarities between these locations and scalloped hammerheads from other parts of the world. By answering these questions, I will have a better understanding of how this critically endangered shark species uses habitat in the Eastern Tropical Pacific, which will hopefully lead to more protections on these areas.
Welcome back. I hope you had a good break. And just any second now, we are going to reveal the results of this year's Sea Life Showdown, the very exciting part of the Young Marine Biologist Summit of the year. We are all on tenterhooks. The results are in. We have counted your votes. It's extraordinary. It's surprising. I mean, how did you choose, frankly? Just between all those 4 fantastic species, I don't know where I would have voted, but the votes are in. And we can now reveal who came in 4th place. I don't actually know. Do we have more pictures on screen? I can't remember if we're going to do it that way, but I can reveal that the 4th place goes to Calanus the copepod. Oh, there weren't enough fish and chip fans. Max, I'm sorry. Maz, I'm sorry. We didn't quite get it over the edge, over the finishing line there. But I mean, as I say, we're all winners really. But there we go. 4th place to Calanus. 3rd place, Rory. I'm going to hand over to you for 3rd place.
Yes, in 3rd place we have Prochlorococcus. 3rd place. Victoria, you made a great pitch. It's come above Calanus, but below the other 2. So, you know, and even with fish and chips, it didn't bump you off the 3rd place. So you're on the podium, but that means that we only have 2 left. Helen, where do we stand?
We have 2 left, and we have some shocking news. We do not have a single winner. We have joint winners. So in joint 1st place, I am very happy to announce that Jasmine's tardigrades and the sea sparkle. So first to you, Jasmine, congratulations. Yay. The tardigrades are, yes, they are cute. They're wonderful. They've been around for a very long time. Your immediate thoughts, do tell us, how does it feel to be the joint winner of this year's Sea Life Showdown?
Amazing, especially after Alocia's amazing pitch. I'd never known so much about sea sparkles before, so I was blown away, and I'm so happy that we get to share the podium, because I think both are such great organisms.
Fantastic. Yeah, I think it's a very fitting place for both of these wonderful creatures to be sharing together. I just like the idea of them sitting next to each other on an actual podium. I wonder how the size work out. We need to work that out, I think, between a tardigrade and a sea sparkle. But yes, and Alocia, I don't think we can. Oh, we can't see your camera isn't working. We've got a lovely picture of you, but congratulations, Alocia. Yeah. The sea sparkle is up there as well. Joint winner of this year's Sea Life Showdown. How do you feel? Tell us. How does it feel?
I feel amazing, and I really love sea sparkles and also tardigrades because they are just adorable, and they are one of the toughest creatures on the planet. So
love it, Alocia. And you said it's very appropriate. Everyone will notice that Alocia's screen is frozen, and it was black just a minute ago. We couldn't see anything. And Alocia, you said that felt quite appropriate, didn't you, for the sea sparkle?
Yeah.
Why did you say that?
Actually, 2 weeks ago, sea sparkles arrived at Colombo, and I saw them.
Oh, wow.
Very amazing.
In Sri Lanka, that is amazing.
Yeah.
Oh, that is very cool. Well, Alocia was saying it's kind of okay that our camera's out, because that is the environment in which sea sparkle shows its magic and lights up. So, and Alocia, you evidently lit up the chat here to get everyone voting for the sea sparkle. So, congratulations there. Great work. Well done to both our winners. Let's have a big virtual round of applause, and for all the competitors for making a great case.
You guys did such a great job. Thank you to everybody, and thank you all for voting and for putting in your thoughts on that. And I think two top winners, 2 runners-up. I think we couldn't have asked for more than that. So thank you again to all of our contributors, our winners, to Jasmine and Alocia, to Victoria and to Maz. Thank you so much for joining us and doing a brilliant job in this year's Sea Life Showdown. Fantastic stuff indeed.
It's so exciting.
It is. Wonderful stuff.
Thank you.
Thank you, everybody. And that was yet again a fabulous showdown. But we still have some more wonderful things to come. The summit continues, and we have one more session for you, which is going to be wonderful, just coming up in a minute. But before we do that, just want to flag up what we're going to have in a little bit later on, which is our careers panel. And we've got a wonderful panel of 3 fabulous marine biologists who've taken their marine biology and their science in lots of different directions, who are going to be coming on to answer your questions. So one thing to think about is just get your questions in now via the Q&A. So check out that box to the side of me. I think if you're online, I think scroll down if you're on your device. Get your questions in there. The people we've got coming back to speak to you are Russell Arnott, who you've seen earlier today, a fantastic educator and science communicator. So if that's something that you want to know more about, do fire your questions, think about what you want to talk to Russell about. We've got Karen Lloyd coming back. She spoke to us this morning by recordings, but she's actually going to come and join us live later, which is wonderful. So she's a professor from the University of Tennessee, specializes in microbes in extreme environments, gets to go to extraordinary places for her work. So she'll also come on and do a Q&A for us. So plenty of chance to get questions in to Karen about her work, about anything you want to know about extreme environments, that kind of stuff. And we also have Dr. Ocean. Jeanette Davis is joining us. She'll be speaking to us in just a few minutes. So you'll get to hear more about her work there. So keep your thoughts on questions you want to ask her, because we'll be inviting her back for the careers panel. She's a marine microbiologist, and she's worked on all sorts of very cool things, including discovering new drugs in marine organisms, a really important part of ocean research. So those are our panelists. Get your questions in. We also are going to launch a poll, another poll. It's going to pop up on your screens in a second, because what we want to know is what are your top questions that you would really love to ask our panelists. So I think that poll is open at the moment. So which of these questions do you really want to hear the answers to? Because as I know, I've been on this panel myself in previous years, and I've seen it last year, and we always have way too many wonderful questions. Not enough time, so we want to know which are the ones you really want us to get to. So do you want to know what your top tips are for applying to study marine biology? How can we protect the oceans? What's the best bit about your job? Fantastic question, I love that one. What skills do marine biologists need? All those sorts of things. So get your votes in now, and I think that's coming up right now, still going on that poll. And we can see quite clearly at the moment already, the winning one is your top tips for applying to study marine biology. So we'll make sure we get through that one. Quite a lot of votes as well for skills to be a marine biologist. Yep, a few more just popped in on that one. Yep, okay. So it seems like all of them are pretty high up there. But skills and top tips look like they're coming out top in terms of those votes. And hopefully we'll get to all of these anyway. Yes, how can we protect the oceans as young people? Another brilliant question. Okay, well, fantastic. Thank you all for your votes on that poll. We'll do our very best to get through as many questions as we can in that panel session, which is coming up a bit later on. So yep, don't forget to get your questions in on the Q&A. We're also going to be asking you for feedback about this whole day of summit and talks. And so we're going to be sharing a survey in the chat a bit later. And you're also going to get an email about this after the event. So there's going to be plenty of chance. What we really want to hear is what you liked about this summit, what was good, what could we do better next year? All that kind of feedback is going to really help us to carry on making this event as good as it can possibly be. So look out for that and let us know your thoughts. And I think it's time for us to carry on with our wonderful panels of speakers. Our topic this year, as you know very well, as I'm surrounded here by beautiful pictures of plankton, is the hidden ocean. There's aspects of the sea that we have to look a bit closer to see them ourselves. But when we do, oh my goodness, what do we find out about? Extraordinary things about how our ocean works. And we're going to dive into that a little deeper with our next speaker.
Okay, folks, speaking first in this session is Jeffrey Marlowe, who's joining me right now. Hi, Jeffrey.
Hello, great to be here.
Yes, great to have you. Jeffrey is an assistant professor of biology at Boston University, and his lab studies the links between microbes and their environment at deep sea methane seeps, salt marsh sediments, and in active volcanoes. That's a pretty cool lead-in, I'd say. Working with the Deep Ocean Stewardship Initiative, he's brought rigorous science to the UN's High Seas Treaty negotiations, so an important link between research and conservation there, and is also the executive director of the Ad Astra Academy, which encourages students around the world to pursue STEM fields. That is a very cool bio, Jeffrey. Now, you're going to be talking about deep sea methane seeps. Is this what got you first interested in our oceans, or did something else lead you down there? How did you end up in the deep sea looking at methane seeps?
Yeah, it was a little bit of a circuitous route. It actually started in very much the opposite direction. I was interested in space and trying to find life beyond Earth. And as you'll see, that sort of leads us to the deep ocean.
Brilliant. Well, I think rather than take away from that brilliant segue, I'll send you straight in. So, Jeffrey, looking forward to hearing all about it.
Great. Well, it is a pleasure to be here, and I am very happy to share a few minutes about our work on microbes that inhabit the deep ocean. So, when you usually think about biodiversity, a lot of people think about the picture on the left, right? This is a tropical rainforest, the best case that we think of, dense life on our planet. But the number of species that we see in that picture is, I don't know, a few dozen, right? There are a bunch of different trees, but that's about it. And then if we look at that picture on the right, that is an individual grain of sand from the ocean. And each of those tiny little green dots is a different microorganism. So on this single grain of sand, these researchers found about 200 or 300 different species. So if we are trying to think about how much biodiversity there is on Earth, what the whole range of different types of genes are out there, microbes are where it's at. So that's what my research group tries to do, is to try to connect things from this individual cell basis, these tiny little organisms that are about 1/1,000,000th of a meter across, and we try to connect them across huge scales. So here we have, these individual organisms that are 1/1,000,000th of a meter across, and seeing how they affect systems that are much, much larger. They live inside animals, they live inside us, they influence ecosystems on a local and regional scale, and obviously across the entire planet. So we're trying to figure out how microbes do this, how they shape the planet in interesting and important ways. And my favorite spot to pursue this question is at the bottom of the ocean. So this is a place where in many spots, there is fluid coming out of the seafloor. You might have seen this at places like hydrothermal vents or methane seeps, as we're going to talk a little bit about. And in all of these locations, there are fluids that have a lot of electrons coming out of the seafloor. And then there are the bottom water at the ocean that has very few electrons. So you can think about this kind of like a battery, where the interior of the Earth is charged negatively, a lot of electrons, and the ocean and our atmosphere is charged positively, so fewer electrons. And in that difference, just like a battery creates power, that allows for life to create its own power and to sustain itself. So we're going to focus a little bit now on methane seeps. That's the spot that I've done most work. And as it sounds, this is a place where a lot of methane is coming out of the seafloor and entering the sediments and rocks, as well as the ocean water. This is a quick sense of what some of these expeditions can look like. This is about 3-4 years ago when we were on a ship leaving San Francisco. We have a remotely operated vehicle, this robot that goes down over the side of the ship, down to the seafloor, and is controlled from a shipping container, essentially, a container that is plastered with video screens. It's the most fun kind of video game you could imagine, trying to control this robot as it collects precious, fragile animals, rocks, sediments from the seafloor and brings them back up to the ship. Once we have them, we sort of put them in bottles, we add different chemicals to see what exactly they're doing. We're very interested not just in which microbes are there, but how active they are, and how their metabolism kind of influences their environment, and then by extension, the entire planet through the gases that it can release and consume. What's really cool about these microbes at the methane seeps is that they can eat methane. And after they do that, they're able to turn it into rocks, which is amazing. It's essentially sequestering this very strong greenhouse gas, methane, into a type of rock that will stay on the seafloor for thousands of years. So that's what we're seeing here. On the left, you see a little weird chimney structure of carbonate rock that is formed by the microbes that we're seeing on the right. This is a electron microscope image, so very, very, very high magnification of these individual organisms. You see them clustered in these symbiotic clumps that are actually touching, directly contacting bits of iron pyrite that are conductive. So it's a very interesting combination of the microbes doing their metabolism, the rocks and minerals doing their electrical conductivity, and it all forms this symbiotic relationship that builds rock on the seafloor and allows these microbes to eat methane at an extremely high rate. So that is, the types of environments that we mostly focus on. And as Rory sort of asked, and I mentioned up top, this has implications not only for keeping our planet sustainable in terms of sequestering greenhouse gases, but also for thinking about the limits of life beyond Earth. And these types of methane-eating microbes could perhaps have been present on Mars hundreds of millions, billions of years ago. And some of these systems are also giving us clues as to what we might find on the ocean worlds in the outer solar system, places like Europa, Enceladus, where we have found these plumes of liquid and gas coming out of these geysers essentially on the surface of these moons. And based on the chemicals in these plumes, it seems like there are these hydrothermal systems perhaps on the seafloor of these outer solar system moons. So it really is kind of a study that spans all sorts of space and time, right? We're looking at tiny, tiny organisms at the bottom of our seafloor for clues of what could be happening or could have happened billions of years ago across the solar system. So I will stop our journey there and would be happy to take any questions that you might have.
Well, Jeffrey, thank you so much. And crikey is kind of the word that comes to mind, because it's remarkable, really. What can we learn about the surface of other bodies in our solar system from the very depths of the ocean? And amazing to think that we've got linkages between those two. So did you actually start then as somebody studying other bodies within our solar system and then got into marine biology via that route? And this is kind of a careers question, but is that really how it happened?
Yeah, I think I was always just interested in what life is capable of. I grew up in Colorado, and we would go up to the mountains, and I was just intrigued by nature. And Yellowstone National Park, these sorts of places where life somehow finds a way. And the limits of that are what I've always wanted to pursue. That someday might involve actual samples from beyond Earth, but for now, all we've got is what we have on this planet, and trying to find what those limits are has always been the goal, for sure. And that's led me down to the deep sea and a few other interesting spots.
And they've personally got black smokers in particular have got a special place in my heart. It's when I started to study marine biology, it's what the habitat that I mentioned in my interview for university. So I'm a big fan, though I never got to research them. So it was a pleasure to see your talk. One of the questions we've had in is about how international research in extreme environments is funded. Is that coming through space funding? How do you find money, Jeffrey? That's the question, really.
Yeah, I think there are kind of 4 routes, to put them in big bins. One is space. That's exactly right. Organizations like NASA fund some of this work to understand the limits of life on Earth and how it might have begun. Another is from sort of, at least in the United States, the Department of Energy, which is interested in how hydrocarbons could be processed perhaps into alternative fuels or different products. A third is just fundamental curiosity about biology, geology. That would be in our country, the National Science Foundation. And then, of course, there are private groups that are really interested in sort of exploring technology, fundamental exploration of just getting a sense of what's out there. So there's kind of a diverse stream of opportunities to make it all happen.
Got you. Someone out there in the chat with an eye on the moolah, looking for where we can fund this research from. So this is a great question that's come in. Do you think methane-eating microbes will be able to help with climate change? We've obviously heard about cow burps in particular, and methane being a really potent greenhouse gas. Could methane-munching microbes unlock some solutions?
Yeah, I think so. The spots that I've mentioned in the deep sea, a lot of that methane doesn't actually get into the atmosphere because there are microbes already eating it. So that's great. In terms of what's happening on the surface in rice paddies and in cow guts, they could play a role. That's a slightly different system that would involve slightly different microbes, but we know they are able to consume methane just out of the atmosphere. It doesn't even need to be that concentrated. So if we could even sort of harness that ability, enhance it perhaps by making these enzymes even more robust, I think it's a very promising opportunity.
Brilliant. And I'm glad that we've had the opportunity of our accents to explore the different ways to say methane and methane. It's
that is key as well. Yeah.
Somewhere across the Atlantic that changes, and I enjoy it a lot. Someone is asking a sort of careers-based question. They've applied to study in the US to Boston University specifically. They're asking if you could talk a bit about your process of studying marine biology in the US.
Yeah, so there are all kinds of different opportunities. So one thing that I love talking about when we go on these expeditions is that it involves so many different types of people. It's not just those interested in the tiny microbes. It's engineers who are controlling or building these robots. It's the mappers who are able to figure out where we're even going in the first place. So there are a wide range of paths to get here. In terms of the sort of fundamental science, it's often, doing graduate degrees. Postdocs often follow that. Perhaps you go into academia, perhaps you go into government-based research organizations. So there's really no one approach. And as long as you're sort of curious about this environment and have something to contribute across any range of those disciplines, there's a path.
Great. And this is something close to Helen's heart. I know she'd be annoyed with me if I didn't mention it, but people are very interested in poking around down there in the depths for rare earth minerals and metals that could be used for constructing things. Batteries in particular is of interest. This concern about deep sea mining, and given how little we know about some of these habitats, the risks that might pose. Could you maybe speak a bit to that and its links with the High Seas Treaty?
Sure. So that is a long discussion, but to quickly put a bow on it, yeah, so you're right. There are companies interested in mining deep sea nodules in particular that have a lot of different metals that could be useful for batteries and sort of more sustainable technologies. And we are still figuring out what's even down there, how they would be disturbed, what critical ecosystem functions they're contributing that we don't fully understand. And more importantly, we don't really know the timeline of their recovery. So it's certainly an open question, an active area of research, and something that is also very much impinging on the international policy world. So there's a little bit of a separation between what's happening in the water column and what's happening in the seabed, but there's a lot of discussion now of whether it should happen, how much it would actually influence both the environment, but also the economics of this green transition. So fascinating area of continued policy and scientific work.
Apologies for just throwing in that bombshell at the end, Jeffrey, but you dealt with it well. And, I didn't want Helen to shout at me after this. I have to big up the deep sea. Thank you so much, Jeffrey. That's been great. We're going to move on to our next speaker now. So I'm going to hand over to Helen. Thank you.
Thanks.
Thanks, Rory, and thanks for getting that question in. I do appreciate bringing it back to the deep sea. That's fantastic. Well, it's my great pleasure now to welcome our next speaker. We are going to start now exploring the world of new medicines in the ocean and what that can tell us. And we have with us Dr. Jeanette Davis, or Dr. Ocean, who's joining us today. Hello, welcome. Thank you so much for being here.
Hello, thank you so much for having me.
It's such a pleasure to have you here. We have a fantastic marine microbiologist with us in the form of Dr. Ocean, but you do a lot of other things too. And before we get into your talk, I'd love to hear a little bit about some of the other things you get up to in relation to the ocean. You've published books, you work on policy. Tell us a little bit more about those sorts of things that you get up to.
Yeah, sure. So I like to say that my 9 to 5 is that I'm an ocean policy advisor, which basically means that I use my knowledge and expertise to try to make good decisions with others around the ocean and managing the ocean resources. So that has been very rewarding. The last speaker kind of talked about policy and so many things that go into decision making. And so I do work with leadership at my agency as well as members of Congress to think about policies that are useful for managing the ocean and its resources. In addition to that, I'm also a best-selling children's science book author of Science is Everywhere, Science is for Everyone, and Jada's Journey Under the Sea. And these books are really designed to allow young children to explore science in a variety of ways and in a diverse way, and making sure that everyone feels included in the conversation of science.
It's fantastic. It's so important, as you say, to open things up and to make the ocean and ocean science open to everybody. And you're doing an absolutely wonderful job of that. And you're very kindly going to come back and join our careers panel. So if any of you watching are interested in learning more or hearing more about work in policy and making these changes in how we use the ocean, in education, writing books, all those sorts of things, then do stick around, and Dr. Ocean will be very kindly answering questions then. But I think now, without further ado, I'm going to hand the screen over to you and hear your wonderful presentation about new medicines in the ocean. Thanks.
Awesome, thank you. All right, guys, thanks again for this opportunity. We kind of talked about who I am in an introduction. My books are located there on the screen, but I'm just going to jump right into the conversation. All right, so the ocean is very important, and I think we already know that. You know, the ocean actually is a little over 70% of our planet. So that means that it's really important overall for our planet and humans overall. And one reason why it's really important is simply life. Also biology, right? And so what we know today is that when scientists are looking for life on other planets, the first thing that they do is look for a source of water, because life as we know it depends on water, depends on the ocean. Another important thing about the biology of the ocean is that it helps oxygenate our environment. And so we learn and know that oxygen comes from plant structures, but the very first plants that formed were in the ocean, and they were actually microbes, the things that you can't see without a microscope. And they are responsible for generating some of the oxygen in our atmosphere. So every other breath that you take, you can thank the microbes in the ocean for that. Another important concept of the ocean is weather and climate. So when you look on your phones and you want to know what to wear when you go outside, you can thank the ocean for that too, because it works with the atmosphere to produce weather and longer weather patterns over time called climate. Also, food is another important aspect of the ocean. In fact, over 30% of the world's population receives its main source of protein from the ocean. And then lastly, the thing that I don't think that most people think about is recreation and community and transportation. So if you ever look at your clothing or probably anything in your house, you'll see that it most likely wasn't made here in the US, and it was made somewhere in another country. And so we use the ocean for trade and transportation and really creating community around goods and services. But today I'm here to talk about one of the most fascinating parts to me of the ocean, and that's discovering new medicines and drugs that we can use when we're sick. And so if you think about medicines overall, it kind of looks similar to the picture that I'm displaying here, where you see a pill or liquid form. So when we're sick, this is what we're used to taking. But medicines are actually just chemicals. They're actually chemistry, right? And so the same way I talked about a microbe producing oxygen, which is on the periodic table, you know, when you think about elements on the periodic table, they come together to form chemical bonds and chemicals overall. So this is essentially chemistry. So medicines are simply chemistry. And when people think about medicines, they often think about medicines in a laboratory setting, right? You think about test tubes, you think about a flask, and that is somewhat true where medicines can come from. They can be synthesized or formed in the laboratory setting. But with the ocean being the largest and oldest habitat on the planet, they have the potential to produce so many different chemicals, right? It has a large biosphere, meaning lots of life there, and with life comes more chemistry. And in fact, most of the medicines that we ingest today actually is a combination of the precursor, meaning the chemistry, the actual structure may have been derived from something in nature and then manipulated in the lab. So it goes back and forth in terms of medicines overall. So as scientists, when we're looking for new medicines from the sea, we do it through a process called bioprospecting. And essentially just means looking at life in the ocean to find new drugs. So with this concept of drug discovery, the first thing you do is you want to collect your samples. And I always get asked the questions, well, how do you know what sample to collect? And the way I think about this is you want to think about the potential chemistry that an organism can make. And so here I have a picture of a coral reef environment, and it's lots of different organisms there. So how do they live together? We call that a symbiosis. How are they able to thrive and live together in this community? And they usually do that by communicating. And they don't communicate via talking the way you and I do, but they communicate through chemical cues. And so it's a known environment where organisms are going to constantly produce chemical cues, whether that is to mate, to eat, to communicate and really, organize themselves. And so that's just one example where you want to go into an environment and think about what are the possible chemicals that are there. So the first thing you do is you collect your sample. And let's say we now have fish A, and we want to know all the chemicals that come from fish A. So then we go on to the next part of that, and that's identifying the chemicals. So again, it's simply just chemistry. So we have fish A, and we want to identify all the chemicals possible in fish A. And then we want to take those chemicals and screen them against known diseases. And that's essentially screening for activity. And the diagram that I have here just represents different shapes, but let's just say hypothetically, each shape represents a known disease. So we can say the blue line up top can represent a virus, and the red triangle can represent cancer, and so on. So now, again, we have fish A that has all of these compounds, and we're now testing them against known activity. And if you're lucky with a lot of research and diligence, you can then confirm the activity from fish A. So essentially, we say that we've collected fish A from the coral reef environment. We found 5 different compounds. We tested them against 5 different diseases, and we found that compound B has anti-cancer activity. So again, this is a very concise way of thinking about this, but essentially, this is the process of bioprospecting. So here are just some examples of marine drugs in the environment. If my slide will go next. Okay, there we go. This is just a list of some of the organisms ranging from a marine sponge to sea squirts, which helps treat cancer, the compounds that has been extracted from there. But the last one I want to touch on is of a sea slug. And that's the research that I've done. Now, these sea slugs are not your typical sea slugs that you see in your garden, right? Not the nuisance sea slugs. These are actually very beautiful, sophisticated sea slugs called saccoglossans. And they get this name from the way that they use their specialized tooth. So it means sap sucking. And they use this specialized tooth called a radula, where they're able to puncture their plant-like structure and take up the chloroplast. And that's a process called kleptoplasty, which means stealing the chloroplast. They also can take up the chemistry, and this is a process called kleptochemistry, where they actively take up the chemistry of those plants. They're pretty widely distributed, but mostly around the tropical regions. So I was able to work on this particular sea slug, Elysia rufescens, in Hawaii. There's a picture of it there. And in the early 1990s, it was discovered that an anti-cancer compound was extracted from both the sea slug, Elysia rufescens, and its plant diet, Bryopsis. And this compound we call Kahala-F, which I will refer to as KF. Very promising anti-cancer compound. But as a marine microbiologist that studies the things that you can't see, my goal of this research was to say, what is the actual role of this chemical in nature? Like I said, compounds are produced, but they have a role in nature, whether that's defense, whether that's communication. So I said, what is the role there? And is it possible that it's bacteria that's actually associated with these organisms or some type of microbe rather than the organisms themselves that's producing this compound? And so essentially that's what we found. And just to walk you through this diagram, the dark green is the Elysia and the lighter green, you can see it's feeding on its plant-like diet there. And essentially those red dots are the microbe. It's an intracellular microbe. And what that basically means is that this microbe can't be found anywhere else, and it only can survive embedded in the plant-like diet. And so essentially the sea slug comes and hijacks this bacterium that produces these toxins and then uses it as a chemical defense. So this is just a really sophisticated, intricate way in which organisms come together to produce these chemicals that we can then use as medicines. So my last slide here, I just want to demonstrate some of the drugs that are currently on the market. So what I showed you was an example of a drug that's in clinical trials, meaning that they're still trying to figure out how best to use this compound. But these are known compounds on the market, dating back to 1969, where anti-cancer compound was extracted from a marine sponge. And a few years later, scientists went back and said, what else can we find from this marine sponge? And they found an antiviral compound. Also, there is a cone snail where a compound was extracted that helps fight pain, or treat pain rather. And then the last four there are all anti-cancer drugs. And so again, this just illustrates how the ocean is essentially this untapped and underexplored resource for marine drugs overall. So thank you for your time, and I'll be happy to take any questions. I can't hear you. Is that just
on
me?
mute. So
Okay, there you
sorry. Yeah,
go.
that was me. Thank you so much for a fantastic talk. Oh, it's getting near the end of the day, you can tell. No, that was so wonderful. I love that you talked about Sacoglossans, one of my big favorites as well. They're such extraordinary looking things. And the
Yes,
idea
they
that they take the chloroplast from their food and they put it in their skin, it's just mind-blowing. And we do actually have rather a good question about those guys, which is, what's the difference between nudibranchs and Sacoglossans?
Oh, that's a great question. I'm wondering if, no, I have some extra slides, but anyway, so they're both mollusks. Let's start there. They're both mollusks. They're both gastropods, right? And so they both fit into this clade of gastropods. Particularly Sacoglossans, what makes them unique is some of the features that I just talked about, that they have this ability to be specialists with their plant-like diets, right? That they have this specialized tooth, this radula, where they can puncture their plant and take up chloroplast, as well as chemistry. Nudibranchs are not known to take up any chloroplast. They are, however, known to take up some of the chemistry of some of their plant-like diets as well, or at least have interesting chemicals associated with them. But with both of them, all of them are known to kind of produce these interesting chemicals because they are slow moving, they are soft bodied. And so oftentimes they are hot spots for drug discovery because they have to think about a way and be creative of how they defend themselves because they don't have shells. So essentially they're different enough because of Sacoglossans being a little more specialized in how they feed and their association with their algal diets, but they are both mollusk gastropods that have interesting chemicals and that are really beautiful organisms overall.
I agree. So beautiful. And it's quite a confusing thing because we talk quite generally about sea slugs, and that's sort of a very general term for a few different types of these
Yes.
shellless mollusks, and and there's interesting things going on there. And as you say, these soft creatures that are so delicious and so beautiful that people other things want to eat them, so they have these amazing chemical defenses. But I have another question about another type of mollusk which you touched on, and another favorite of mine, actually, the cone snails, ones that do have shells, and they use their chemicals in quite a different way. And the question is, well, how do cone snails treat pain, and what chemicals do they use? So maybe you could talk a little bit about what's going on with these wonderful but maybe slightly deadly creatures.
Yes, that's a great point because going back to defense. So, and I always try to emphasize this, when we think about, medicines from the sea, it's exciting because we think about how the ocean could potentially be this way of revolutionizing medicine as we know it. But it's really important to think about what is the actual ecological role that they play. And going back to, like you said, some of these are very deadly. And so the cone snails produce, I think it's called ziconide, which is a really interesting compound that can be deadly, right? And it is used to deter their predators. But when used in the proper dosage for humans, it actually is really good for treating pain. And it has been known to be used for people who are really in severe pain. It kind of has like this numbing effect that it can have also if you step on a cone snail. But the impact here is that it's kind of using some of the numbing property while still getting rid of some of the things that potentially could be deadly. So I think it was the first slide or so, I had this arrow that pointed from the lab into basically our planet. And so with some of these compounds, and the cone snail is one of them, you can take the precursor, but then manipulate it in lab so you can make sure that you have just the potent amount that can treat whatever disease you want, but also get rid of the components that could potentially be toxic or deadly to humans.
Fantastic. Thank you so much for that wonderful explanation. We've got lots more questions, but I know we're bringing you back for the career panel, so I think we might poke some of them, shove some of those your way when we are talking a bit more about careers and work in marine biology. But for now, just
Sure.
a huge thank you for that wonderful introduction and discussion of the mollusks and what they do, and the fantastic medicines and all sorts of useful things we can find in the ocean. So thank you, Dr. Ocean, and we will have you back shortly.
Yep, thank you.
Hello again, folks. And of course, as soon as the word nudibranch is mentioned, the chat absolutely lights up. There's a lot of questions. I'm very pleased to see that. I know we're a bunch of salty marine nerds around here, but that's very encouraging to see all those questions come in. So, but like Helen says, there's plenty of time to chat to Dr. Ocean when we come to the careers panel, and I'm sure you can squeeze in some bonus questions that aren't career related if there's something burning on nudibranchs that you want to get in there. I am joined by Karen Lloyd, who earlier you saw give a fantastic presentation. Karen is an associate professor at the University of Tennessee. Welcome, Karen.
Hi, can you hear me okay, or am I broken up?
You're loud and clear, and you're completely visible also, so we're ticking all the boxes here. Now, Karen, you study microbes in deep oceanic subsurface sediments, deep sea mud volcanoes and cold seeps, terrestrial volcanoes and hot springs. That's a pretty cool little list of habitats there, if I do say so. Oh, I've missed some as well. Serpentizing springs, Arctic marine fjord sediments, and ancient permafrost. Geez, oh, I thought that was a cool list, and then I've added 3 more. Serpentizing springs, come on. Yeah. So that actually leads me neatly into the very first question for you, which is, what is the most dangerous place that you've been to? Which I think is a cracking question.
That's hilarious. I mean, I guess you have to look at danger. I mean, you could say that going inside an active volcano is pretty dangerous. You always take a risk with that, but of course you don't do it stupidly. You look and make sure that it's not actually actively smoking on the day that you're going in, things like that. Probably the most dangerous place that I've been is standing in a dry river just when the rains came in Panama, sampling a hot spring coming out of a rock, and all the rocks in the river were just getting the fresh rain for the rainy season, and they were sort of tumbling down on top of us as we're trying to sample. So that probably ended up being much more dangerous than volcanoes, actually. But I don't seek out the danger. I just go to where the samples need to be gotten and try to mitigate the danger.
Look at that. That's a laser focus and a very sensible answer. And I don't think many people will be saying it. You know, like going into an active volcano might be considered dangerous, but you just have to do it right. Really enjoyed that. Sophia from London has asked, would you say that we've explored space more than we have the ocean?
If you say space, then no, because there's just that's everything, right? Everything that's not on this planet. And there's so much that we don't know. The James Webb telescope pictures that have been coming back really have brought that into startling focus, that we just don't know most of what's out there. But there are surfaces of planetary bodies outside of the Earth that we do know much better than the seafloor, like Mars, for instance. And that's something I think about a lot, is that there are plenty of mysteries right here on our own planet that are just, to me, just as exciting as the mysteries that are outside our planet.
Yes, and not least because a bunch of them probably are the solutions to some of the vexing problems that we have today in terms of continuing to exist on this lovely green and blue space. Now, we've got probably time for one more question, I think, and then we're going to slide straight into the careers panel. And I have to ask this question because it's a cool question. How did you get to be able to research in such cool places?
It was gradual, you know. It's just a question of looking at what you can do in the moment and trying to do as good of a job on whatever thing it is you're doing. You know, I didn't set out to say I'm going to get a degree in oceanography, get a degree in chemistry, and then end up in a jungle and a volcano and permafrost and all these different places. It's just simply that in the moment, these things made sense, and I worked very hard, and then they happened.
Magic. Well, that's a good message to bring us into the careers panel. So we're going to get everyone lined up, but first we've got a little sting and we'll go.
Wonderful. Oops. I am on. Yes, I'm on. Hello. Sorry, I thought I was muted. So slick. Right. Welcome back, everyone. The final session we have today is really a wonderful opportunity for all of you out there to pick the brains of a wonderful panel of diverse and fantastic marine biologists who I'm very pleased to see. We've got arranged by the side of me right here. We've done our poll and we've asked you what kind of questions you want, which were your biggest question. And so what I want to do first, we're going to try and get through as many of these as we can, but I would like to go through each of our wonderful panelists in turn. If you could just briefly reintroduce yourself. We have met you all already, but just maybe briefly reintroduce yourself to us, and at the same time answer the top question which came up in the poll, which was, what are your top tips for applying to study marine biology? So perhaps quickly remind us who you are and have a think, each one of you, in terms of studying and getting that foot in the door in terms of getting those qualifications as a marine biologist. So Russell, let's welcome Russell back in. You were speaking to us a couple of hours ago. Lovely to have you back. Thank you so much. What are your top tips?
Okay, what are my top tips? So hi, I'm Russell. I research, my background's in physics, and I research how physics impacts biological systems, specifically how the physics impacts phytoplankton. My top tips about how to get into marine biology, I would say it's all about networking, okay? I think that at school, and that when you're young, it's sort of, you need the good grades, you need the good grades, you need the good grades. But because marine biology is one of those jobs that unfortunately a lot of people want to do, it can be very, very kind of oversaturated, and there's not that many positions available for the amount of students that want to do it. So I'm not trying to dampen anyone's enthusiasm, but what I am saying is that because it's quite a small community of people, it's very easy to talk to people and find people who are into the same things you are and talk to different people about things. And it's just about getting your name out there and coming to things like this and taking part in the chat and networking with us, finding out what we do, and then obviously getting in contact with us if something that we've said is interesting. All of these things are really good. So I would say, don't be afraid to put yourself out there, to ask questions, to send emails to people, and to be kind of bold and brave, and don't be afraid to ask for things. So.
It's a great message, Russell. I would absolutely echo that. Be bold and brave. Ask. Put yourself out there. It can be scary, but it definitely can pay good dividends. So yeah, definitely a good ideas there. Dr. Ocean, let's come back to you. What are your thoughts? Hello again. We've just heard from your wonderful presentation. Thank you for sticking around. What are your thoughts on that idea of applying to study marine biology?
Yeah, I want to second Russell, because I think networking is huge. It is about who you know, but it's also about who knows you. And so I echo, be brave and put your name out there. The thing that I would absolutely add, though, that I don't think a lot of people realize, and I think Russell kind of alluded to this point too, he studies physics, right? And so marine science, marine biology is a lot of science together, right? Like, the reason why I came to love the field is because I love science, and I had to be strong in chemistry and biology and physics and all of these other sciences. And so sometimes if you don't have necessarily a program next to you, because I grew up on the East Coast and I wasn't fortunate to have like a lot of the programs that are in California or Florida, but because I was strong in science, it allowed me to get into marine science, right? And so the other thing that I would add is that make sure that you are strong in your sciences, because believe it or not, it's still connected to the ocean environment. And so even if you can't find a program next to you, find the chemistry program and talk to the teacher or your advisor about trying to add a marine component. And so I guess the overall message is that your path is not linear. There's not just one way of doing it. Find the thing that you're passionate about and link that to marine science, however or whenever you can. And that goes to the point of being brave and asking questions and, letting people know your interests and you're up for creating and trying something new in the field.
That's fantastic. Thank you, Dr. Ocean. I really liked as well your point about networking isn't just about you meeting people, it's about people meeting you. I think it's a very good way of putting it. Karen, let's bring you in. Do you have anything to add? Any more thoughts on that first step?
That was perfect. Those are the two things that were in my head, really, was get a really good background in sort of basic science. I majored in biochemistry. It's, we don't come to this often as marine biologists, but then I always say that it's a prerequisite to be very good at what you do, but then after that, it's all about who you know and who knows you. And maybe just one tiny tip I can add is that when I was starting out, going to a big meeting where I knew that people wouldn't know me and they wouldn't know anything about me, I would always have a question preloaded in my head about their research in particular that showed that I understood their research and that I was curious in it and I had an insightful question. And I think that went a long way to sort of making me known. So you don't have to know everything, but knowing about what other people are doing and being able to showing that you can contribute to it can often make yourself known.
Yes, asking good questions is very important, and this is what we're getting you guys to do right now as well. So keep your questions coming in. We'll keep bashing through them. Rory, I don't know, would you like to pick out another? We've got lots to get through. What do you think? Where should we go next?
Yes, well, actually, the next most popular question, I think all of you have kind of answered already, which is what skills do you need to be a marine biologist? And you've talked about some of the academic, but you've also talked about some of the other skills that sit alongside that in terms of networking and speaking to others and ways of approaching it. So I think what we'll do is move on to another question that gets asked a lot, not just within the chat here, but outside of the conversation among the young marine biologists. It's this idea of work experience, right? And that it's often difficult for people to access work experience, right? And we know that lots of marine biologists have done other jobs as part of their journey. So it'd be great to hear about how you can explain the skills that you've learned, some of which you've described, in other jobs that have helped you get into the marine biology realm. And I'll start with Dr. Ocean this time.
Okay, that's a great question. I always, think about that when you think about applying to a position, it's not just the title or the status or the position. You're really saying, I have the skills to do this thing. And so you want to highlight the skill over just a position or a job description. And so the way that you can do that is, one thing that I always think about is leadership. Have you done any community outreach? Have you organized anything? If you are a student, do you demonstrate leadership within your student organization? Are you the president, vice president of something? In your church, in your local organizations, like all of these are skills that I would say are leadership skills, right? And so you want to be able to highlight those things. Collaboration, that's another one. Like, no one in the field of marine science works alone. And some of the speakers talked about that, even going on a boat. It's not just the marine biologists, it's the engineers, it's, all of these different people. It's the people who are actually just steering the ship, right? And so, collaboration and communication is another big important skill that you want to be able to play up. And I'm going to give you one last example, because I come from a huge family. I have 4 older siblings, 14 nieces and nephews. My mother is one of 12. My father is one of 8. And I always say that because I say to people, if I can communicate in my family, then I can communicate anywhere else. So you can even take personal stories and talk about your skills overall. And those are the stories, the storytelling you want to do and how you want to sell your skills overall.
That's great. Thanks, Dr. Ocean. And I very much relate to having lots of siblings. I'm one of 4 myself, and it helps bring your voice up for sure. Let's go over to Karen now. Karen, what are your thoughts there?
I don't know, other jobs that I've had that helped prepare me. I had other jobs that I really didn't like, so that helped me know that I wanted to do this other thing. I think it's also important to know that, like, when you look at the adventure aspects of being a marine scientist or, whatever environmental scientist, know that it's not all that. We do sort of the more boring sounding bits too, but you kind of have to prepare for both sides of it, and just kind of be prepared for everything.
That's a great answer. And the journey's wavy as well, right? It's not a straight line. Sometimes you're doing the exciting stuff, sometimes you're doing the less exciting stuff. All of it's part of figuring out what you find exciting, which might not be what the next person finds exciting, right? Russell, you've had a really interesting career path and followed some different routes. Great to hear your thoughts on this as well.
Yeah, I guess what experience have I brought to the table? I'll be honest, I've just kind of made it up as I go along. I wish I was a bit more like I meant to do this. I mean, I started off doing a degree in fine art painting and was just like, I didn't even do any science A levels. So that's a shush. Okay. So don't tell anyone that I'm a fake scientist. But yeah, I was a teacher for a bit, and I had all kinds of weird and wonderful jobs when I was at university, working in factories and things. And I think, to echo what Karen was saying, like, that really made me think, okay, I really want to make sure that I don't end up building keyboards or packing car seats or making exhaust pipe brackets. I did all these things. So one of the things that really helped me when I was at university, and I think this is an important thing, I think within marine biology, because there's not that much money available, I think as a community, a lot of organizations rely very heavily on unpaid internships and expecting you to give up your time for free. And there's a balance between getting experience, but also recognizing that that puts barriers in place, because not everyone has the time and the money to be able to be like, yeah, I'm going to go work for free and do volunteering. And I wasn't in that situation at all. And there was organizations that used to come to my university and be like, come and get experience. You only have to spend $7,000 and pay for your own flight. And I was like, mate, I can't afford that, right? So what I would do, again, this idea of networking, putting yourself out there, I couldn't afford the snazzy let's fly abroad and do coral reef stuff, and I didn't want to work in a factory. So what I did is I just emailed everyone in the Oceanography Centre, National Oceanography Centre, and was like, hi, I'm here, I kind of know about this. And every single summer I was able to get paid work as a research assistant. And all of that experience throughout my whole time at university meant that my first job that I got as an oceanographer from leaving university, I actually got because I'd worked with some PhD students who then left academia and gone to start become commercial oceanographers. So again, just kind of feeds back into, I guess, know your own worth and know your value and what skills you've already got, and kind of, you know, just be like, no, you should be paying me, you know, if you feel if you feel like that. But I mean, if you're in a position to be able to do it for free, then, you know, good on you. But also, again, just don't feel free to ask questions, put yourself out there, make contacts, and just be bold and ask questions.
Thanks, Russell. Yeah, that's really great. It's such an important thing that comes up again and again, which is, you know, how to get that experience, and there's lots of different ways to do it, and that's great. I want to pose another question, which I think we've touched on a little bit during the day, and I'd love to see what other thoughts any of you three have, which is either from your experience or from other things you've come across in your time working in the field, which is, well, the question is, are all marine biologists academics who do lectures and research? And I know the answer to that already is definitely no. So what other jobs are there in marine biology? Maybe I'll turn that, I think, first to Dr. Ocean, because I know you already do lots of very interesting different things. And I wonder if you could speak a bit to that idea of, you know, what can we do with marine biology out there in the world?
Yeah. So yeah, there's so many different things that you can do. I think obviously academic is one particular, I guess, path. Government, I work in government as an ocean policy advisor. That's absolutely a path. Government is always looking for people to help them problem solve. So that's an avenue. Entrepreneurship. You know, I'm a children's science book author, and that is definitely more entrepreneurial of me. I mean, I work with the publisher, but that was pretty much my idea, and working with an illustrator to come up with concepts. Also nonprofit, that's another arena that you can go into. Lobbying, like there's just so much you could do, from teaching. Consulting is another one that you can do. I recently just kind of got into consulting, and you can do consulting everything from like companies to actually like movies that use the ocean environment. So there's so many different things that you can do. And I think one of the great things about being a marine biologist is your role is to problem solve, right? And so there's so many problems to solve. And when I think about things like climate change and how we all kind of have to put our hat in, or put our, you know, our knowledge in to figure out something as big as climate change, I think that requires scientists, artists, social scientists, you know. So there's so many different things that you could do, and I probably made things more confusing. So I would say, what do you want to do, and lead with that, and that'll help direct your path.
That's fantastic. Yeah, I mean, I remember when I was setting out as a marine biologist, the thing I would get told by so many people all the time was, well, there's no jobs in marine biology. And you've clearly shown us that that's not true. And there's so many different creative ways of getting involved. Russell, I don't know if you have any extra thoughts, because you've obviously had a very creative path through all different sorts of things. Have you got any more thoughts on that idea of what marine biology is and where it can go?
Well, I think because you're doing science broadly, you can basically, you're kind of proving that you're clever to a certain degree to employers, you know, that's what it is. So just because you've learned marine stuff doesn't mean that you have to stay in marine stuff necessarily. Like the skills that you've got there, you know, working together, coding, report writing, you know, driving boats, like all these random skills that you pick up on route, you can apply them to different situations. And, you know, I mean, when I think back to the people that I did my undergraduate with and the jobs that they've gone on, and, you know, they've literally just done anything. Like they were like, I guess they saw it as like, this will be a fun degree and I'm just going to use that and as a foot in the door for whatever job I wanted to. I mean, I remember there was one girl on my course, and she was like, yeah, after university, I want to go and work in PR for a Formula One racing company. And I was like, so why are you doing oceans if you know that that is this? She was like, I just thought it'd be a fun thing to do. So yeah, so that just because you are studying about the oceans, it doesn't mean that you have to stay there. But I'd also say what has been really nice in recent years is the importance of the ocean in climate regulation has really, it's becoming more and more known. And with now the massive emphasis that's being put on trying to solve the climate and understand the climate, ocean, atmosphere system, the oceans are kind of being pushed into, instead of it being like, I'm a weird ocean nerd, now you're kind of a broader environmentalist and can take part in these conversations about bigger things instead of it just being stuff like ocean plastics, for example, which is like what we were kind of pigeonholed into before. So it's nice to be, I'm hoping, you know, we'll continue this trajectory and the oceans will continue to be seen as an important thing that more people, including politicians and stuff, need to know about. So.
Yeah, absolutely. Great thoughts there, Russell. Thank you. I'd like to come back to Karen. I don't know if you have any more thoughts on the kind of job aspects of being a marine biologist, but I also have another question, if not, that I think would be rather fun to ask you, as I know you are the active researcher very much in our midst. And the question is, how do you know what to study? So any thoughts on that, or any more job thoughts as well, if there are anything else on the
No, you guys did such a fantastic job answering that question. I have nothing to add. What they said. So how do you figure out what to study? That is a fascinating question. For me personally, it's a combination of what I'm interested in, which is hard because I'm interested in literally everything all the time, always, which is an impossible thing to narrow down. But then you look at what has traction, like what has, you know, a community, a nascent community that's starting to get interested in it. So for instance, there's, I don't know if you've been following much about carbon capture and storage, but we're not really going to be able to do much to mitigate climate change without actually pulling more CO2 out of the atmosphere. So decreasing our emissions is necessary, but we're going to need to actually draw some CO2 out as well, which is a much harder proposition than, you know, making fuel efficient cars, which is also very hard. But anyway, storing it underground is the big thing that I am getting interested in myself. And it's simply because there just aren't a lot of people working on this. So I personally try to look at a question that's relevant, that's tractable, that's interesting to me, that also is not already saturated with people. And so that's where I tend to move into, just intellectually. That's what I like to do. And then so much of it is who you know. Like, do you know the people who are doing this thing, and can you get access to this type of research? Can you get access to a drilling well? You know, how much proximity can you get to this stuff? But that's sort of how which is not, you know, there's nothing prescribed about like what I study or where I go. It's just whatever makes the most sense in a moment.
Thanks, Karen. And that leads quite nicely actually into one final question, which I think is particularly relevant for you. So I'll come back to you straight away on this one. What's the best way of becoming a marine scientist working in the field specifically? And what tips do you have for being able to travel whilst working? So notwithstanding everything we've said about, you know, taking the opportunities as they come and everything else, for someone who's really interested in doing field work and getting a bit of travel in there, what are your tips for that?
That's also a great question. And maybe this goes back to the jobs question. You know, there's a way to only do field work. If that's really your focus, we need whole people who live on ships, you know, really the support staff, which, you know, these other guys alluded to as well. You can do logistics for an Arctic station. You know, when I go to the Arctic, I don't, you know, bring all my stuff with me. I am going to a research station that has a permanent staff that live there. So that's something that you can do if that's really a priority. But then if you want to do things sort of how I do them, where I mix the science, you know, I'm writing the grants and I'm writing the papers and I'm doing the field work all together. Advice is to get really good at being productive on planes. in airports, you kind of have to just like, you definitely can't consider your travel time as your non-working time. That is like where you have to be productive, because otherwise you're going to get nothing done.
Absolutely. I can relate to that. Some interesting places I've worked, especially on planes with my knees up my nostrils being 6'3", slightly awkward. Folks, we're just about out of time for the panel. You've been amazing, such insightful answers, really helpful, I think really valuable for our young marine biologists gathered here today to hear everything you've had to say for those different experiences and the ways of bringing different experience to bear on how to get a career in marine biology, and all of your stories being so completely different yet so completely brilliant and important in the world of marine biology. So big thank you from us for joining the panel, and thanks so much for your great answers.
Thanks for having us.
Okay, folks. Well, that's us almost right at the very end of the day. We have 10 minutes left. Thank you for sticking with us this whole time. I hope it's been fulfilling, enjoyable, that you've learnt lots, that you found it compelling and interesting and everything else. I really appreciate all the interaction we've had with you. We come to the very end of the day and we are joined by MBA Director Willie Wilson. Willie, welcome.
Hi. Oh, what a fantastic day. I can't believe I've got to give a closing statement here because I don't want it to close. You guys have done such an amazing job. I've got several pages of notes that I want to get through here. I can't believe I've only been given 10 minutes to come up with a closing statement. This has been absolutely amazing for me. I mean, I've been a marine microbiologist for 32 years. When the team came to me and said they wanted to do a sort of hidden oceans theme, and they were a little bit nervous when they came to me. Do you think it'll work? I said, of course it'll work. It's what runs the planet. It's got to work. People have got to get an understanding for what goes on under the microscope. So for me, I've just been glued to the screen all day. So this has been amazing. I guess as a closing statement, I'm going to start. There's going to be like a bit like the Oscars a little bit. There's tons of people I need to thank, and I'll just rush through some of that. But first, Rory and Helen, you've done an amazing job emceeing today, just bringing all this together, just bringing the best out of everybody, which I think is in some ways been quite an easy job because you've had such a brilliant group of speakers that have been here all day. So that great segue on to saying thank you very much to all the speakers. There's every single talk I've absolutely loved, and I've got tons of new ideas from some of the things that have been getting said today. Also, these conferences or these meetings or summits can only really work if everybody gets involved, and there's been huge amount of involvement from people asking questions and then everybody answering questions as well. So thanks very much for participating in that way, because it just makes it a really sort of vibrant meeting. There's also I'm sat here in a sort of virtual green room. I'm just amazed at how many different people there are in the background here that are helping. And what you probably don't see, it seems really smooth at the front there. There's a production company that's helping us, the SSTV, and we've got Alex, Harry, Simon, and Joe. I'm not sure who it was. I had somebody speaking in my ear, sort of just guiding me through all this, and that's why it's been so smooth. So thanks for the team there. And then of course there's the MBA team, the membership team, and the comms team at the MBA, in particular Kathy, Lise, Mari, and Neri. I see they've been running around in the background over the last few months, and I can see this from my own office. So that's a huge thanks to everybody. So as a closing statement, oh my word, how do I sum all of this up without offending anybody or missing anybody out? I was trying to think of a way of sort of bringing all this together. I mean, there's a great analogy I use when I talk about marine microbiology. I often talk about, when I give talks, about the great engines of planetary control. And if you use that, and there's two big engines. One is the absorption of heat energy, which drives all the physical processes. And then there's the absorption of light energy, which drives all the biology in the ocean. So you get absorption of light, starting with the photosynthetic processes, and we've heard talks about the food chain, food web, and all of marine biology in the ocean derives from that. There's some chemolithotrophy. You could maybe argue maybe some of that doesn't come from there, but then you've got all this, what is it, Cordelia put it, the poop that sinks down to the bottom of the ocean. So there's it's all interconnected. But if you use that analogy of the engine, I love using the engine analogy, and quite often why do people get into marine biology? Because they love the whales and the sharks, these charismatic megafauna. Well, if you think of a Rolls Royce or a Mercedes, that's your charismatic megafauna, isn't it? But what is it that really makes a Rolls Royce work? If you lift the bonnet and take everything apart, you're going to get lots of nuts and bolts and cogs and the diesel or the petrol or the gas that helps make it run. That's what we're talking about today, is nuts and bolts that make the ocean, that really bring together the ocean. It's the bits that you don't even think about normally, the bits that you can't really see in your Rolls Royce. My favorite analogy, of course, is the oil that you put in the car. Now, why do you put oil in your car? It's because it's lubricating the car. It's the lubricants of the engine. So, and when I talk about the lubrication of the great engines of planetary control, that's when we start talking about the microbiology, the viruses, the plankton, and everything together. And that's what we've really been talking about today. And it's not thought about enough. And there's been some amazing talks today that have really made this sort of quite difficult, complicated, and quite frankly, critical subject much more accessible. So and everybody's done an amazing job. There was some great videos. I thought it was I forgot Sally. And now Sally had this video of that predator-prey dynamics. I remember as a young kid watching David Attenborough movies, and you sort of watching the lions going up, sneaking up on a gazelle, and everybody remembers that kind of thing. I wonder if way back then, if David Attenborough had something sort of shown these predator-prey dynamics at the microbial level, which is happening all the time, every day, all over the planet, all over the ocean. And if we got to see this on the television all the time, then maybe people would actually start getting interested in marine microbiology. And that's that kind of the part of our vision at the MBA, is just to try and make this type of really complicated work much more accessible. Another key word, and Cordelia reminded us of Charles Darwin's quote around adaptation. And I think adaptation is maybe one of the key words I think connects all the different talks today. We heard, you've got such a huge diversity of microorganisms that can survive in the deep ocean, inside the Earth's crust, yeasts in the Arctic, high diversity in tropical coral reefs. I'm sort of reading this from the notes that I took here. You got shape adaptation, kleptoplasty. I mean, these are all. Kleptoplasty is one of my favorite words, by the way. So thank you, Jeanette, for actually bringing that up. How do they adapt? We had Yasmin was discussing some of the molecular mechanisms, such as diatoms sensing the environment, and she gave an excellent talk that. This really sort of brought to light what is an incredibly complicated topic. So well done for really sort of making molecular biology accessible in that way. We heard a lot about some of the processes. Microbes are the ultimate recyclers. I heard the term microbial gardening today. You know, if you think of if you have a garden at home and you sort of throw all your waste in it, you can get all this organic waste, all the vegetables from, you know, a potato peel and everything. You throw it into the compost heap, and then magically a year later it turns into this lovely soil. It's inorganic material that provides nutrients. And that's what's happening in the ocean on a daily basis, and it's all been driven by the microbes. And we were hearing all about that. The other thing, I guess, is if you're going to take one thing home today, it's really about the sheer scale of it. And this was something mentioned by David in terms of the plankton, where there's plankton everywhere. It's the biggest kind of migration of animals in the entire planet that happens on a daily basis. When you've got zooplankton that go from the depths right up to the surface, you know, they're in the depths during the day, and then they can travel about 200-300 meters up to the surface. And for something that's about 1 millimeter long, that's just phenomenal. That happens all at the same time. It's the biggest vertical migration. So the scale is massive. You have phytoplankton all over the ocean who are doubling, the productivity doubles on a daily basis. It doesn't keep doubling and getting bigger and bigger and bigger like a tree would, but as it doubles, then that gets grazed down. And that's what drives the productivity in the ocean, and our entire planet provides half the oxygen. It's the second lung of the planet. And it's all driven by microbial processes. And we heard all about that today, and I probably missed lots of things, and sorry for that, but I think I've reached the end of my 10 minutes. So I just want to say massive thank you, amazing day, and just power to the microbes. That's all I'll say. Thank you.
Fantastic. Thank you, Willie. And just wonderful thoughts to end a wonderful day. I don't quite know what to add to that. We've done all of the thank yous I think that need to be done. There's so much going on behind the scenes. Rory and I are really just the pretty faces that have helped move this all along. And it's been an absolute pleasure. I think my personal perspective on it all is this feels like a roller coaster ride, and I want to get straight back on it. I want to go right back and watch all these again. I've learned so much. I hope you guys have too. The biggest thank you really is to all you guys at home, all around the world, for joining us. We couldn't do this without you. We wouldn't do this without you. It makes it all feel so special to know that we can all connect, share our ideas and thoughts, and then get back out there and be the voices for the ocean. So thank you all very much.
A couple more things, Helen. I think we've got to also remind folk to fill in the survey as well. Trust me to do a bit of housekeeping. Keep exploring, obviously. Keep looking out for the marine environment. Don't forget the Young Marine Biologists Club as well. And I think Kathy, who's been the absolute powerhouse behind a lot of this happening, is somewhere here and can maybe appear on screen for a moment. Is that correct? No, she's not. No, I'm being told. That's the behind the scenes magic happening. She is not here. So folks, yes, don't forget to do the survey, Young Marine Biologists Club, and don't forget to keep exploring. Thank you all very much. Thank you, Helen, and thank you, Willie.